Fine. Good evening, everyone. I welcome you all to the third day of Biochemistry Centennial celebrations. This session will include Biochemistry alumni and student talk, which will be shortly followed by a session on immunology and a session on translational biology. In the end, the session will be concluded with two plenary talks. The session, including Biochemistry alumni talk, will be chaired by Professor Jaya Bhaskaran, an honorary professor in the department. He joined the department as a faculty in 1985 with an interest in plant metabolites. His research group in the department is inclined in developing cost-effective anti-cancer and anti-thrombotic drugs using secondary metabolites of medicinal plants and their endophytic fungi. May I now request Professor Jaya Bhaskaran to kindly introduce the speaker for the Biochemistry alumni talk. Before proceeding to the talks, I would like to inform everyone that if you have any questions, you can raise your hand in the Zoom box and put your questions in the YouTube chat box. Your questions will be addressed by chair at the end of the talk.
Okay. I welcome all of you for this BC alumni talk number 3. The speaker is Dr. G. Sudarshan. Let me introduce the speaker. Dr. G. Sudarshan obtained BSc degree from Sri Bhagavan Mahavir Jain College, Bangalore, and MSc from Dayananda Sagar College of Arts, Science and Commerce, Bangalore. He joined the Department of Biochemistry, Indian Society of Science in 2007 to work for his PhD degree with Professor Anjali Karande, and obtained his PhD degree in 2012. During his stay in Professor Anjali's lab, he was involved in research focusing on targeted cancer therapy. Currently, he works as postdoctoral fellow at the Department of Genome Integrity, RNA and Cancer, Institute Curie, Paris, France. At the Institute Curie, he works on microtubules and implication on organogenesis and cell signaling. He has 27 publications to his credit. Today, he will talk to us on his work, and his presentation is entitled Insight into the Structural and Functional Role of Tubulin Post-Translational Glycosylation in Mammalian cilia and flagella. Now I invite Sudarshan to give his talk. Please.
Thank you, Professor Jaya Bhaskaran, for this very kind introduction. And I would also like to thank the organizers for giving me this opportunity to present my work on this very nice occasion celebrating the centennial year of the Biochemistry Department. Let me just share my screen. I must say it's an honor and a privilege to be sharing my work with you all today. I hope my screen is visible and I'm audible as well.
Yes, Sudarshan, you can go ahead. You can see.
Yes.
So I will talk to you about a couple of stories that have recently come out from the lab where I was involved, which includes the understanding of the role of tubulin post-translational glycylation in cilia and flagella. But before I get into the details of the work, we all know that microtubules are involved in diverse cellular functions. In eukaryotic cells, they regulate the shape and the size of the cell. They are involved in cell division, cell migration. They play an important role in regulating the functions of neurons and during their differentiation, and they are the core structures of mammalian cilia and flagella. Now, the fascinating question is, how is it that a structurally conserved polymer made up of alpha-beta tubulin heterodimers showing such functional diversity? Now, the diversity of microtubules is regulated mainly by their interaction with several microtubule-associated proteins and motor proteins, as well as their dynamic and mechanical properties. But an emerging concept that seems to play an important role in regulating the functions of microtubules is the presence of the tubulin code, which consists of two key components. First, there are multiple isotypes of tubulin that are expressed in mammals. To be precise, there are 9 alpha and 9 beta tubulin present. And also, both the alpha and the beta tubulin are a hub of multiple post-translational modifications, not just on the tubulin body, but also on the C-terminal tail. Now, in the lab, we are interested in understanding the role of two key modifications, polyglutamylation and polyglycylation, which is the enzymatic addition of either a glutamate or a glycine on the unstructured C-terminal tails of alpha and beta tubulin. Now, both these modifications are catalyzed by enzymes that belong to the family of proteins called the tubulin tyrosine ligase-like, or TTLLs. Now, these enzymes have a reaction specificity. Now, if you look at glutamylation, there are 3 enzymes that initiate the reaction, adding the first 1 or 2 glutamates, after which there are 4 enzymes that can elongate the side chain. Similarly, for glycylation, there are 2 enzymes, TTLL3 and TTLL8, that initiate the reaction, and there's 1 enzyme, TTLL10, that elongates the side chain. Now, we all know that when we talk about an enzymatic reaction, when there is a forward reaction, there is always a reverse to balance the equation. In case of glutamylation, this is brought about by the family of proteins called the cytosolic carboxypeptidases, or the CCPs, which also have a reaction specificity. Now, most of the CCPs remove the long-chain glutamates, and there's only 1 enzyme that removes the branch point glutamate. Now, in case of glycylation, so far, no such deglycylase has been identified. So the aspect of regulation for this modification is still an open question. Now, if you look at the distribution of these 2 modifications, glutamylation, which is seen in red here, is seen on most of the specialized microtubules, like in the neurons, in the centrosomes, during the cell division in the mitotic spindle and the midbody, as well as on the axonemal microtubules of mammalian cilia and flagella. Glycylation, however, intriguingly, is seen exclusively on the axonemes of cilia and flagella. Now, evolutionarily as well, if we look at the presence of these modifications in eukaryotic cells, we see that right from the unicellular biflagellate Chlamydomonas right till humans, all those species that have motile cilia in them do also have the presence of these modifications. And it's only species like yeasts and plants that do not make cilia that don't have these, which indicates to us that both these modifications seem to play an integral role in regulating the functions of cilia and flagella. And now, when I talk about cilia or flagella in humans, the first cell type that comes to mind is the sperm, which has its long tail, which is required for the cell to beat so that it can move in the female reproductive tract and go and fertilize the egg. But this is not the only cell type in mammals that have cilia or flagella. Now, shown here is the video of an ependymal cell, which is a multiciliated cell present on the ventricles of the brain, which have these cilia that are beating in coordination, which is extremely important for the movement of the cerebrospinal fluid. A similar kind of cell is found in the trachea, which is the tracheal epithelium, which is also multiciliated and is important for clearance of mucus and to prevent infection. Apart from such cells which have motile cilia, most of the epithelial cells across the different tissues, like in the kidney, colon, retina, liver, heart, etc., have what is called the primary cilium, which is a single cilium per cell, which is non-motile, but has a very important role as it plays a key role in chemosensing, mechanosensing, signal transduction, and so on. And of late, understanding the functions of both motile and primary cilia has become extremely important in the field, as disruption of either of these can lead to several disorders in almost every organ in the human body, and these disorders are collectively called ciliopathies. For us, the question is, how are tubulin modifications playing a role? Now, for that, I need to tell you about the overall structure of the cilia. And if you look at the core structure, be it the motile or the primary cilium, the core structure called the axoneme is rich in microtubules. And there are 9 doublet microtubules, which are shown in orange and green circles here, that make up the structure of the cilia. And in case of the motile cilia, the beating is governed by accessory proteins like the inner and the outer dynein arms, the radial spokes, and the Nexian dynein regulatory complex, which of course are not present in the primary cilia, and hence they're non-motile. So as I said, we in the lab are trying to understand which functions of cilia and flagella are controlled by tubulin modifications. And for this, we're using the mouse as the model system. And for today's presentation, I will talk to you about the role of tubulin glycylation. Now, glycylation was discovered in 1995, but its role is still not very clear in mammals. And an earlier postdoc in the lab, she was trying to understand at what stage during development does glycylation happen on the cilia. And for this, she looked at the ependymal cells in the brain, and these cells originate from precursor cells called the radial glial cells, which have primary cilia in them. And when she looked at the presence of the modification, am I still audible?
You're audible now. Go ahead, Sudarshan.
There's some issue with the network. Sorry for that.
Yeah.
So what Monse saw was that in case of the primary cilia, she was not able to detect any glycylation, but only when the cells differentiated to form multiciliated ependymal cells was she able to see glycylation in them, which indicated that the modification occurred much later during development. And so she wanted to know what happens to these cells if this modification is removed. For this, she did a combination of a genetic knockout of the TTLL3 combined with a knockdown of TTLL8 by using shRNA. And what she found was that when both these enzymes were absent, there was a loss of glycylation, as you can see from the immunofluorescence staining here. And this loss concomitantly led to the loss of cilia in these ependymal cells, which made her conclude that glycylation stabilizes motile cilia. Now, it was around this time that I joined Carsten's team. And what was intriguing for me was that when Monse looked at the cells, she found clear modification on the motile cilia, whereas glycylation was almost never present on primary cilia. Now, this was intriguing for me, and I wanted to know if this modification is more specific to motile cilia, or is it just that the antibody that was being used at this point was just not able to detect the modification on primary cilia. So to answer this, I just developed a new antibody against the branched glycylated peptide and recapitulated the study that Monse did, wherein I differentiated the glial cells to ependymal cells in vitro. And when I stained them with the new antibody called GlyPep1, I was able to see that the primary cilia was also getting glycylated, while the commercial antibody was only detecting glycylation when the cells became multiciliated, which told me that, yes, glycylation does happen on primary cilia. Now, to confirm this, what I did was to look at epithelial cells that do have only primary cilia. And for this, I chose 3 cell lines, the MDCK, which are dog kidney cells, RPE1, which are human retinal epithelial cells, and IMCD3, which are mouse kidney cells. Among these 3, MDCK is special that it makes long primary cilia. And it was only in this that I was able to detect glycylation even with the new antibody, while in the RP and IMCD3, I was not detecting anything. Now, this was interesting, and I thought that the presence of the modification depends on the length of the primary cilia. Now, to test this, what I did was to ciliate the MDCK cells, followed by a deciliation process, and then put them back into a ciliating process and look at the cilia when they're short and once they become much longer. And what I indeed did see was that when the cilia were shorter, I was not able to detect glycylation even in MDCK cells. And only when they became longer was the modification observed. So I went ahead to quantify this and saw that for the modification to happen on the primary cilia, the length has to be at least 3 to 6 microns. And as the length increased, so did the extent of modification, which told me that the modification depends on the length and the maturity of the primary cilia. Now, what happens if there is no modification to these cells? So to test this, again, I did the same assay as Monza did, wherein I took the MDCK cells and knocked them down for the enzyme TTLL3. And here again, like what she had seen, I saw that on the absence of this modification, the cilia became extremely short, which I quantify here, thus confirming that similar to motile cilia, even in primary cilia, glycylation seems to have a stabilizing role. Now, a physiological relevance for this was obtained in case of the retina. Now, the retina is a specialized tissue that expresses only one of the glycylating enzymes, TTLL3. And in the retina, you have the rod photoreceptors, which have a special primary cilium here called the connecting cilium, which is required to transport proteins and other components from the inner segment of the cell to the outer segment, as the outer segment does not have any protein synthesis. And this connecting cilium is rich in glycylation. Now, when in the mouse that was knocked out for TTLL3, what we observed was that with time, the length of the connecting cilium started shortening. And this shortening of the connecting cilium was coupled with mislocalization of rhodopsin, as well as apoptosis of the cells in the outer nuclear layer of the retina, both of which are indicators of retinal degeneration. And if you see here, this required a lot of time, about a year to 14 months, which indicated that absence of glycylation in the retina causes an age-dependent retinal degeneration. Now, retina, as I said, is special in that it expresses only one of the enzymes, while most of the other tissues express all the 3 glycylating enzymes. So to get an overall view of the physiological role of this modification across tissues, what I did was to go ahead and develop a mouse model that was genetically knocked out for both TTLL3 and TTLL8, with the hypothesis that removing these enzymes leads to total abrogation of this modification across the different tissues. And this is exactly what I observed when I looked at the different cells by an immunoblot, that compared to the wild type and the single knockout, in the absence of the 2 enzymes, there was no glycylation that was observed in any of the cells. However, what was quite interesting was that when I looked at the tissues like the testis by an immunofluorescence, what I observed was that though there was absence of glycylation, the cells still had their cilia flagella, like you can see with the sperm cells here with their flagellum stained for acetylated tubulin. The similar thing was observed in the brain ventricles, where there was no glycylation, but the ependymal cells still had cilia, in the tracheal epithelium, which still had the cilia, as well as in the kidney connecting tubule, where despite the absence of glycylation, the primary cilia was still present, which indicated that genetically removing the enzymes, though it leads to a loss of glycylation, it does not impact ciliogenesis. So I immediately wanted to know if it impacts the functions of these different cells. Now, among the different cells, the sperm cells gave me an interesting phenotype. Now, we all know that the sperm is required to fertilize the egg, which is an extremely challenging process. And this depends on the quality of the sperm, which is governed by 3 parameters: the sperm count, the sperm morphology, and its motility. So in the double knockout mice, when I looked at the sperm count and the morphology, I observed that neither the count nor the morphology was affected in the absence of glycylation. But when I was propagating these animals, which I do as homozygote couplings, I observed an interesting phenotype, wherein compared to the couplings that I did with the heterozygote animals, which gave me a litter size of about 8 to 9 animals per birth, which is the average for a black 6 mouse, in the absence of glycylation, this number reduced to about 5 to 6, which indicated that these mice could be subfertile. Now, to know if this subfertility was because of the sperm losing its effect, I went ahead to test this by an in vitro fertilization experiment, since in vitro fertilization is much more sensitive to changes in the quality of the sperm. And for this, I worked with Ahmed and his team at Institut Cochin in Paris. And what we indeed saw when we did the IVF is that compared to the wild type sperm, in the sperm that had no glycylation had a much lower in vitro fertilization index, indicating that these sperm did have defects in their ability to fertilize the egg. Now, this indicated that this is most likely because of the changes in their motility. So to test their motility, I then worked with Aminata and her group at Institute Koshan, where we looked at the sperm motility by using computer-assisted sperm analysis. Now, when a sperm is swimming freely in medium, it either swims in a straight line or takes a curvilinear path. And when we do the CASA analysis, it gives out numbers indicating both straight line and the curvilinear velocity, along with various other parameters. And we saw that in the absence of glycylation, both these velocities were reduced. And also what was reduced was the progressive motility of the sperm, which is an important component that people who are doing IVF look at to say if the sperm are indeed viable and fit for in vitro fertilization or not. So now, since we saw these defects in motility, we immediately wanted to know, why was this motility happening? Is it because of the way the flagella is beating, or is it just because of the way the sperm are inherently swimming in medium? So to do this, I then worked with Luis Alvarez and his team at University of Bonn in Germany, who do biophysical analysis of the sperm cells. And first, we wanted to look at the way the sperm flagella is beating. Now, for this, what we did was to look at the flagella by fixing the sperm through its head to the glass imaging chamber. And what you can see here in the video on the left is the wild type sperm, which has its characteristic whiplash beating of the flagellum. While in case of the cells that lacked glycylation, you see a biphasic beating, wherein near the head, it's quite stiff, while at the tip, it's much more flexible. Now, what we also saw was there was an increase in the curvature of the flagellum in the absence of glycylation compared to the wild type sperm. And we also determined the amplitude of the flagellar beat, which is the overall displacement of the flagellum with respect to the head. And this was also reduced in the absence of glycylation compared to the wild type sperm. And as I said, when we calculated the beat frequency, we found it biphasic, that near the head of the sperm, there was a reduction in the frequency of the flagellum, while at the tip, it was much more enhanced compared to the wild types. Now, since there was a change in the curvature of the flagellum, what Lewis and we did was to see if this is the case, how are these sperms swimming freely in medium? And when we looked at this, this is where we found the drastic phenotype, that compared to the wild type, which are swimming in a linear or a curvilinear fashion, in the absence of glycylation, these sperm were predominantly swimming in circles. And when we quantified this, we found that about 86% of the sperm were circular in their swim pattern. Well, there were about 14% that was still linear or curvilinear. And we believe that it is this 14% that is keeping these animals subfertile. Now, the question for us was, how is it that glycylation, which is a modification that's happening on the microtubules that form the flagellum, loss of this modification leading to such beating and swimming defects? What are the ultrastructural changes that are causing this? To know this, I collaborated with Gaia Pigino and her group in Dresden, where we analyzed the sperm flagellar architecture by cryo-electron tomography, where we plunge froze the sperm in liquid ethane to retain the overall axonymal proteins in their native conformation. And initially, when we looked at the arrangement of the different proteins within the axoneme, we found no major defects, whether in the wild type or in the absence of glycylation. But what we did find was defects in the conformations of the outer dyneins and the inner dynein motor proteins that are required for the flagellar beat. Now, the outer dynein proteins, which have two motor domains, regulate the frequency and the amplitude of the flagellum, while the inner dyneins regulate the waveform of the flagellum. Now, be it the outer dynein or the inner dynein, in a normal actively beating flagellum, these two motor proteins exist in two major conformations. One, which is called the pre-power stroke, where the motor domain is bound to the microtubule doublet, while the stalk is free from any binding. And once the beat cycle is complete, they shift from a pre-power stroke to a post-power stroke state, where the stalk also firmly binds to the microtubule doublet. So there is a constant binding and unbinding event, which leads to the active beating of the flagella. Now, in case of the wild type, when we looked at these two conformations, we found that the dyneins were predominantly in the pre-power stroke state, while there were some dyneins in the post-power stroke state as well. But when we looked at it more closely in the mice that lack glycylation, this is where we found that in the absence of this modification, the number of dyneins in the pre-power stroke reduced, and there was an increase in the dyneins in the post-power stroke state. What was also interesting was that there were dyneins that were in a mixed conformation, wherein one of the motor domains was in the pre-power stroke and the other was in the post-power stroke state. And this was for the outer dynein arms. And when we looked at the inner dyneins as well, we found the same result, that most of the inner dynein arm proteins were also stuck in a post-power stroke state conformation, indicating that the dyneins had lost their coordination between them, and this loss in coordination was leading to the kind of defects that we were observing with respect to the flagellar bead and the swimming patterns. And what we believe that if a similar defect was to happen in humans, it would lead to total sterility, because the human sperm is not as robust as the mouse sperm. Now, with all these studies, what we were able to conclude, giving the first ever molecular evidence for glycylation in humans, in mammals, is that this modification controls the sperm motility and hence fertility by regulating the dynein motor proteins. And this story came out as a cover story of Science in the beginning of the year. And its public health relevance was also highlighted by a number of articles that highlighted the work, not just in Europe, but across the world. Now, till now, I've spoken to you about the role of glycylation in sperm, but that's not the only modification that happens in the cilia or flagella. They're also rich in glutamylation. And there are multiple enzymes that are responsible for this modification. And what our team and other groups have shown is that if you remove a single enzyme that is responsible for glutamylation, most of these lead to male mice that are sterile. But if you look at the phenotypes, some of the enzymes affect the axonemal structure of the sperm. Some enzymes lead to total loss of spermatogenesis, while others lead to defects in motility, similar to what I showed you today for glycylation. Now, the question is, how is it that a same modification or loss of the same modification is leading to such diverse phenotypic defects? What are the mechanisms that are regulating this diversity? Now, this is something that is not yet understood and something that we want to look at in the future. But more importantly, what we also want to understand, or what I also want to understand in the future, is if these different tubulin modifications also affect the primary cilia functions. The primary cilia, as I told you, are present on most of the epithelial cells across different organs. And they are required right from the state of development for organogenesis and organ function. And in the adults, they're also playing a key role in tissue homeostasis and regeneration and repair. And this is possible because the primary cilium is a hub of different signaling and sensing pathways. Now, I give you a list of few of the signaling pathways that happen through the primary cilium, but this is not an exhaustive list. And the way this signaling or sensing process happens is because of an intrinsic trafficking process that happens within the cilium called the intraflagellar transport, which is brought about by a complex of proteins called the IFT complex. Now, these IFT complex proteins carry the cargo like the signaling molecules or the receptors or the transcription factors that localize within the cilium by taking it into the cilium through the anterograde IFT, which takes the help of the kinesin motors, which walk on the microtubules. Now, once the process is complete and the proteins have to be trafficked out of the cilium, the same IFT complex carries the cargo out of the cilia with the help of the dynein process, dynein motors through the retrograde IFT, and these dyneins also walk on the microtubules. And what has been shown by different groups that work on cilia is that if you impact the intraflagellar transport process, it can lead to several disorders of the human organs. But what is not known is whether tubulin modifications regulate this intraflagellar transport and thus functions of primary cilia. Now, this is where the future of the work with respect to tubulin modifications and the cilia and flagella in the lab and in my research work will go ahead.
Yes, so just 1 minute.
Yeah, I would like to thank all those people who have been part of this work, especially Carsten for giving me this opportunity to carry out this wonderful work in the lab, and all the lab members for their continued support during the stages of the project. The fantastic core facilities at Institute Curie, a special mention to the histology, microscopy, and the animal facility, and all our different collaborators for their help during the work. And I thank Institute Curie and the funding sources for their continued support. And I thank you for your attention, and I'm open for questions.
Sudarshan, it's a very nice talk.
Thank you.
Thank you very much. Now, his presentation is open for discussion. Anybody can raise the question or any clarification if you want, please do so.
Yeah, I can answer. Hi, Sudarshan. So a great talk. So I have two questions. So number one is that, so the C terminus, right? It's enormous modification happens. So are they doing some kind of a priming for like priming state, like this many glutamylation should happen, then the glycosylation happens, or is there any priming?
Actually, if you look at glutamylation, we don't know if there's any priming happening, but what we know is that both these modifications are in a kind of a tug of war. They compete for each other for the same sites. So we still don't know if how one enzyme or one modification is regulating the other.
Okay. So what about this tyrosinization? You know, that is also one of the modification now people are looking at in the kinetochore levels, that it is also happening at the C terminus. Do you think it is also coordinating at the sperm, like when in axonome?
I don't think, I mean, from whatever I know, till now, there is no tyrosination that is present in the cilia of flagella. They're all detyrosinated microtubules.
Okay. So my last question is a simple one. So when you say the cilia, the primary cilia, right? So the BB-some and IFT complex, they are normally connected with the CEP164. So do you think that CEP164 are recognized by these modifications? Do you think they do have some role on that?
It could be. We still don't know. There is also a possibility that the CEP164 itself could be modified, because even though we say that tubulin is the protein that is getting modified by these enzymes, we know that there are other proteins that are also substrates for these enzymes. So we are actually in the process. I'm actually now doing a project where we're trying to identify other proteins that these enzymes can modify. So there is, and the in the centrosomes, there is a high level of glutamylation that happens. So there is always a possibility that even CEP164 could be glutamylated. Okay. So once we identify this, then only we will be able to address this.
Yeah, sure. Thanks, Sudarshan. See you in April.
Bye. Yeah, see you.
Yeah. Professor Ganesh will ask.
Sudarshan, one short question for you. So yes, in the mice, whatever you see, the infertility with the glycylation problem. So do you also see in the human, so with the infertile or less fertile males, whether they have a similar defect with the glycylation, now also with the mutation with the TTL3 and things like that?
I wish I could say yes. We still don't know. The thing is, now that we know that this can happen, actually, we've been contacted by a few clinicians who wanted us to test these samples. It's just that we have not been able to do it till now. So only once we have a look at it, will I be able to say yes or no for your question.
Okay, great.
Thank you.
Are there any other question? Anybody wants to ask?
Yeah, there is a question by Aditya. Aditya, yes, you should go ahead.
And also in the chat, there are questions in the chat also.
Hello, Dr. Sudarshan. Nice talk. I have two basic questions. One is regarding the TTL specificity with the alpha beta subunits, because you said 9 subunits are involved. So does that have a specificity with TTLs? That's the first question.
Yeah, okay.
The second one is because the polyglutamylation and glycylation, because of the charges, do glycylation help in stability and glutamylation help in the mobility of these axonemes? The second one.
Okay. So to answer your first question, yes, these enzymes do have a specificity that some of them, for example, if I talk about glycylation, the TTLL3 specifically modifies beta tubulin, while TTLL8 has, though I say specifically, it's not that it does only beta tubulin, it does also a little bit of alpha, while TTLL8 predominantly does alpha tubulin. The same thing is there for the glutamylases as well. Now, whether it does alpha 1, alpha 2, beta 1, beta 2, those isotypes, we still don't know that. We are trying to do it by mass spec to identify which of the isotypes it modifies. Now, the second question is, this was about the charge, right? So what we think is the glycylation, because it is not charged, it's kind of acting as a scaffold where the dyneins can bind. While in case of glutamylation, there could be a charge repulsion as well. Also, what we believe is because the glutamylation has that negative charge, the dynein is kind of not able to interact. And so that's why when there is no glycylation, we see an increase in glutamylation as well. So this also might be impacting the way the dynein is binding to the microtubules and thus affecting the sperm motility.
So there is no specific structural change because of the glutamylation and glycylation?
No, we don't see anything.
Thank you.
You're welcome. I think there was one more question from Girdhari Lal. Maybe I could just answer.
He says that great talk. Just curious, does PTM changes in sperm also make it more immunogenic?
This is a good question. We haven't tested this. It would be interesting to test. That's a nice point. I would probably just check this out. Yeah.
Yeah, there is no other question. I thank Dr. Sudarshan for the excellent work and the excellent talk. And now I hand over to Dr. Kesava for the next question.
I think Bix can take over and then Keshav can take.
Thank you, Dr. Sudarshan, for this insightful talk. And I also thank Professor Jayabhaskaran for chairing the session. We will now move ahead with the biochemistry student or alumni talk. This session will be chaired by Dr. Keshavardhana Sanulla. Dr. Keshav is an assistant professor in the department. He has recently joined the department in year 2020. His research interests lie in studying the mechanisms involved in RNA sensing leading to cell death or inflammation during virus-host interaction. I request Dr. Keshav to kindly chair the session.
Thank you very much, Neetu, and a very good evening all, and good morning to those who joined from US and Europe. And we started with an excellent talk from Dr. Sudarshan. And now we are going to have 4 student talk sessions in which 4 of the alumni from BC is going to talk and 1 of our current student will talk. And let's start with our first speaker, Dr. Satpal Singh. Dr. Satpal Singh recently completed his PhD from biochemistry department under the supervision of Professor Jayabhaskaran in 2020. And during his PhD, he worked on identifying novel non-toxin anti-cancer secondary metabolites from taxol producing endophytic fungus. And I welcome Dr. Satpal for his talk for the Centenary series. Please go ahead with your presentation.
Hello, thank you, sir. Thank you very much. First of all, I thank all the organizers for giving me the opportunity to speak in this conference. And I'll be presenting my thesis work plus some of the other ideas which we have been exploring in the lab for some time now. I'll be presenting basically the studies on the endophytic fungi which produce the blockbuster drug, anti-cancer drug Taxol. And this drug was actually initially, is my video visible to everybody?
Satpal, you need to share your screen.
Okay, my mouse is having some trouble, so let me check it out.
Okay.
Okay, so yeah, just give me a moment, sir. My mouse is having some trouble, so I'm not able to, navigate the things on the screen. of that. So I hope now it is settled.
Yeah, now we can see your presentation. Please go to presentation mode.
Yes, let me put it on. Okay, so
Can you put it in presentation? Yeah.
so I'll be presenting the work on the production of Taxol, which is an anti-cancer drug by endophytic fungi. So we have learned some lessons because this drug is basically very predominantly used in the mainstream anti-cancer clinical use, and the sources of this drug were kind of main concern with the supply chain. So so this drug actually falls under the category of secondary metabolites. So primary metabolites are those which are required for the normal growth and development, reproduction of the organism. However, the secondary metabolites are those compounds which are not required for these regular day-to-day activities, but they are required for their competence in the enhancing their survival in the ecosystem. And they have diverse sources like plants, microbes, fungi, and they come to protect all these producing organisms from a diverse set of challenging environments and conditions in their ecosystem. And they have diverse biological activities. And in the traditional plant-based medicines, these were the drugs which used, these majorly employed these secondary metabolites, and some of these are in clinical use in the current setup. And the secondary metabolism basically is a derivative of a primary metabolism. And so basically from the primary metabolism, many compounds, they get acted upon by various specialized enzymes in the producing organisms and give rise to various category of secondary metabolites like alkaloids, terpenoids, and phenylpropenoids. So these are some of the examples of the drugs which have been derived from the plants as secondary metabolites, and they are in clinical use. For example, we have Taxol or paclitaxel, which is an anti-cancer drug, and some of its derivatives, and vinculcolides, which are produced from the periwinkle, and tropin alkaloids, tropin and others, as well as the camptothecin and its derivatives are anti-cancer drugs. So endophytes are those organisms which colonize the internal living tissues of plants without harming them. And they, if I spend some of the or most of the life cycle within a plant tissues, and they include various categories of microorganisms. And this also is basically for the mutual benefit of both the host and the endophyte. And you can see sometimes in the insert picture over here, an endophyte being stained with a lineal blue within the leaf sheaths of tall fescue plant. So fungal secondary metabolites, initially everybody might remember that in 1928, the first fungal secondary metabolite which came to limelight was the penicillin antibiotic. And following that, there have been a plethora of drugs which have been derived from the fungi for various clinical uses. So Taxol was the drug which sometimes in the late 70s and onwards had literally revolutionized the field of anti-cancer secondary metabolic drug discovery. And it was obtained initially from the Texas brevifolia bark in an NCI program to find anti-cancer drugs. And it was approved by 1992 as by FDA as an anti-cancer drug. And is also used currently in various combination therapies for several new cancers in these days. But its presence in the bark is quite very minuscule, like 0.02% by dry weight. So that supply chain becomes the main bottleneck for its continuous supply to the world. So demand is quite heavy, like around 250 kg per year, but the production is quite limited by the supply. And this led to the search for the various alternate sources like semi-synthesis or some of the plant-derived Taxol intermediates which can be converted by synthetic chemistry through semi-synthesis. So chemical synthesis, though it had been attempted sometimes back, but it's not commercially feasible. So there are still efforts worldwide underway to find the alternate sources of this drug. So the structure of the compound here, you can see it's a highly oxygenated cyclic, multicyclic compound. And there are various groups which are required for various biological activities of this compound. And presently, the major supply comes from the semi-synthesis, whereby the intermediates like diacetyl baccatin-3 or baccatin-3 are used to convert them to the Taxol. And you can see there is still quite a lot of interest in this field, as per year you get around 2,000 publications which has Taxol in their title abstract. So it basically binds in the beta tubulin part of the alpha tubulin beta tubulin dimer, and thus it blocks the progression of the metaphase to anaphase, because it doesn't allow the depolymerization of the microtubules, thereby inducing apoptosis. So these are some of the other compounds which do the same job of blocking that cell cycle by binding to tubulins. So initially, as you can see, in 1971, the first paper reporting this drug came out. Subsequently, there were a number of reports on its mechanism of action. And then 1993, the first paper reporting an endophytic fungus, Taxomyces andrini, was reported to produce this drug. And of course, its biosynthesis pathway was also characterized in the Texas plants later on. So endophytic fungi, they had been heralded as a kind of new source and a very promising source for the Taxol production. But so far, even there are more than around 150 fungi known in the literature, but commercial production is still not in sight. So that is because of a lot of challenges in terms of dealing with these enigmatic organisms. So in the lab, I'll be basically talking about the 3 fungi which we have shown to produce Taxol. So I'll be majorly focusing on Fusarium solani, which was part of my work, and this was the other work which was on the same topic in the lab. So, Taxol production by this fungus Fusarium solani was initially done by one of my seniors in the lab, Dr. Chakravarti, and he had isolated the isolated the fungus from the Texas Celebica stem cuttings, and of course later on shown to produce this fungus to produce the drug. And the drug was also characterized functionally, biologically, by its activity on the cancer cell lines, which will kind of prove that it is indeed the original comparable to the standard Taxol. So, so there is another fungus which later on was studied in the lab, which also produced Taxol, and another fungus which produced Taxol and one of its derivatives. So basically, as I was talking about the supply of the drug through microbial fermentation and other approaches, so this can be done by various approaches like either the metabolic engineering of the biosynthetic pathway, which could also be impacted by the global regulation through other epigenetic or global TF transcription factor regulators, or some of the stresses which could be applied to the culture as well as illustration by the various factors like plant hormones. So as you can see over here, there are a lot of approaches which could be used for improving the production of these microbial hosts, which could be either by co-culturing or by attempting the enzyme engineering or metabolic engineering. And for this, this was attempted in Fusarium solani, the main fungus which I worked upon. And you can see over here that the first thing was done to find the various media were supporting its production, prolific production of the drug. And as you can see over here, a media named FBM, which was flask-based media, with some of the components modified, resulted in the highest production of Taxol, baccatin-3, and total taxanes. So it is noteworthy that there are more than around 200 to 300 taxanes of different types, but Taxol is the main one. So then I studied the effect of pH optimization or stress on the Taxol and Taxol production in Fusarium solani. And as you can see over here, total taxanes were found to be somewhere in the neutral range, says neutral range, whereas the biomass was quite correlating with the same range. And as you can see over here, so there is a lot of indication for the lipid peroxidation, which was earlier reported in the Texas cell cultures. So we also studied this. So in the lower acidic range, you can see the fungus is experiencing some kind of lipid peroxidation, maybe because of the pH effect at the lower pH. And we also saw some of the other stress phenotypes, like the formation of chlamydospores in this pH range, as well as the lipids being higher in concentration in the lower pH range. and also the indication of some extensive ROS production as the pH was lowered. And then I studied the effect of cadmium on this fungus for its production. So first, I found the IC50 of the cadmium, which was 228 micromolar, and that was applied to the two different phases of cell culture, which was at the log phase, which is day 12, and day 18 at the stationary phase for 3 days. So the fungus was harvested, and we studied the production of total taxans as well as the individual taxol, baccatin 3, and
I have 1 minute more.
so as you can see over here in the log phase, there was a marked enhancement in the total taxane production, which was when challenged with the cadmium at IC50.
I have 1 minute more.
Hello, can I continue? Is it audible?
Yeah. Please conclude your talk. Just one minute.
Yeah. So we also studied the various media for the production of spores and how the spores performed in various germination media and all that, because that will be relevant for the industrial fermentation. And we found some of the media like especially the M1D medium supporting highest germination. And another group which studied another fungus in our lab, which also studied its supplementation of the salicylic acid in the liquid fungal cultures, which was shown to produce the enhanced taxol production. And at 300 micromolar of salicylic acid, you can see it also correlated with the one of the GGP gene, which was GGPPS, showing the enhancement in the taxol production, which is around 45-fold over the control. So, so these were the people which were involved in the taxol work, and some of the publications from our group. And so the pathway for taxol starts with the geranyl pyrophosphate, which is the precursor to all the diterpenoids. And the first step, which cyclizes this, this compound into taxadiene. So is the taxadiene synthase enzyme, and subsequently other enzymes come over for oxygenations and hydroxy. So then you can see that there are various reports of different pathway genes being reported from several endophytic fungi known to produce taxol. And I also cloned one of these genes, which is over here, DBAT, the partial gene. And another member from our lab, our group, also cloned the same gene from a different fungus, and also characterized the enzymatically synthesized baccatin 3 by the purified enzyme to be shown as cytotoxic towards HeLa cell lines and Hep G cell lines. And interestingly, after some years of cultivation, what I found was the original fungus stopped producing taxol, which is of course known in the literature for secondary metabolites. And there were particularly two reports which concern my fungus of interest, which is Fusarium solani. So these two reports showed presence of some extra chromosomal kind of supernumerary chromosomes, which can be dispensable for the organisms. So then I tried to kind of list it, this fungus with various plant hormones, but we couldn't succeed in restoring the production of the fungus taxol. And this brings an interesting question to us, that what are the regions of secondary metabolites in plants as well as fungi? Because the plants keep on...
Can you hear me?
Hello, sir.
Yeah, your time is up.
Okay, sir. So only, sir, 2-3 slides left. So we also found some genomic indication of the absence of the pathway genes. And as you can see over here, we also studied the tubulin gene from this fungus and its host plant, and found some of the changes before the production loss and after the production loss. And I also studied the compound at this point. The fungus was studied for the production of another novel non-taxane anti-cancer compound, which is characterized. And at present, we are focusing on putting the intermediate taxadiene into the yeast for attempting for the scale-up production of taxadiene into yeast. So these are some of the unresolved questions, like what are the exact molecular pairs in the biosynthesis of this taxol in fungi? Could it be used for effective production at the industrial scale? And why do they lose this drug over extensive periods of propagation and production? So, so I acknowledge my professor, Professor Jabaskan and Professor Patrick, for supporting me, and also the funding from various agencies as well as ISC. Thank you so much.
Thanks, Satpal. Are there any questions? If there are no questions, we'll move on to next talk. Thank you, Dr. Satpal, for your talk. And we'll move on to next speaker. Next speaker is Dr. Anujit Kumar. He's also one of our alumni. He did complete his PhD from Professor in the lab of Professor Dipankar Nandi in 2007. And he's currently an associate professor at the Manipal Institute of Regenerative Medicine. And his group currently works on understanding neural lineage specification, and especially how can you generate neuronal cells from pluripotent stem cells. I invite Dr. Anujit Kumar for the Centenary series, and please go ahead with your presentation.
Okay, so am I audible as well as the slides are visible?
Yes, yes.
Okay, thank you. First and foremost, I would like to thank the organizing committee for allowing me to share the research work which is going on in our lab. Our lab majorly focus on the different aspects which are involved in neurogenesis using human induced pluripotent stem cells as well as the embryonic stem cells. But in today's talk, I thought of giving the glimpse on the work which involved in assigning a novel function to an antioxidant enzyme. So the embryonic stem cells, which are isolated from the blastocyst, the inner cell mass of the blastocyst, has the ability to differentiate into multiple cell types, provided proper cues are given. So these cells has got immense therapeutic applications. The application of these cells can be realized only if the quintessential property of these stem cells, that is pluripotency or self-renewal, are well maintained. So several signaling pathways participate in maintaining these properties, and the major ones are the LIF-STAT3 pathway in mouse embryonic stem cell and basic FGF in human embryonic stem cells. So we would withdraw the LIF, the mouse embryonic stem cells goes for spontaneous differentiation. In a similar way, if the human ESCs are cultured in absence of basic FGF, they go for differentiation. In the process of identifying the novel STAT3 downstream targets, we identified the SOT2 antioxidant enzyme to be one of the key downstream target. So this SOT2 is also called as manganese superoxide dismutase, which is the resident of mitochondria, plays a role in dismutating the oxygen radicals. By using the luciferase assay by overexpression of constitutive STAT3 or by using the JAK inhibitor, we found that the SOT2 promoter activity is modulated. As I told in the previous slide, withdrawal of LIF makes the mouse embryonic stem cells to go for differentiation with a change in morphology. But as you could see that upon overexpression of SOT2, the morphology is regained as good as that of cells cultured in presence of LIF. Even the pluripotent gene SSCA1 expression is rescued upon overexpression of this SOT2 antioxidant enzyme. So the take-home message of these results is that SOT2 is the downstream effector of STAT3, and it is involved in the pluripotency of embryonic stem cell. Once any of the proteins which have been assigned with the function of pluripotency, the obvious question is that, does this protein play a major role in the reprogramming process? In 2006, Professor Yamanaka's group came up with a new recipe where you have the cocktail of 4 transcription factor, which can switch back or otherwise reprogram the terminally differentiated fibroblasts into embryonic stem cell-like cells, which they named it as induced pluripotent stem cells. So we also thought of joining the bandwagon and asking the question that, does SOT2 contribute anything for this reprogramming process? With permutation and combination of the Yamanaka factors, along with SOT2, we found that just 2 factors, that is OCT4 and SOT2, was sufficient to generate the colonies from embryonic fibroblasts. Upon characterizing these colonies, we are surprised to see that they lack the expression of pluripotent genes. That means they are no more iPSCs. So we went ahead in further characterization whether they represent any of the three derm layers, that's ectoderm, mesoderm, or endoderm. And in that process, we found that these cells expressed the ectoderm neural genes. And you could see that the ZIC1, which is a pan-neural marker, is highly expressed in these cells compared to that of the starting cell material. Upon global gene expression analysis, we also found that approximately 66% of the genes overlapped with that of these neural progenitor cells, which was obtained from fibroblasts. So a combination of these two factors was sufficient to drive the fibroblast towards that of neural identity. To further authenticate, indeed these are neural progenitor cells, we differentiated these cells to get the mature neurons in vitro as well as in vivo. And in vitro, these cells express all the mature neural markers upon differentiation, as well as GFP labeled these progenitors when was transplanted in the cortex of the mice, they further matured, saying that these cells are probably the neural stem cells or neural progenitor cells. So the SORT2 has the ability to incline the fibroblast towards the neural progenitors. So if the SORT2 has this proclivity, we wanted to further corroborate. Indeed, yes, SORT2 has the neural inclination. So we took up the mouse embryonic stem cell model system and differentiated to all the three derm layers in presence and absence of SORT2. And in the absence of SORT2, as was observed before, only in ectoderm neural genes there was compromised expression, but not in the ectoderm or the mesoderm. Even upon the gain of function, when the SORT2 was overexpressed, there was enhanced expression of neural genes. So it confirmed that, okay, indeed, the SORT2 has the proclivity towards the neural lineage. If that is the case, then what is the temporal requirement of this SORT2 during neurogenesis? So we generated the inducible overexpression as well as shRNA construct and found that it is only during early neural genesis where the SORT2 requirement is there, but not during the late neurogenesis. MAP2 is one of the late neural marker where it was unmodulated. Even with shRNA condition, we observed the same. So SORT2 modulates early neurogenesis. So we know that now SORT2 is involved in neurogenesis. So then it intrigued us to understand what could be the mechanism. So different parameters we tested like cell cycle, proliferation, ROS, because it is an antioxidant enzyme, does it play a role? But all these parameters failed to address the mechanism. During that time, SLAG group came up with a new thought process where mitochondrial dynamics dictate the mitochondria, the neuronal stages, or it gives the neuronal identity, whether it is a progenitor, whether it is a stem cell or a matured marker. So this caught our attention and we asked the same question that does then SORT2 modulate the mitochondrial dynamics, and in that process, does the neurogenesis is enhanced? So again, we sought after the knockdown as well as overexpression of SORT2 and found that whenever the SORT2 is downregulated, the mitochondrial length is reduced, as it could be seen over here. However, in the presence of SORT2 overexpression, there is increased length of the mitochondria. So the fusion process, in other way, I would say that the fusion process has been increased whenever is SORT2 overexpressed. And when you looked at the contacts of mitochondria, whether they touch each other, and that's where the fusion is increased, as upon overexpression of SORT2, we did find the mitochondrial contact is increased. And very surprisingly, as I told before, ROS didn't facilitate in addressing the mechanism. Here also, we generated a mutant SORT2 where antioxidant the activity is reduced. And we found that even the mutant SORT2 behaved like the wild type, ruling out the role of antioxidant activity of the SORT2 in this mitochondrial fusion process. So in this video, we can see, I think we will appreciate. Oh, sorry. So in this video, you can appreciate that in the control, the mitochondria is just rotating in its location, whereas when it comes to SORT2 overexpression, beautifully the mitochondria are fusing with each other and they are elongating. So confirming that probably the SOT2 is facilitating the mitochondrial fusion process. To conclusively show that SOT2 is indeed involved in mitochondrial fusion, we did a biomolecular complementation assay where we took the N-terminal leucine, the venous fluorescence, as well as the C-terminal in another cell set. So in one set, we transfected the N-terminal leucine zipper, and another set, we transfected the C-terminal leucine zipper and cultured them in presence and absence of SOT2, isolated the mitochondria and did an in vitro fusion process. Individually, these constructs failed to express the fluorescence, but upon combination of the both N-terminal and C-terminal, you could see the fluorescence happening. So with this approach, when we looked at the SOT2 overexpression, indeed facilitated the mitochondrial fusion process. So with all these things, we can conclusively say that SOT2 is enhancing the mitochondrial fusion process. If this is the case, then what are the different proteins which are at the molecular basis? What are the different proteins which are associated with this process are modulated with SOT2? To address this question, okay, we did the protein analysis and found that mitofusin 1 and 2 are involved in the fusion process.
Dr. Anujit, you just have 1 more minute.
Pardon me?
You just have 1 more minute.
Okay, thank you. So specifically, MFN2 is expressed or enhanced expression in SOT2 overexpression condition. So in a similar way, when we did the MFN2 shRNA, but not in MFN1 shRNA, the overexpression of SOT2 could rescue the mitochondrial length. When we did the mitochondrial length calculation or evaluation, we found the same thing where only in MFN2 shRNA condition, the wild type, the SOT2 overexpression can rescue the process. To translate this in the neurogenesis, when MFN2 shRNA was performed, there is a decreased expression of neural genes, but upon expression of SOT2, yeah, indeed there is rescue of the neurogenesis happening. So SOT2 induced neurogenesis, it operates via modulating the MFN2 mediated mitochondrial fusion process. To put all together, SOT2 enhances neurogenesis. A new function has been assigned over here. It also facilitates the transdifferentiation, and the underlying mechanism is, I would say that just one of the underlying mechanism is probably by enhancing the mitochondrial fusion via enhancing the MFN2 expression. With this, I will conclude my talk, and I would like to acknowledge the funding agencies, that is Department of Biotechnology and ICMR, where this project was run by, and also Mahe supported with the intramural fund. I would like to thank Professor DPN Lab and the past members especially. I would like to thank, these are Preeti as well as Smita are my students who are in the driving force of this project. Preeti is my first PhD student who is doing her postdoc at University College London. And also I would like to thank Professor McBride, from whom we got all these mitochondrial constructs. A special thanks to Dr. Jyoti Prasanna, who is the founding member of MIRM. And also happy to say that she is the alumni of IASC and also my senior in Professor DPN's lab. She majorly works with the immunomodulatory function of stem cells, where she sees the transition of macrophages from M0, M1, and M2 with stem cell, how it regulates it. And also she works on wound healing, how stem cell modulates the wound healing. Before I conclude the talk, I would like to thank my past teachers as well as the institutes which made me to be in this place today. I did my basic education at Sri Satyasai Higher Secondary School, Puttaparthi. Then had my post-graduation in biochemistry from JJMC Medical College, Davangere. Then I got graduated from ISC from DPN's lab. Then had a stint of postdoc from in Professor Catherine Werfel's lab from KU Leuven, Belgium. And all the mentors, because of their effort, I could find my place in MIRM Institute, the institute which is dedicated exclusively for the stem cell research. Thank you very much, one and all, and please feel free to ask if you have any queries.
Thanks, Dr. Anujit. It was a nice talk. Are there any questions? Yeah, Dr. Anujit, I have a question for you.
Yeah.
Yeah, so you mentioned that when you use ARC4 and SAR2, you get these neuronal progenitor cells.
Yeah.
But it actually test them whether they secrete neurotransmitters and they do behave like neural cells with respect to electrophysiology and with respect to neurotransmitters.
No, with electrophysiology, we couldn't do it at this juncture, but we are in the process of doing it. And also this is the mouse, and we are also translating it to the human fibroblast and asking similar question, whether with human fibroblast also similar thing happens. But at present, yes, the electrophysiology study is lacking.
Okay, that's great. If there are no questions, thank you, Dr. Anjit, for participating. Let's move on to the next speaker. The next speaker is Dr. Mridula Nambiar. So Dr. Mridula is also our alumni, and she did her PhD from Professor Satish Raghavan's lab, and she completed her PhD in 2011. Currently, she is an assistant professor at IISc Pune, and her lab is working on understanding the molecular mechanisms of chromosomal segregation during cell division. So I welcome Dr. Mridula, and we would like to hear your talk.
Thanks, Keshav. So at the outset, I'd like to thank the organizers for giving me this wonderful opportunity to present my work in this meeting. And so my talk today is going to primarily focus on my postdoc work on understanding the repression of recombination at centromeres during meiosis, followed by a brief introduction to the kind of work that we're doing in the lab currently at IISc Pune. So the model system that we use in the lab is fission yeast, Saccharomyces pombe. And once meiosis is finished, the 4 haploid products can be visualized encapsulated within asci. So recombination in pombe and in other organisms begin with the generation of double-strand breaks. In pombe specifically, we have meiotic-specific cohesin subunits which get loaded onto the chromosome after replication is completed. And this is followed by recruitment of linear relevant proteins, which are orthologs of synaptonemal complex proteins present in other organisms. Once the double-strand breaks are generated, they are repaired via homology-mediated recombination, where crossovers are a predominant intermediate. And these crossovers are important for 2 reasons. They are crucial to generate genetic diversity in the daughter cells. And this is because they are involved in exchanging information between the 2 parental chromosomes. And another important function is for accurate distribution of chromosomes, that is segregation. This happens because crossovers are the only physical links that connect the 2 paternal chromosomes during meiosis 1. And the crossovers help in the proper orientation of these paternal chromosomes and the kinetochores associated with them to allow reductional division after meiosis 1. And meiosis 2 is essentially equational division, which gives rise to 4 haploid products. Now, crossovers are not uniformly distributed across chromosomes. It has been found across species that centromeres are specifically avoided around the centromeres. And this happens because there's increased correlation of missegregation events associated with centromere-specific crossovers. So some of the questions that have been in the field broadly have been to understand the mechanism of repression of these crossovers at centromeres, and also how exactly do these crossovers result in chromosomal missegregation. So fission yeast is an excellent model to understand this because centromeres here are complex and they have heterochromatin. So we have a central kinetochore binding region just flanked by repetitive sequences which are organized into the heterochromatin. Heterochromatin, as we all know, are marked by H3K9 methylation. To which proteins which have a particular chromodomain are associated. In pombe, the orthologs of HP1 are SY6 and SHIP2. And one of the functions of SY6 is to load cohesins in this region during meiosis and even otherwise. So just to take you through the steps that are involved in meiotic DSB formation, once Rec8, Rec11 containing cohesins are loaded, Rec11 gets phosphorylated by a kinase, which helps or is essential in the recruitment of these linear element proteins. Rec10 is sort of an activator protein, which is needed to activate the SPO11 complex, which is the main DSB-inducing protein during meiosis. An important note here is that there is a differential distribution of these cohesin complexes within the chromosomes. In mitosis, both in the chromosomal arms and pericentric region, the composition of this cohesin is the same, which means that the two ring-shaped proteins, SMC1, SMC3, are brought together by the glycine subunit RAD21, and this is associated with PSC3. However, in meiosis, two changes happen. First, RAD21 gets replaced by a meiosis-specific subunit called REC8 in the pericentric region specifically. In the arms, in addition to this REC8 switch, PSC3 also gets replaced by the meiosis-specific paralog REC11, and then this is needed to initiate a double-strand break formation pathway. So when we started to sort of look at the mechanism of how this repression occurs, we explored a lot of different options, but I'm going to talk about specifically the one which forms the basis of the model that we have currently, and that is absence of certain activating proteins for DSP formation. So as I mentioned before, REC10 is an activator for the SPO11 complex, and REC10 gets recruited because of the presence of REC11 in the centromeric region. So we hypothesized that what if the levels of REC10 itself are low in the pericentric repeats, and we confirmed this using a chip analysis. So to genetically test this out, what we did was to specifically target REC10 to the pericentric repeats by using a chromodomain fusion, which because of its affinity to the H-taken methyl marks would get enriched in the heterochromatin regions, thereby activating SPO11. And when we verified this using genetic assays, we tried to measure the recombination across centromere 3 in POMB, which is the longest centromere. And we found that indeed, in strains which had REC10 now targeted specifically to the heterochromatin, there was a remarkable increase in the recombination frequency. And this went down when we used a REC10 inactivating mutation, or as well as a CD inactivating mutation, which no longer binds to H-taken methyl marks. And also physically, when we look at the double-strand break formation in these strains, we see that presence of REC10 at the centromeres helps in generating sense-specific double-strand breaks. So this definitely tells us that REC10 is a limiting factor, but then how is REC10 actively excluded from the pericentric region? So we went back to the protein which recruits REC10, which is REC11. And when we look at the levels of REC11 at the centromere, we see that the levels are much lower as compared to the chromosomal arms. And this was independent of SY6. SY6, if you recall, is the protein which is essential for depositing cohesins in the first place. So we see that if you remove SY6, REC11 levels do not automatically go up. So when we had to show or sort of prove our hypothesis, we went back to our CD fusion strategy and now deliberately targeted REC8, REC11, which is the pro-recombinogenic cohesin, so as to say. And once these are now in the pericentric region, when we look at the recombination levels, there is now a remarkable increase in recombination across centromeres. So this suggests that the mere switching of these cohesin subunits between the arms and the centromeric region is sufficient to activate recombination at the centromeric region. So which brings me to the model. So SY6 is the central player in this regulation because SY6, as I mentioned, is needed to deposit cohesins in the first place. But then when it does that, it sort of creates a bias because it's depositing PSC3 specific cohesin complex, thereby inadvertently excluding the REC11 containing complex, which is more pro-recombinogenic. Thereby, we don't see any breaks or even recombination events happening at the centromeres. So this model definitely helps us resolve the problem of spatial crossover control during meiosis, but we also think that it sort of addresses a bigger question, which is why species like POMB and even mammals have multiple cohesin complexes in the first place. So this is something that we are currently focusing in the lab. We are trying to understand it at a mechanistic level, how these multiple cohesin complexes are either formed, loaded differentially across the chromosomes, as well as the conservation of function with respect to the mitotic and the meiotic paralogs. But in the interest of time, I just want to talk about our sort of efforts to understand the problem of how exactly do these pericentric crossovers result in misaggregation. So for this, Sneha, who was a MS thesis student in the lab, she first just started off to look at the effects of centromeric crossovers on spore formation. So a simple propydium iodide staining of the asci in these different genotypes showed that when you have centromere-specific recombination, because we have now the rect NCD allele here in the strain, we see that these strains are highly defective in spore formation. And we see most of them are either 2-spore viable or 3-spore viable, and these were confirmed by tetrad dissection as well. To further sort of understand the type of segregation errors that can happen when we have centromeric crossovers, we are using a fluorescence-based assay, which will sort of help us to visualize these segregation errors. So what we do here is use 2 different fluorophores, which are under spore autonomous promoters, and they are tagged to the centromeric, to CEN1 specifically. And upon meiosis, now we can actually visualize these spore products because of the chromatid or the parent that they inherit. And this assay is very useful for us to bin the different segregation errors and sort of understand the source of these errors. So are these errors generated in meiosis 1 or meiosis 2? So when Sneha did this assay using the 3 different strains that we have been analyzing, what we see is that there is a remarkable increase in events which are happening because of M1 non-disjunction, which means that the 2 homologous chromosomes are not separating during meiosis 1 itself. And those form the majority of the segregation error types that we see after meiosis in these specific strains. We also see a significant proportion of missegregation that occur because of the premature separation of cohesins of the sister chromatids itself. So what we think is that this assay has definitely given us a lot to think about, and it forms the basis of sort of understanding what are the different types of segregation errors that can arise because of centromere specific crossovers. And so currently in the lab, one of my graduate students is sort of fine-tuning this process. She's using a LAC-I-LACO based system to specifically target breaks within the centromere. That is either in the centromere.
We have one minute left.
Sure, I'm almost done.
Yeah.
So, using this LAC-I-LACO system, we are going to specifically generate breaks under the central region, which is where the kinetochore binds, and also when the pericentric repeats. And we think that this sort of differentiation is going to help us narrow down sort of the mechanistic, on a mechanistic basis as to how crossovers can impede proper segregation during meiosis. And this assay, we have used it in a locus and chromosomal arms, and we see that we can very nicely visualize double-strand breaks. And I think the most crucial one is to sort of understand recombination intermediates in a temporal manner using this assay. So with this, I'd like to conclude by thanking, especially for this work, my postdoc mentor, Professor Jerry Smith, and all my lab members. And here at ICER Pune, Sneha was the MS thesis student who recently graduated, and hers is the work that I predominantly talked about. And a special thanks to Chinmay and Nikita, who were instrumental in helping me set up the lab during the very tough pandemic initial phase, because I just joined the institute right before the pandemic hit. And I also acknowledge the funding sources, both from my postdoc as well as here at ICER Pune. So thank you, and I'll be happy to take questions.
It was nice talk, Dr. Mridula. Session is open for questions.
Yeah. Mridula, so you mentioned that you presented with the centromeric recombination events of segregation errors are happening. So is it that aneuploidy sort of a thing, or what exactly segregation error means?
Right, so segregation error results in aneuploidy. So the ultimate end product that we see is aneuploid chromosomes. Okay. So we're trying to now understand at what level is this segregation error happening?
That is my next question. So, right. Is it the problem in the kinetochore assembly, and then other, all that secondary events are subsequent?
Right. There are a lot of things happening. So right now, as I just mentioned, we have a hint that most of the errors that we see are happening at M1 itself. So now we are sort of systematically going to now figure out, is it the kinetochore assembly or the orientation of the kinetochore itself? Or is it just a simple resolution defect? Because the pericentric regions are rich in cohesins, and we know that cohesin removal is essential to resolve these, chiasma. So that could potentially be a mechanistic basis for this. So we are trying to figure that out.
Yeah, you can try to look at by chip analysis with assembly of all these kinetochore proteins, correct. Then assembly.
Yes.
So from there onwards, you can try to look for,
Right,
what
right.
could
So
be the defect. Yeah.
exactly. Yeah. So that's in the works now.
Yeah. Okay. Great.
Yeah, thanks.
We have one more question.
Hi, Mridula. It was a great talk.
Hi.
So I have one question. So the SPY6, it's tend to be phosphorylated by casein kinase 2, right? So that's known that phosphorylation affects the heterochromatin structure and so on. Right. So do you think that this will also dismantle the complex of the REC11 and the 10, this phosphorylation? Has anyone looked at it, or?
No one has looked at it so far, and that's exactly some of the things that we are doing currently, because SPY6 also gets phosphorylated by DDK kinases. So we're looking at the role of these different kinases in ultimately cohesion deposition, and probably deciding this switch between whether to load a PSC3 or a REC11 containing complex.
Yeah, sure. Thanks.
Thank you.
Thank you, Mridula, for having you here, and we hope to see you here physically soon.
Sure, I hope so too. Thank you.
Yeah, bye. Let's move on to the next speaker. Next speaker is Dr. Kumar Samajit. He is currently an assistant professor at the Functional Genomics and Metabolic Research Unit at the University of Southern Denmark. He is also an alumni of the department. He completed his PhD in Professor Ganesh Nagaraju's lab recently, and he completed a postdoc in Denmark, and now he has his independent group over there. So his group is studying especially interconnecting mechanisms of mammalian cell replication, repair, and metabolism, and with the goal of defining how mammalian cells respond to intrinsic stress responses. So I welcome Dr. Kumar. He was also my batchmate in PhD. I'm happy to have you here. Please go ahead.
Thank you, Keshav. Can you hear me?
You're good.
Yes. Yeah. Okay. Yeah. Thanks a lot. It's great that I see you. So that's great. And also, I'm very happy that I got this opportunity to talk, and more so, that after one of my favorite senior, Mridula. So yeah. So I also think that it's a great opportunity for me to introduce our group. So we are a new research team. We just started this August at the University of Southern Denmark, which is in Odense, very close to Copenhagen. And here what we want to do is that to understand, to carry out cell biology of genome maintenance. So we are interested in understanding how human replisome function, but not in isolation, how they talk to natural changes in metabolic cues. And then, how they shape the pace of cell cycle, as well as the extent and the quality of DNA repair. And for this, we use cell culture model system and combine them with advanced light imaging. And then, keeping this conceptual framework, we ask specific question that how cancer context or early development, metabolic plasticity can impact on ongoing genome duplication as well as cell cycle. So why this is important? Why is it important to understand DNA replication per se? And then it is beautifully summed up by a very provocative, still debatable paper from Bert Vogelstein, suggesting that actually 2/3 of all cancers are due to bad luck during DNA replication. Unlike what we thought that it is either hereditary or environmental factor. So it is important to understand how we correct these errors and then prevent the disease such as cancer. On a second thought, it is not very surprising, notion that cancer, the replication is a cause of cancer, because when we replicate, our replication folks encounter plethora of problems. I'm not going to detail them. Probably better than me. But the good news is that we have a response mechanism, that's called replication stress response, that protects our genome, fighting against these problems, including limiting components to collision, with transcription, or dealing with the secondary structures. And one of the key, player in this response is RAD51 protein. And then, just that, I could have picked other stories to present today, but, I'm very happy to talk to you about our latest work, which we just published this year. And this also fits today, in today's context, because here in this project, I could harness all the training that I actually got from a Nagaraju lab when I was a PhD student. So what is RAD51? So as Mridula said that, during meiotic recombinations, the different players are involved, but if there is a problem, if there is a troubled DNA structure found in somatic cell, RAD51 is your friend. It can sort out sister chromatid to search for the right sequence which you have lost if you have a double-stranded break, but it can also stabilize replication forks which are stressed, cannot move on, and then it can also use catalytic power. It has this ATPase activity that can remodel replication structures for their restart, as well as can help repair bypassed lesions which arise if you have a template damage, for example. So all these is good, but where's the knowledge gap? So the knowledge gap lies in these discoveries itself. We know how RAD51 function. We know that it chooses different substrates, but all these knowledges are derived from using high loads of DNA damage, replication inhibitors, double-stranded breaks, and so on and so forth. So we decided to carefully look the role, and pursue, what RAD51 does in unperturbed, context. And for this, we needed a technology which will allow us to thoroughly scan RAD51 in live cell, as well as during a cell cycle. So here we use a technique we call quantitative image-based cytometry, where we do high content imaging and then automated single cell analysis, extracting multiple parameters. And here, when I say multiple parameters, the parameter was to look at spontaneously arising RAD51 foci, which is also at a biochemical equivalent of RAD51 filament. And particularly in cells which are replicating. So these are, with EDU pulses. And then, you can extract these typical QIBC plot, which look like flow cytometer plot. Each dot is a cell. And what you see here is that, rate of DNA synthesis, on a y-axis, and DNA content on an x. So you see G1, S, and G2 cell cycle. And on third color, what you have is a number of RAD51 foci. And these are totally untreated cells. So you can appreciate that even during normal replication, we make use of RAD51. And so there must be some sort of stress that RAD51 is dealing. And these are not just the random non-specific binding of RAD51 on chromatin. These are bona fide foci. As you take away the RAD51 mediator like BRCA2 or RAD51 paralogs, and they are gone. As you can see here, that they are totally wiped off without affecting global rate of DNA replication measured by EDU here. So now, what are they? What are these foci? So in order to look more into that, we zoomed in. We used high resolution microscopy, and to begin, we were surprised, because these RAD51 structures, what you see here, they didn't co-localize with a replizome proxy timeless, which is a replizome fork accelerator, as well as short pulses of nascent DNA. Marked by EDU again here. They are totally excluded from these replication sites. And I'm just going to cut a very long story short, that it actually turned out that these RAD51 spontaneously arising RAD51 foci are actually lying behind the fork. And then, hypothetically, one can imagine that, if a replizome has problems which it can bypass, it will bypass with leaving some gaps behind, and those will be coated by single-stranded DNA binding protein like RPA, and which will be replaced by RAD51 through the help of RAD51 mediators. And the RAD51 can do what it does best. It can do template switching behind the fork and still find the right sequence, and then sort these or repair these gaps in an error-free fashion. And if it is true, then if you do not have BRCA, for example, or RAD51 foci, then you will keep replicating with these gaps, right? The gaps in the nascent DNA, and that's what actually happens. So now here is a simple technique. This is a DNA fiber technique where you stretch DNA on a glass slide, and now we combine it with S1 nuclease, which will go and find these gaps if they are in a newly synthesized DNA. And what you see here quantified in this plot is that without S1, replosome, the track length is similar as you see in a control cell. But as soon as you apply S1 nuclease to DNA fiber, you start to see fragmentation of the track length. So suggesting that indeed these foci formed through the help of Rad51 mediators, actually repairing daughter stand gaps forming behind the replication forks. So now, this is all good. It was again, robust, but still moderate. So where could it be? Where could it matter? Now that was again the next question, and then for that again we needed a context, and that could be chemotherapeutics, hormone-induced cell proliferation that we are exploring at the moment. But another thing which inspired us was actually tumor microenvironment. And when I say tumor microenvironment, I'm not an expert, but as we all know, one feature of the tumor microenvironment is the exhaustion of oxygen, and that is hypoxia. So hypoxia, as you know, shapes metabolic environment. It can provide cancer cell with the environment, with the cues that helps them to proliferate and also evolve. And it's still a mystery, that's this still we are trying to understand the Warburg effect in detail. So we thought, why not to explore hypoxia, and around 1 to 3% of the oxygen percentage is actually regarded as a natural episodes of oxygen fluctuations in all cancer types, so at least solid tumor types. So we started to shift our experimental condition from 20%, which is there in the normal regular incubators, to 1%, and we could start to see the biomarkers of hypoxia, as you see here, glucose receptor, as well as HIF, the central regulator of hypoxia response, beautifully upregulated in normal or in 1% oxygen, which was missing in 20%. So after the setting was ready, we applied these cells to we went to perform deep proteome analysis, and then we found that, as predicted, the basic hypoxia signatures were captured. This is all good, but then we found that also it targets DNA repair, it attenuates DNA repair mechanisms, and the DNA repair mechanism that it was targeting is actually HDR. So you can see it is not affecting NHEJ, while it particularly suppresses the protein level of BRCA Rad51 without touching even the replosome component. And then we needed a condition where yes, okay. So we needed a condition when we can where we can actually explore what happens if you lose BRCA but you are in a hypoxic environment. So we started to characterize 3 days hypoxia where cell cycle looks fine, so replication is still happening, and then there was no double-stranded break formation by 3 days. So it was a perfect condition to explore now daughter stand gaps. And then we turn to again S1 nuclease assay, just to again remind you that under normal oxygen concentration, if you lose BRCA, then you have these gaps, can be monitored by S1. But in hypoxia, we observed the same, but surprisingly, they were totally fragmented. It turned out that they are not just simply gap because you lose BRCA, but they are also processed by a nuclease, MRE11 here. And that then the next question was that we can the common denominator of gaps during replication is like loss of BRCA. But then on top you have activated nuclease, which is MRE11 in hypoxic condition. And again, it turned out that it was actually through ROS-ATM pathway, which processes these gaps, provide platform for TLS polymerases, which can get loaded on chromatin. Example is Paul Kappa, and they drive mutagenesis scored by HPRT-based mutagenesis assay. So in summary, want to say that when we replicate, we always make stochastic errors, and those are those create daughter stand gaps. They are in a good hand of Rad51, which sort these problems in error-free fashion. Well, the same luxury is not there when we divide in a hypoxic environment, because we do not have BRCA proteins. They leave the gaps behind, and they are overprocessed to allow fit in TLS polymerases, which can explain that why hypoxia induces high mutation burden and instability. But one thing is that you can say that they are it's an ingenious process. Cancer take good advantage of all the microenvironment, but it's also their Achilles heel, because if you take away now TLS polymerase, you have mitotic catastrophe seen here by gamma H2AX foci in mitosis. With this, want to thank Nagaraju Lab first, because it allowed me to create a platform. It helped me to he introduced me to the field, which I feel that I will continue to do, continue to pursue next 20-30 years. And want to thank Lucas Lab, which is a textbook research environment. Particularly want to thank Jerry Lucas for his support and mentorship. And now, with the similar notion to develop this environment, we are building our team, we are growing. So if you are interested in our research program, please come along and write to me. And then this is only possible because we have funding support from university as well as the Lundbeck Foundation starting grant. Thank you very much.
Yeah, it was a nice talk, Dr. Kumar. And the floor is open for question. Dr. Sudarshan, you want to go ahead first?
Yeah, hi Kumar. That's a great work. It was just a very naive question. So in your work, you're looking at when there is no BRCA2, where you're either knocked down or you've done a knockout. Do you see a similar thing when you do a point mutation, which would probably be what's happening in a tumor scenario, where you still have the protein but its function is lost?
Yes, so that's a fantastic question, and so when you talk about cancer scenario, it's only one copy that is mutated. The other copy is still functional, and it's only lost in aggressive subtypes. Yeah, so if you make a point mutant, which are typically in a C-terminus part of BRCA, which affects actually HDR thoroughly, it actually also causes gap. So this is true, and now we are trying to pursue whether it is now where if you put now a hormone induced cell proliferation pressure, whether these point mutants actually go to the same mechanism as happens in a hypoxic environment, and that will be a road towards understanding why BRCA loss or mutation leads to cancer. Yeah.
Okay, great. Thanks.
Just to extend that Sudarshan question, so in fact, in the tumor cells, you see loss of heterozygosity. You see the lose of losing the another one intact copy of the BRCA1 or BRCA2 also. So that way,
yeah.
So that is the situation.
Yeah, but that happens quite late. The first thing will be to induce mutagenesis so much, or to induce or to create heterogeneity so much that now you start to lose your key checkpoints and so on, and then you lose the second copy. So that comes quite late in cancer. Yeah, but it's that happens. LOH. Yeah.
Hi Kumar, lovely talk.
Hi.
I was just wondering about the track length data that you were showing when you put them exposed to hypoxia. In one day it goes down, and by day 3, do you see some improvement in that? I was just curious, what does that mean?
Yeah, no. So yes. So sorry, I didn't make it more simpler, So what we saw actually is that by day 3, when you do not have BRCA RAD51 pathway, you actually create gaps, but excessively processed gaps, which you can improve or you can rescue by taking away nucleases. So that what you see here is the rescue when you take away MR11 nuclease. So
I'm talking about the second
this one
in both day 1 and day 3. Yeah.
Yeah. So by day 1, you do not have so much of gaps because you still have BRCA protein intact. While in day 3, they are totally gone. So when they replicate, they keep leaving these gaps. That's why you have excessive fragmentation, excessive processing of the gaps.
Right. Thank you, Kumar.
If there are no further questions, thank you, Dr. Kumar. It's nice having you here. Yeah.
Thank you.
Let's move on to the next talk. Dr. Sarnan will lead this next session.
Thanks, Keshav. Our next speaker for the session is the last one, is Sharat B, who is a first-year student who joined Dr. Keshav Sanula's lab in January 2020. And he's been working on characterizing SARS-CoV-2 proteins that interfere with the host cell death. Sharat, are you there?
Yeah. Can you hear me?
Looking forward to your talk. Yeah, continue.
Yeah. Good evening, everyone. Firstly, I would like to thank the conveners of this centenary celebrations for giving me this opportunity to talk about the work that is going on in our lab. Our lab is interested in viruses, inflammation, and cell death. Coming to the mimicry, it has been there on Earth since a long time. Organisms try to mimic other organisms for their survival. And today I'll be talking about the mimicry that happens at the molecular level, and the viruses, the organisms that are of our interest are said to be the masters of mimicry. Coming to the regulated form of inflammatory cell death, it is an innate host immune response upon viral infection that triggers cell death. It not only eliminates the virus infected cells, but also recruits the cells of adaptive immune system that results in viral clearance, tissue repair, and also tissue homeostasis. There are multiple forms of regulated cell death, and these three are the ones that are mostly studied: the apoptosis, necroptosis, and pyroptosis. Among these, necroptosis and pyroptosis are an inflammatory form of cell death. Coming to the necroptosis, which is a form of cell death that we extensively study in our lab, it starts with sensing the virus or PAMs associated with the virus or viral genome by a sensor, resulting in the formation of a higher order functional complex called as the necrosome. This necrosome is made up of proteins called as RAPK1 and RAPK3, which are nothing but kinases. These, upon formation of necrosome, phosphorylate a downstream protein called as MLKL. Upon phosphorylation, the MLKL goes and oligomerizes on the plasma membrane, resulting in the formation of pores. And this leads to ionic imbalance, the osmotic disbalance, and culminating in a lytic form of cell death. So necrosome is a very crucial part in the necroptosis signaling pathway. And what is important in this necrosome is a motif called as RIP homotypic interaction motif, shortly called as RIM. And this contains a tetrad amino acid residue that form beta amyloid-like sheets, that helps in stacking each other's. So these proteins which have rim-like domains can interact with themselves, resulting in the formation of an amyloid-like necrosome complexes. Those are so big that we can see in TEM images. Then these viruses are shown to interfere with the necroptosis pathway. And how do they do it? This is where the molecular mimicry comes into picture. These encode for proteins that interfere with the formation of necrosome. So they can either inhibit the formation of necrosome or promote the formation of necrosome. But what these papers have shown that these viral encoded proteins hinder necroptosis. Interestingly, these viruses are DNA viruses. So they have the genome size ranging from 150 kilobases to 250 kilobases. So they have enough genetic material to encode such proteins. So with this background information, and given the fact that the SARS-CoV-2 causes a severe cytokine storm, inflammation, and acute respiratory distress syndrome, we hypothesized that SARS-CoV-2 with a 30 kb genome might encode for some molecules like this, which mimic the host proteins. And we went in search of it, and luckily we found not one, but 3 rim-like sequences that are encoded by ORF1AB non-structural proteins. And these are the rim sequence that are well characterized in the host proteins, and these are the viral encoded rim decoy proteins. Coming to coronaviruses, 7 different coronaviruses have infected humans so far, among which the SARS-CoV-1 and the MERS-CoV caused the epidemics. And SARS-CoV-2 is the reason for current pandemic. And there are other human coronaviruses like HKU1, NL63, 229E, and OC43, which have infected humans but have caused very mild symptoms, and they cause there is no disease severity as such. Coming to the coronaviruses, they are classified into alpha, beta, gamma, and delta coronaviruses based on the protein sequence encoded by ORF1AB. But when we do this phylogenetic analysis based on the rim-like sequence that we found, there are several interesting results. So one can see the human coronavirus HKU1, which is a non-pathogenic, less pathogenic coronavirus, is placed very far away from all the pathogenic coronaviruses. So this is because we have found some mutations critical for probably critical for necroptosis in rim-like sequence 1. So we did similar phylogenetic analysis in rim-like sequence 2 and rim-like sequence 3. What we can see in this phylogenetic analysis using rim-like sequence 1 is that it categorizes all the pathogenic coronaviruses at one place and the less pathogenic coronaviruses at the other place. Then we went on to look into the structure of these non-structural proteins. We used the crystal structures of SARS-CoV-1 for our understanding, and we see that the rim-like sequence 1 and rim-like sequence 2 are present on the surface of the protein, meaning that they are amenable for interaction with the other host proteins, whereas the rim-like sequence 3 is present deep buried inside. So coming to this slide again, one can see that the severity of the SARS-CoV-2 can be correlated with the presence of the rim-like sequence, which we call the rim decoy sequence 1. So with this all information, we wanted to test these proteins for their functionality. And we used the site-directed mutagenesis approach, where we have done a tetrad mutation where the rim-like sequence 1 is converted into a tetrad alanine, which is a well-characterized mutation to study the rim-containing proteins. This tetrad alanine abrogates the beta structure and hence the interaction with other proteins. We have constructed another important construct, that is by swapping mutation of rim-like sequence 1 that we have found with a less pathogenic rim-like sequence found in the less pathogenic human coronavirus HKU1. So we tested this with these constructs. First, we tried transfecting these constructs into Calu3 cells, but the transfection efficiency was much less. And so in order to have a stable expression, we used the lentiviral transduction system. And interestingly, what we found was the cells with NSP wild type constructs started dying, whereas the tetrad mutant the cells with tetrad mutant are stayed alive. And we think that these RNA viruses acts differently compared to the DNA viruses. And this is a very preliminary data that we have right now. And further, we would like to test rim-like sequences 2 and rim-like sequence 3 also. And we would also like, based on the results, we would also like to prepare viruses in our institute's own BSL-3 facility and infect hamsters and do in vivo studies, hopefully. And then we have published recently a review article that talks about the dysregulated innate immune and inflammatory responses during COVID-19 infection. And here we talk about the pyroptosis, the netosis that have been shown to cause COVID-19 severity, but the role of necroptosis in COVID-19 severity is still enigmatic. So we want to dwell into this, and we would like to ask questions how SARS-CoV-2 is sensed by a ZBP1 as ZRNA binding protein, or how is the necrosome formation happening and how the necroptosis is happening. Well, another important cell death pathway that we study in our lab is the inflammasome induced pyroptosis. Yesterday, there was a wonderful talk by the pioneer himself, the Dr. Vishwa Dikshit, who crystal clearly explained this pathway of inflammasome, and it has a sensor called NLRP3 and an adapter protein called AS, which results in the activation of caspase 1 that cleaves gasdermin D, which goes and forms spores in the cells and helps in the release of the leaderless peptides. So we also know that the bats are said to be the reservoirs, and it is from the bats that there is zoonotic transfer of SARS-CoV-2 into humans through an intermediate host. But the bats are reservoirs to like lots of viruses, and why isn't there any inflammation in the bats? So we had this question. So we went on to look into the components of inflammasome, that is the NLRP3 ASC and Gasdermin D, and also another Gasdermin called as Gasdermin E, and we found interesting results there. We analyzed the sequences and we found at critical residues there are some changes and mutations in bats that results in dampened inflammation in bats, resulting in tolerance towards the viruses. So I would like to conclude with this slide saying that as a whole picture, we would like to see how the viruses are being transmitted from the reservoir's host, and what's the reason for this zoonotic transmission, and why does this cause severe human disease? And we would like to answer these questions using cell death as our experimental approach. So we would like to use both necroptosis and pyroptosis and the inflammasome mediated pyroptosis to answer these questions. So this is our lab, and I would like to thank Dr. Keshwardhana Sanula and all my lab mates for helping me do this presentation and all the work. Thank you. I'm open for questions.
Thank you, Sharath. That was a wonderful talk. Are there any questions from audience? Okay, there's a question from Shrish Tibari. Yeah, Shrish, can you go on?
Yes, Keshav, Sharath, it was a wonderful presentation. I just want to ask you one question.
Yes.
In regard to the upcoming variants in the COVID, like the Delta variant and the Omicron variant.
Omicron variants, yes.
Yeah.
Yes.
Do we see any kind of similarity in the rim domain?
Yeah, that's a very good question. Like, we have been following up all the variants and also the other SARS-CoV-2 isolates that were from different geographical regions, and we have found that there are no such mutations in the rim domain, and we feel that rim domain is very much necessary for the survival of the virus. So I don't think there is mutation happening there, and we are following up, and it's not there.
Okay, so if anyhow we can target this rim domain, I think it could be a possibility of getting a more suitable drug target or a vaccine for this virus. I guess so.
Yeah, there are many pharmaceutical agents that target necroptosis. Necroptosis is not just in SARS-CoV-2 infection. Necroptosis has been implicated in many autoimmune disorders and several pathogen-induced diseases. So there are many pharmaceutical agents that can be tried in future. If
okay, thank you, Sharath. Thank you so much.
Yeah, is it Nikita?
Hi, Sharath. Can you hear me?
Yeah, I can hear you.
Great talk. I just had a question. Can you comment about the efficiency of the different decoy proteins based on which virus they're present in and the severeness there?
I think the efficiency depends on the conserved motifs. If you see here, there is a 4 tetrad region, amino acid region. So these red ones indicate that they are highly conserved, and the blue ones are less conserved. It can be replaced by other amino acids, and the black ones are least conserved. So it all comes down to the rim sequence is what we feel, rather than the viruses. And even during in the virus encoded proteins, you can see that there are some places where there are less conserved amino acids.
But between the different types of decoy protein present, 1, 2, and 3?
Yeah, that also depends on the sequences, and I have not shown the sequences. So it depends on the sequences is what I would like to add on.
Okay, thanks.
Yeah.
Okay, thanks, Sharath. Is there no further questions? Thank you for the talk, and I'll thank all the speakers who was in the session, both current students as well as alumni. They were all wonderful talks, and I'll now move on to the BICS to take over the next session. Thanks.
I would like to thank all the speakers for their wonderful talks and Dr. Keshav for chairing the session. We now welcome you all to the session 5 of this talk. For this session, I welcome Deeksha to introduce.
Thank you, Neetu. We now welcome you to session 5, which is focus on immunology.
The session will have two talks by Professor Sara Gaffan and Professor Ali Albedi, and the session will be chaired by Professor Dipankar Nandi. Professor Nandi joined the Department of Biochemistry in 1997. His lab focuses on microbial stress response and host response involving T-cell biology, as well as inflammatory response mediated by nitric oxide. May I now request Professor Nandi to kindly introduce the speaker.
All right. Okay. All right. Thank you, Diksha. So it's my pleasure to welcome you to the immunology session. And the current pandemic has shed light on the role of immunology, and it cannot be overemphasized. I have great pleasure in welcoming Sarah for this talk. Sarah and I did our PhD together at UC Berkeley. She was in Marian Caution's lab, which was next door. And Sarah, Doug Caution, her son, gave a talk here some time back. So it's a nice connect, yeah. So it's great to have Sarah. Sarah's made striking contributions in the roles of IL-17 in immunology, and she's been awarded the Bill Paul. Bill Paul Award in recognition of understanding of IL-17 by the International Cytokine and Interferon Society. Great pleasure to have Sarah. So I will not take any time, and over to you, Sarah.
Thank you so much. It's really wonderful to be here. Good evening to all of you and good morning to me. Let me go ahead and share my screen here. All right. All right, computer's thinking. There we go. Okay. Well, welcome. And I caught a couple of the talks beforehand, but this looks like a really exciting symposium. I'm, again, truly honored to be here. So I'm going to talk to you today about some of the work my lab's been doing on IL-17 signaling. When you work on something that's a prime number like IL-17, we tend to look for that number everywhere. So we have lots of little tokens. I'm just going to point out this corner. This is a street corner in Pittsburgh, and you know it's Pittsburgh because we have the Steelers football team in the background. That's American football. So it's the corner of Sarah and 17th. This is my current group, and we're always looking for new additions. So if anybody at the end of this talk gets excited, please feel free to send me an email and we can talk about possibilities of joining us as a postdoc. Okay, I'm going to start with a story about one of my former lab members. Rachel Bailey was an undergraduate who did a gap year in my lab working as a technician. And it's really a story about Rachel's mom. So her mother had psoriasis and psoriatic arthritis pretty much lifelong. And it was unfortunately not very well controlled by all of the available drugs that were out there until 2015. And the reason that's significant is that's when the anti-IL-17A biologic drugs were first approved. And they kind of took all of us by surprise with how well they worked in psoriasis. Rachel's mom was no exception. Her plaques cleared, she felt better, life was great. And I wish I could stop the story there. But unfortunately, not too long after, she developed recurrent oral candidiasis. This is a somewhat common side effect in this disease. I mean, it's not terribly common, but it's there. And it was fairly severe for her. That alone probably would not have made her stop the medication, but she also developed inflammatory bowel disease, which was a much more concerning issue. And I think this really illustrates in a single individual all the many aspects of this cytokine. And when we try to intervene medically, sometimes we come up with surprises in what these cytokines do. So I'm, for time, only going to talk about one aspect of the work my lab's doing, and that's on autoimmune disease. Let's put my laser pointer here. And so I'm going to talk to you today about some of our work on autoimmunity and the basic signaling mechanisms that drive this. But about half my lab also works on oral candidiasis and other oral manifestations of disease. But as I said, I really won't touch on that today. So as I alluded to, biologics to IL-17 or its receptor were approved more than 5 years ago now, and they work very well in psoriasis. They have been attempted or are being attempted in a number of other conditions where it's not quite as much of a game changer. But I would say the field is still very early in trying to understand how all of these work. I will tell you today about some preclinical work we're doing in autoimmune glomerulonephritis, so manifestations in the kidney. But more globally, my lab is really interested in signaling and downstream signaling. And these biologics, of course, block everything that the cytokine does. Our goal would be to try and understand these downstream pathways so that we could begin to intervene perhaps selectively and target pathological effects while sparing beneficial ones. And that's a little pie in the sky, but really this concept has proven to be valid with various drugs that are now very widely used that target other signaling pathways. The JAK inhibitors, for example, target the JAK-STAT pathway. It's very early days for IL-17. This is quite a different cytokine to most of the other known ones. And so that gives us a lot of areas for delving into what this cytokine does and how it does it. This journey started in 1999 when I started my own independent lab. This is pretty much what we knew at the time. We knew that cells had receptors, and we knew about one receptor subunit anyway, which had been cloned just a few years before. You could buy IL-17 from R&D Systems, and it began to be clear that the cells that respond to IL-17 tend to be non-hematopoietic, so fibroblasts, epithelial cells. And that's a concept I'll come back to later. And one or two genes were known, but that was pretty much it. And my first student, Matt Ruddy, took this on, and he talked me into doing a very cutting edge, hot experiment at the time. And that was to do an Affymetrix gene chip. And of course, our reasoning was we needed endpoints of this pathway, because if you're going to study a pathway, you have to start at the end and then work your way backwards. And so we did this and came up with a long list of genes. Among them was IL-6, which was one of the few known targets for IL-17 at the time. So that was reassuring. And we also came up with a lot of other genes. And frankly, we were agnostic about what we would find, because it didn't really matter to us what the endpoints were. We thought, okay, that's just a platform for understanding this pathway. But of course, at the same time, understanding the genes that you find does start to illuminate what the physiological event significance of your favorite cytokine might be. So just a little introduction. IL-6, I'm sure this audience is pretty aware of, very pleiotropic cytokine. It does connect to Th17 differentiation. Lipocalin-2, I had not been familiar with at the time, but it's an antimicrobial protein and it targets gram-negative bacteria. But it's also a very potent marker of kidney damage, and it's considered, as I said, to be a clinical biomarker of disease. And so that was really our first hint that kidney might be important here. And we also found the cat enhancer binding protein delta. This is a basic leucine zipper transcription factor known to regulate lots of inflammatory genes. But I would say even today, we know relatively less about how this family of transcription factors is activated compared to, say, NF-kappa B or IRS. So our very first paper, this is summarized on this, published in 2004. By Matt. And essentially what he showed was that CBP delta is induced very potently by IL-17, and you need it for regulation of downstream genes, including IL-6 and lipocalin-2. And if in more modern days, when we use RNA silencing, we demonstrated, in fact, it is absolutely non-redundant for regulation of those genes. So the stories I'm going to tell you tie together these target genes, but we're really also partly focusing on how CBP delta is regulated and what any of this means physiologically. So signaling, of course, is complicated. I could have thrown up a really messy signaling diagram, but I'm sparing you that. I think when most people think about signaling, you think about recruiting adapters to a receptor. You have various events happen and you end up with transcription factors that turn on genes. And of course, that's true. But I think what sometimes does get overlooked is the fact that the RNAs that get produced are themselves subject to a lot of regulation, and it can be very complex. And so really the story doesn't end with the transcription factors. And it turns out that IL-17 is kind of a modest activator of transcription, but is a very potent activator of RNA regulation. So that's going to be really my story today. The first vignette I'll tell you is about an RNA binding protein called IMP2 and how it regulates the CAT enhancer binding proteins. It regulates both beta and delta, but I'm just going to focus on delta today. All right, so that's the punchline. How did we get there? Well, for many years, we've been dabbling with how CBP delta is regulated and coming up with nothing. And so everybody who did the projects, tried, moved on and did something else. Until Rami Bashara joined the lab a few years ago, he just became an assistant professor himself at the University of Paris. And what Rami did was he asked the question of whether maybe the RNA that encodes CBP delta is itself subject to regulation. And to do this, we prime cells to induce, or in this case, CBP delta with a different stimulus, here TNF. We stop transcription with actinomycin D and then just track expression over time to measure the decay rate. And what you can see is that in cells treated with nothing, there's a certain decay rate of CBP delta, and this is increased about 50% with IL-17. Maybe at first that sounds kind of modest, but if you do the math, that actually turns out to be sufficient to account for the pretty potent increase in CBP delta protein that we observe. And this was really very surprising to me because genes that are regulated at the level of RNA stability tend to have some very characteristic sequences, particularly in their 3' untranslated region, that determine this. And the CBP delta didn't have that. It's an interesting gene. It's a single intron gene. And as I said, it doesn't have any of the obvious sequences that might make you think it was unstable. But Rachel Bailey, one whose mom I talked about, she was working in the lab at that time, and she started on her own initiative to do some sequence analysis of the 3' untranslated region of CBP delta. And she came to me and she said, Sarah, did you realize that there are consensus sequences for RNA methylation in the CBP delta UTR? And I was embarrassed to admit that I had not appreciated the extent to which RNA is methylated. We all know DNA is methylated, very important in its regulation. But actually, there's a huge literature about methylation, and particularly on adenosine. And this is a shorthand to M6A. So there were multiple M6A consensus sites in this 3' UTR, 2 of which are very prominent. And in this biochemical pathway, methylase enzymes called writers add these methyl groups, but it's a dynamic process because there are erasers enzymes that can reverse it. And so this can determine the fate of this RNA, including in particular RNA stability and translation. And I will say at the time, most of the studies that had been done showed that methylation at M6A tended to destabilize RNAs. Okay, so we first asked, well, is CBP delta methylated? And to do that, we did what's known as an RNA immunoprecipitation, or a RIP. We take antibodies against M6A, pull down a cell lysate, and then measure the presence of the RNA of interest. And what you can see is, first as a control, IgG does not pull down any CBP delta mRNA. CBP delta is constitutively methylated. It comes down even when cells are not treated with IL-17, and substantially more so upon IL-17 treatment. CCL20 is another RNA that is induced quite potently by IL-17, but doesn't have any consensus sites for M6A. And sure enough, it didn't come down. So that was a very reassuring control. All right, so CBP delta is methylated, so what? The important question in this regulation is, what is the reader? And I'm skipping some data showing that if we block the methylase or demethylase enzymes, that does influence the output of CBP delta. So what's the reader? We did a lot of work to rule out some of the more common known readers in the YETH family for anyone aficionados out there. But what we really came across was a protein called IMP2. also known as IGF-2 mRNA binding protein 2. So I will refer that just to it as M2. And it was known actually fairly widely in the cancer field and the development field as a protein that regulates translation, as the name suggests, the IGF-2, in that case by binding to a 5' untranslated region. And as a consequence, it can control thermogenesis, and there are some genetic links to type 2 diabetes. But for us, the breakthrough really came with this beautiful paper in 2018 that demonstrated that IMP2 also binds to M6A and as a consequence can positively control RNA stability of target genes. And MYC was one of the genes that they describe in that paper. So we asked, does IMP2 have anything to do with the IL-17 pathway? And I showed it here in this volcano plot. If we knock down IMP2, and it knocks down very efficiently, we see a lot of our favorite IL-17 target genes. And for example, here's IL-6, CBP delta, lipocalin, as well as a number of others. I'll just point out CCL2 here for 1 second. It's going to come up a bit later. But not all genes were regulated this way. And we also did RIP-seq to eventually to show that there, of these genes, only a subset directly binds to IMP2. Some are indirectly regulated by virtue of control of upstream genes. So I'm going to spare you all the details, but biochemically, what we were able to demonstrate, and this is all published, is that IMP2 does in fact bind to this 3' UTR. It binds to these methyl sequences. These methylase sites are essential, and it will not bind to a non-methylated RNA. So it absolutely requires this methylation event. And it has a cofactor called HUAR that is also required for expression of CBP delta. And all of this is required to stabilize CBP delta mRNA and therefore to allow its expression. So what does any of this have to do with autoimmunity? I referred to in the beginning that we've started to get interested in autoimmune glomerulonephritis, and in part because lipocalin is such a strong IL-17 induced gene. Now, there are many forms of autoimmune kidney disease that have very different epidemiology and background, and they're in many senses very different diseases. They're characterized by different autoantibodies. However, the terminal events that happen at the level of the kidney are really quite shared among all of these conditions. So there's shared inflammatory, fibrotic events, and so forth. And this is really if we can intervene at the level of the kidney damage, then this might be a way to help ameliorate disease, even if we don't know what the autoantigen is, or if it's not possible to prevent development of the autoantibodies. And a very exciting paper implicating IL-17 in this as a possibility in AGN came out last summer from this group from Germany, who showed that in humans with autoimmune glomerulonephritis, there was significantly elevated levels of tissue memory Th17 cells. And interestingly, these TRMs are recognized, commensal pathogens, including staph, Candida albicans, and E. coli, so they're generated in response to commensal organisms, but they end up accumulating in the kidney and persisting, and therefore driving inflammation in the context of cytokines, inflammatory cytokines that would amplify a TRM17 response. And of course, since the other half of my lab works on Candida, we were very excited about this. So there is a really nice mouse model of this disease that was introduced to me by my close colleague, Partha Biswas. And what Partha's lab had shown a number of years ago was that, surprisingly, actually, is that IL-17 signaling is essential for development of AGN pathology. So the way this mouse model works is you immunize mice against rabbit antibodies. So you inject them with rabbit IgG in complete Freund's adjuvant. We then follow that with rabbit serum that targets the kidney. So it's against the glomerular basement membrane protein. And within 2 weeks, we get antibody complex formation that deposits in the glomeruli and causes pathology that actually is very similar to human disease. And among those genes are lipocalin-2 and IL-6. The terminal events that we see in this mouse AGN model are a very good reflection of human crescentic glomerulonephritis. This is the most severe form, again, that's common to all these different diseases. So using this model, Partha's group, with some help with us, had shown that the IL-17 receptor not only is a global knockout resistant to this disease, but if you delete the IL-17 receptor just in tubular renal epithelial cells using a CRE-LAC system, it's the cadherin 16 CRE, these mice are completely protected, as you can see. This is a blood urea nitrogen in a wild-type mouse, littermate control, and loss of this 17 receptor here completely protects from disease, suggesting that targeting IL-17 signaling might be a way to approach treating this condition. So what about IMP2, which I've neglected for a few slides? So IMP2, again, is in a wild type. Wild-type mice are very susceptible, but IMP2 deficient mice are largely protected. It's not 100%, it's not completely wiped out, but it's definitely reduced. And ACT1 deficient mice are basically the same as an IL-17 receptor deficiency. And this correlates beautifully with CBP delta expression. I'm showing you RNA here, but it's true at the protein level as well, and also IL-6 and lipocalin. So all of this pathway seems to be operative. Not only that, but if we this is a whole body knockout, and IMP2 has a lot of effects on development. So we asked, well, what if we delete IMP2 just after we initiate disease, which would be more of a therapeutic modality? And to do that, we crossed IMP2 flox mice to a constitutive but tamoxifen-inducible CRE so that we could delete this gene at will by administering tamoxifen. We did that after we initiated AGN, and you can see that those mice are protected very similarly to the total body knockout. So this showed that it's not a developmental problem, and moreover that agents that target IMP2 or its pathway might be beneficial here. We also looked at other models of disease, and this is a paper actually that got accepted on Friday afternoon, so hot off the press. Also work done by Rami Bashara with my colleague Mandy McGeechy, looking at the mouse model of multiple sclerosis, or EAE. And just very quickly, you can see in red the IMP2 knockout mice are completely protected from disease score, very similar to a 17 receptor knockout, whereas wild-type mice have very severe symptoms of the EAE-like disease. But interestingly, in this model, the mechanism seems to be really different. We thought it would be IL-6, and we did a lot of work to try and see if that was true, and it really, really wasn't. Turns out the major effector gene in the IMP2 pathway seems to be the chemokine CCL2, or MCP1, which of course regulates monocytes, and monocytes are defective, we found. There may be some direct effects on Th17, but the effects are probably more indirect through monocyte activation of this pathway. And all of this accumulates to regulate RNA stability, including of CCL2 mRNA that potentiates this pathway. So in the last part of my talk, I'll tell you a second vignette. So I just showed you IMP2 regulates CBPs, but there's a lot of other RNA binding proteins in this space. So my next story is another positively acting protein called ARID5A. ARID5A, well, I'll just tell you how we got there, but the punchline is it regulates several transcription factors, including CBP beta and IKB zeta. We found this because it's induced in autoimmune kidney disease. This was done by Yang Li, very strongly induced. And we've seen it induced in other IL-17 conditions as well. So it's an RNA and DNA binding protein, a fairly unusual family. And it had been previously reported to stabilize the IL-6 message by binding to the 3' untranslated region. And of course, that made us very interested. And it had also been shown to regulate lipocalin too. So several years ago, we started working on this. This was work by Nilesh Amatia, who's now at one of the many RNA companies in Boston. And what Nilesh had shown was that ARID5A did not really affect the RNA encoding all these transcription factors that we were interested in. And I'm showing you here IKB zeta. It's a non-canonical member of the NF-κB family. You knock down ARID5A, really nothing much happens. But the protein is very strongly affected, as you can see. And he saw exactly the same thing for CBP beta. And so this suggested that ARID5A regulated not transcription or stability, RNA stability, but translation. And we have some evidence for that showing that ARID5A binds to the eukaryotic initiation factor complex, EIF4G, and seems to, therefore, it's helping to control translation. And without ARID5A, the translation is very inefficient. Again, so does any of this matter in vivo? We made CRISPR knockout mice lacking the ARID5A gene, and we've shown that these mice are also resistant to AGN. And this was a bone marrow chimera that showed that the effects are in the non-hematopoietic compartment. I'll just draw your attention to this very last bar here. This is a total knockout, and these mice have very low blood urea nitrogen compared to a wild type. And as I said, the activities are entirely in the non-hematopoietic compartment, which is exactly what we see for IL-17. And so finally, I don't know how I am for time. Hopefully I have another couple minutes. The third vignette is, I've told you about two RNA binding proteins that turn on signaling. But in autoimmunity, a big feature is how you turn it off. So I'm going to tell you a story about an RNA binding protein called regnase, which negatively regulates some of these transcription factors. And this time I'm going to really focus on kappa B zeta.
Yeah, quickly, Sarah.
Oh yeah, sorry. All right, but I'm nearly done. So a couple years ago, a really talented student in my lab, Abhishek Garg, they're all talented, had shown that this endoribonuclease degrades target RNAs. And the consequence of that was to negatively regulate IL-6, kappa B zeta, and the consequence was that lipocalin-2 got regulated because it's downstream of kappa B zeta. So does that matter in AGN? Well, this is a negative regulator, remember. So the prediction would be if you lose the restraint on IL-17, now you would see more exacerbated disease. And that's exactly what we see. So ZC3H12A is the gene, and elevated levels of regnase do indeed lead to very high levels of AGN disease. And that happens in the renal tubular epithelial cells. And I'm not showing you the data, but this seems to converge on the kappa B zeta expression, again, upstream of IL-6 and lipocalin. And so finally, of course, if you don't have regnase, that might be a good thing for an infection where IL-17 is required, and indeed that's true. So again, I'm not going to show you any data, but Klebsiella pneumonia, that's a new story that just came out from that's on bioRxiv. In our original paper, we showed that was true for Candida albicans too. So we put all this together. Generation of IL-17, at least in part from pathogenic TRMs, but probably also other innate lymphocytes, leads to production of IL-17. This drives transcription, but it also, more importantly, drives post-transcriptional events. And these have crosstalk because a lot of these RNAs encode transcription factors. And so we have an intricate interplay between these. And all of this converges on kidney damaging gene expression, either positively or negatively. And of course, conversely, the reverse would be true in protection from infection. And I think I acknowledged everyone as I went along, so I won't dwell on that. I'll just draw your attention to this conference, which we all very much hope will happen in person next year in the lovely Hawaii, the International Cytokine Society. And we have lots and lots of travel awards, so I encourage people to apply. All right, and I hope I have a few minutes for questions. I apologize if
great. Yeah. Great, great talk, Sarah. Thanks a lot. Are there any questions?
I always encourage students.
Yes, yes. All right.
Sorry, in there.
So Sarah, so maybe I can ask a quick one. So how do you, the IL-17 has so many different effects. How do you sort of put it all together? Sometimes it helps, sometimes it hurts. How do you put it all together?
Yeah, I mean, I think being aware of what the side effects are is really the most important thing, I think, when you're starting to intervene clinically. And all the preclinical data really pointed in the right direction. I mean, we found Candida, but the early models of DSS, that wasn't done by us, all suggested that IL-17 was protective in gut colitis. And people criticized the DSS model, but it was all there. And sure enough, once they started giving it to people, they began to have these issues. How do you go in and intervene? Well, I will say that the ARID5A knockouts, although completely protected from AGN, seem to be fine when we infect them with Candida. So very simply, if maybe targeting ARID5A rather than the entire pathway could be beneficial. Obviously, very early days.
Okay. Yeah, there's one question by Dr. Das. Please go ahead, Dr. Das.
Hi, Sarah. Actually, you said that ARD2 actually interacts with EIF4G. And so is this an interaction through HUR? Can you elaborate on how is the interaction?
Yeah, we're still trying to really understand all of the players that are in there. So we haven't looked too closely at who are with ARID5A, but I think we need to. What we're trying to determine is, are the interactions RNA dependent between EIF4G and ARID5A? And my guess is they will be, but we're still trying to understand that. So yeah, we're moving into the scary world of RNA translation.
So in the context of infection, there is a translational downregulation? In the context of infection, EIF4G, if the interaction is affected, then the translation should downregulate, isn't it?
Well, in the absence of ARID5A, we see all of these genes of interest going down. Yeah, no, yeah, absolutely.
All right. Okay, one more question. One more question from ISC Biochemistry.
Hi, Dr. Sarah. It was fantastic talk.
Thank you.
So just have a simple question. So this IL-17 signaling also goes to TRAF6 and NF-kappa B activation.
Yeah.
And these T cells respond to multiple cytokines. Do you think once a cell respond to multiple cytokines, do they crosstalk at intracellular level and decide which cytokines should dominate the response?
Yeah. So quick reminder, and I didn't emphasize this, all the 17 doesn't, as far as we can tell, act on T cells, maybe very rarely, but all the cells we're looking at are epithelial or mesenchymal. But your question is valid, because we see the 17 synergized with lots of other cytokines, right? IL-17 is made by T cells, but acts on non-hematopoietic cells predominantly.
Okay, great.
Thanks. Great.
Yeah, thanks a lot, Sarah. Thank you so much. I know it's really early morning in Pittsburgh. Thank you so much for joining us. All right, see ya. Okay, we'll keep in touch. Thank you. Thank you so much.
Thank you.
Sure. All right. Okay, so we'll move to the next talk by Dr. Ali Al-Bedi, who's an associate professor in the Department of Immunology and Pathology at the Washington University in St. Louis. Dr. Al-Bedi did his PhD at St. Jude's and then did his postdoctoral research with Professor Rafi Ahmed at the Emory Vaccine Center in Georgia. So his research focuses on two aspects. One, defining the cellular and molecular mechanisms that regulate memory T-cell heterogeneity and fate. The second one is on understanding the rules for eliciting broadly neutralizing B-cell responses against rapidly evolving pathogens such as influenza viruses. And given the pandemic, Dr. Al-Bedi has geared his efforts towards understanding the durability of antibody and germinal center responses in response to SARS-CoV-2. Dr. Al-Bedi, it's a great pleasure to have you with us. Please go ahead.
Well, thank you very much for the invitation, and obviously it's great to be. And I was hoping to be attending in person, and hopefully at one point this will be possible. First, I enjoyed Sarah's talk, this beautiful talk, and it's really tough act to follow. So I feel like it's going to be already. I'm disadvantaged, but I will do my best here. But so before I start, I would say that I was really reading about the history of the institute and how it first started. It just really very inspiring that we are 100+ years now, and we're still remembering the efforts of those who made all the effort to make that institute a reality. And we still celebrate their names, and I feel that having the right vision and having the desire to help and make something useful for humanity makes people, even if they are not billionaires and they are not sending people to space, we still remember their names hundreds of years later, and I think this is really inspiring. So I'm really honored to be part of this celebration. So I will start by saying that these are my disclosures. They're really not related to the data I'm going to present to you today. So as you know, that these cells, when they respond to infection or vaccination with any of the antigens that are involved in that we get exposed to, they have to make two decisions. The first fate decision they make is either they become what we call a short-lived plasma cells, obviously after receiving the appropriate CD4 T cell help and recognizing the cognate antigen, then they either they differentiate into short-lived plasma cells. And those are the cells that produce all of the antibodies that are responsible for providing the early wave of protection. Those usually become a very low, they are low affinity antibodies. You can think of them, this is our early trial to really limit the replication of the virus or control the presence of the foreign antigen. But some of those clones that responding from the very early wave from the B cell, responding B cell clones, see what we call a germinal center reaction in the draining lymphoid structures. And this reaction is plays a central role in our how we develop a long-term immune memory. During this reaction, those responding B cells divide extensively and then introduce through the help of an enzyme called activation-induced deaminase, D-deaminase. That enzyme actually introduces mutations in the B cell receptor genes, and that allows the B cell receptors to be tested for those that have higher affinity. So affinity-based selection within responding B cell clones will allow us to promote the cells that are responding to vaccination or infection that have higher binding affinity to the antigen. And then the second fate decision happens at that stage because some of those cells either become a long-term high affinity memory B cells, or they become a long-lived switch now plasma cells. And those are the cells that reside in our bone marrow. So after vaccination, our goal is to produce these two subsets of cells, obviously along other effector T cells and other immune cell subsets. So having plasma cells in our bone marrow maintains our antibody levels against, even if it's a low level, but it's a persistent level of antibodies against the invading pathogen or the vaccine antigen. And having memory B cells ready and poised to fire up once you have exposure to produce more antibodies through differentiation to short-lived plasma cells or engaging in a second round of germinal centers. So what I'm going to talk to you about today is really our effort in the last almost 2 years now, looking at what happens at SARS-CoV-2 vaccination, actually a year now with vaccination. So we look at the generation of those cells, the plasma plus generation of germinal center after vaccination in humans, maturation within the germinal center, because this maturation is very important for generation of those two subsets and high affinity cells. So in humans, if you want to study all these subsets, it has been hard to do this over the last 100 years because we always sampled blood in humans because blood is the easy accessible compartment, very easy to do this in a large number of people. But in blood, what you can see early on is really the plasmablast, and at later time point you can detect the memory B cells. But the plasmablast is what really easy to access in blood. Those come very early, and those are short-lived, as I mentioned in the previous slide. But if you really want to look at lymph nodes and germinal center reaction, you need to track the draining lymph nodes and to be able to sample that. And that's what I'm going to explain how we did it. But also, if you want to know exactly the fate of responding B cells, you need to look in the bone marrow and to look at really the final stage. This is the winner clones that went to become long-lived plasma cells. So how we did this? We did a study very early in the pandemic, and my apologies, I know that this study was all about RNA vaccines, and I know that RNA vaccines are not really the only vaccines out there, and there are many vaccines, especially in India. There are lots of other kinds of vaccines that are being used, especially early in the pandemic. So all my study here is really showing the data from RNA vaccines, these, especially with Pfizer, because this was the first available in the US, and that's what we used in our study because that's what's available. So it doesn't mean that we are not interested in sponsoring other vaccine or we didn't want to study them, but it was a matter of what's available at the time. So our healthy volunteers here received 2 doses of Pfizer vaccine. Some of them, 7 people, were having already confirmed infection. All of those are healthcare workers. So those are the first people that were eligible for vaccination. Then we collected blood sample early before vaccination, and then multiple time points after vaccination. We also sampled, as I'm going to describe shortly, lymph node from those individuals. In that case, we sampled the draining lymph nodes, the axillary lymph nodes, also at multiple time points, and we are sampling the same lymph node to track the evolution of the germinal center response. And then at the end of 6 months after second vaccination, we from some individual who consented, we sampled the bone marrow, and this is to really look at the outcome of that evolution within the germinal center, looking at those long-lived plasma cells. And that's basically what be my talk today for you, showing you all of these populations and how this, even if we having the vaccine, how our immune response is getting refined over time. First, to look at as I mentioned in blood, the plasmablast. The plasmablast responses has been, this is during my time as a postdoc with Rafi, and I'm assuming that the majority of the audience know who is Rafi. And so Rafi, he's a world-class immunologist. He worked on T-cell responses, but he also worked in the last 15 years working actually more, working on B-cell responses and viral infections. And his focus was mainly plasma cells and plasmablast. And he, in his lab, before even I joined as a postdoc, his lab defined this plasmablast population in blood after influenza vaccination in humans. And you can define this population, that short-lived population, as CD27-38 double positive population. And as you can see, it quickly disappears from blood after vaccination. And this is now published 13 years ago or more. And this population also can be defined based on what we call ELISpot assay, which is very simple. You use the fact that those cells are spontaneously secreting antibodies, so you can just put them in plates that are coated with the antigen and let them incubate overnight, and you count the spots. So those, as you can see, come shortly after vaccination and disappear quickly, but they are responsible for the acute increase of antibodies. So we wanted to look at this is a population in blood that we can measure after SARS-CoV-2 vaccination. We did this with the individuals that I mentioned to you that received 2 doses. After 1 dose, this is 3 weeks after the first dose, you can detect a plasmablast in blood that are IgG secreting, specific to the spike protein, which is the protein that's expressed by the RNA. But then after 1 week after the second immunization, that population increases. But as I told you, this is short-lived, so they slowly decrease. But what we can see is that if you look at antibodies in serum against the IgG, you can see that a nice increase after first vaccination in those that never had been exposed to the virus. And then second vaccination adds another boost for that. And then you start the natural decay. But for those who are exposed before, 1 dose was enough to get them to the high level. And then second dose didn't add much, and then you have the decay. So now we know that the vaccine is inducing a robust plasmablast response. Very useful and very important for driving that early antibody response. But what about the germinal center? So germinal center, this has been the challenging part because this is not easy accessible for blood. So we worked with some wonderful radiologists here at WashU in St. Louis. And what we did is we used ultrasound to access the draining lymph nodes in the axillary region in humans. And there are many, there are up to 30 lymph nodes in that region in humans. So we had to learn the way, which ones are draining the upper arm area, which is where the deltoid muscle, where we get our intramuscular vaccine. And then within the lymph nodes, we sample only the follicular part, which is the part that has the B-cell follicles, and the center of those follicles will have the germinal center reaction here. And this is an example of one of our subjects. I guess I'll have to go for the regular. If I just go for, yes. So you can see here the needle sampling the cortex part. This is we call a pass, and then we take the needle out, and then we just take the cells for flow and for downstream applications. This is the lymph node, and this is the cortex part. So after that, the first sign you can tell there is a really robust response if the node gets much bigger, and that's the case with the RNA vaccine. As you can see, the node is really enlarged in size, very has a lot of vascularity, means it's a very active node. And that was very helpful for us to know that, okay, we are looking at the right node, obviously. But then to really look at the germinal center response, we looked by flow. And just this is an example for how the lymph node, after the regular seasonal flu vaccination, which is really non-adjuvanted, relatively weak immunization. So if you look at that, before vaccination, the germinal center reaction defined as isotype switched B cells, but also intracellular expressing BCL6 and CD38 intermediate cells. So those cells before vaccination, very low background. 2 weeks after flu vaccination, in some really good responders, you can see a nice expansion. If you look at the same population after 2 doses of Pfizer vaccine, this is the first 4 participants. You can appreciate how more impressively immunogenic that vaccine compared to our regular flu vaccine, almost 10-fold increasing the frequency of those germinal center B cells. Another thing that we knew right away that this is a massive immunization is that you can see it persisting for longer period of time compared to flu, but also we can see it in multiple lymph nodes in that region. In flu, we were lucky if we see one node responding to vaccination. Here we saw multiple nodes across the beads, and they are all firing up basically with a lot of immunization. And this work we published earlier this year was showing that surprisingly and also strikingly, that vaccination is not surprisingly produce a lot of antibodies early on because of the plasma process, but also it definitely drives a lot of maturation. And the one thing that we recently reported is that if you look in animal models, and we have been looking animal models of immunization for many years, and you never see germinal center for longer than 2 months. For example, after immunization, here in our participants, in 10 of 15 participants, we see germinal center up to 6 months after second immunization. And not only just germinal center, this is all spike binding. So we have a lot of, so this is NG-specific maturation going on for amazingly that long period of time after 2 immunization of this vaccine. And we see this in many individuals. This is just to show you the frequency that we see only in multiple lymph nodes. So 10 out of 15 individuals have that persistent germinal center reaction. So as I mentioned in the beginning, what this means for the immune response, we mean that we driving those cells to become long-lived plasma cells. So to do that, we take the plasma cell aspirate from the iliac crest here, and then we isolate plasma cells, CD138 positive cells. We put them on ELISpot assay, and then very simply, either the cell will secrete IgG, will be in blue, or IgA will be in red. If you coat the plate now with the flu vaccine or tetanus diphtheria vaccine or the spike protein, and this is done 6 months after second vaccination, we always had a lot more influenza-specific plasma cells because we have way more exposure compared to the tetanus diphtheria, compared to the spike, which is the first-time exposure for those individuals. And these are the frequencies. So in 10 out of 12 individuals, we see a nice induction of those long-lived plasma cells in the bone marrow, indicating that this is the result of this persistent germinal center reaction. So everything I mentioned to you so far is showing that even if you haven't been exposed to the virus and you got 2 immunization of this RNA vaccine, you have a nice plasma-plus response early on that drives the antibody to be very high in blood, anti-spike IgG response. And then they slowly decay because those cells start to die because they're short-lived, but then they are replaced by those long-lived plasma cells that really sustain that IgG level. And that's what I have shown you so far. But now I haven't shown you if there is any difference in quality due to this persistence of germinal center for 6 months. So to do that, we had to really dissect the B cell clones responding to vaccination and really ask the question, are we looking at an improved B cell or B cell receptors over time and improved antibody affinity over time? And then to do that, we had to really define those clones that are responding to vaccination. And then after we define them, and we know that this is a responding B cell clone, sequence the B cell receptor, and then track the evolution of that receptor over time in the germinal center and in the bone marrow. and then determine did it improve functionally as well. So to do that, we picked 8 people that we have the 3 different compartments, 3 different all the time points: blood, lymph node, and bone marrow. At the end, we did sequence the responding B cells either by single cell RNA-seq or bulk PCR analysis, wherever it's possible. And then we to be able to really definitively say that those cells, those B cells, are antigen specific without just using just a spike probe that could have some background binding. We extracted those cells either from the germinal center B cells. This is from the single cell RNA-seq data. So we know exactly what are within the lymph node, which are the germinal center based on the transcription profile of those cells. What are the germinal center B cells? What are the plasma cells within the lymph node? We call them lymph node plasma cells. And then extracted from each single cell, we have obviously we have the BCR information, the B cell receptor, as well as the transcriptional data. So we extracted the B cell receptors. We took the heavy chain, light chain from each single cell, cloned those into expression vectors, produced the corresponding monoclonal antibody from each B cell receptor, and then tested that antibody in a simple ELISA assay to make sure that we are looking at binding cells. And then verifying that those cells are antigen specific by ELISA. So, and we did this for the 8 different individuals, and we generated massive number of monoclonal antibody that are clonally distinct. So we can actually look for each individual and say, clone number 1, 2, 3, all the way till we have a representative clonal pool from each individual, and then we tested those antibodies here. But what that means is for each line here is an antibody that we verified binds to the spike protein that correspond to a B cell that we know is antigen specific. So now we have the information, the ID or the fingerprint from that particular B cell, and we can go back and track the evolution of that in those 8 individuals. So we use that information now and went back and tracked how, using single cell RNA-seq data from multiple time points for each individual, and say what happened to those PCRs, early time points, late time points. So first, but we had to, given that I told you there are multiple compartments, plasma plus we isolate from blood early on, and they are short-lived, but then germinal center can persist for 6 months, as I showed you. So we did a very simple. Now that we have the BCR information, we track the cells that share the same BCR. So they came from the same precursor, but then they obviously had a divergent fate. Some cells became plasma plus, some participate in the germinal center. So we track the fate of those cells. As a plasma plus, they have a dead end. As a germinal center, they are evolving. And you can see for the 8 different individuals, there are, if you just compare the plasma plus, there's a very small compartment from blood here. And each line here say that clone from the plasma plus correspond to a similar clone, have very similar feature in the BCR in the germinal center at multiple time points. So you can see some people have a lot of sharing, like this individuals or that individual. Some people have some little sharing in those 2 compartments. And this could be due to sampling or other technical issues. But the fact that we had some sharing between the 2 compartments allowed us to say, can we now see the level of mutations within the plasmablast and compare that to what happened to the same exact clone that now participated in the germinal center over time. So plasmablast is also 1 time point, germinal center is multiple time points. And now we can see how the evolution of germinal center accumulate more mutations, and you can see that here. So this is, here we're looking at the level of mutations in the heavy chain, which is these somatic hypermutation contribute to how mature the B cell over time. So within the plasmablast, we can see there are variation, but mostly are low mutations. This isolated 1 week after 2nd vaccination. At the same time, in the same individuals, germinal center, we still have relatively similar level of mutations. Now, if you look a week later, you can start to appreciate that the germinal center clones that are similar to the plasmablast start to accumulate more mutations, and over time it accumulates more and more. And that's exactly what we wanted the vaccine to do, to mature those cells, to respond early with a robust plasmablast, but also mature these cells in the germinal center so we have a better memory at the end. But then I mentioned to you that briefly that there is a population that we didn't appreciate before because it's only in the lymph node. We call it lymph node plasma cells. And those are the cells that we think are likely exiting the germinal center and on their way to become a long-lived plasma cell in the bone marrow. So they are slowly getting out of the germinal center and migrating to become a bone marrow plasma cell. They are different from the very early plasmablast cells that we isolate from blood that are short-lived. But if this is the case, if those cells are indeed coming from the germinal center, they should be clonally very close, not something like the germinal center and plasmablast. Those cells should be clonally very similar because they are just in the same vicinity, basically, and they are just coming out from the germinal center, but also it should share some similar level of mutations. So first, clonally, we do see that those cells, the lymph node plasma cells and the germinal center, are basically identical, clonotype-wise. So there are lots of sharing, and also mutation-wise, you can see that they are evolving together at multiple time points after second vaccination. So they are evolving together. So if those are indeed, if the lymph node plasma cells are indeed the cells that are seeding the bone marrow plasma cell, and that's the cells now that will stay, that's the compartment that will stay with us for the rest of our lives. So those could be, or most likely be, the cells that have accumulated more mutations compared to the cells that we generated early on, which is what we want. So to do that, we compared the level of mutations early on from the plasmablast compartment, looked at the different time points of the lymph node plasma cells, and then looked at the mutation levels in the bone marrow 6 months later. And as you can see here, plasmablast and blood very early on, there are some mutations started to accumulate, but on average, it's a very low level. Then you can look at the lymph node plasma cells at multiple time points after immunization in the draining lymph node. And those individuals, and you can see that the slow accumulation of mutations, and then you get to the bone marrow, and obviously the numbers there are much lower because the frequency is very low. But what's amazing, or what's very striking, is that we had basically left the best cells, drove the best cells to become a long-lived plasma cells in terms of high level of mutations, and those are the cells that established residence in the bone marrow. So just to remind you, I mentioned that most likely those antibodies coming early are coming from this population of plasmablast, and those that are at 6 months are coming from those bone marrow plasma cells, mostly derived from those cells. So in that case, although we have a decreased, so that's why I always say that we shouldn't be worried much about decreased level of, or decreased titer of antibodies early versus late, but the one thing that we should always look at is the quality of the antibodies, even at those later ones. So if this is indeed the case, that those antibodies are coming from higher mutated cells that have likely high affinity, then they should be better in quality compared to those antibodies. And to test that, and this is my last slide before the summary, that we did a very simple ELISA, but we added a urea wash that only washes antibodies that bind with low affinity. And to compare that, you can compare the early time point antibodies here at week 5 compared to 6 months. At the early time points, most of the antibodies are weakly binding. They're easily replaced by the urea wash. At 6 months from the same individuals, those antibodies are now coming, although they are less in quantity, they are much higher ability, have much higher ability to binding to the spike antigen, because they are a smaller fraction of them got replaced by urea wash, and that's what we are showing here. So in summary, I showed you that after SARS-CoV-2 vaccination, especially the mRNA vaccine that we used, we had a robust plasmablast response that led to this early increase in antibodies, but also we had this really first time to be shown in humans. We've known this from animal models for 40 years at least, that we have this maturation happening in the germinal center of B cell responses, but to see that this is happening in humans in this really strikingly long period of time, 6 months of continuous maturation, leading to seeding our bone marrow with this highly refined plasma cells that will live with us for a long time, and with a much better functional profile. So this is work not done. I didn't generate a single data set in this whole set of slides. What done by a lot of heroes in my lab, Jackson and Usab are two fantastic postdocs. Julian and Aaron are terrific specialists that really did the cloning and the tracking. Terrific clinical colleagues here in the hospital to be able to do the sampling lymph nodes, sampling bone marrow, blood, and multiple time points. Very generous volunteers, very generous funding from NIH and multiple institutes here. And we are also expanding in our lab. So if you're interested, please reach out. And I'll be happy to take any questions.
Thank you so much, Dr. Albedi. Can you please can I please ask others to unmute? Please. Okay, there's a okay, go ahead. There's someone in IISc Biochemistry who's raised their hand. Okay, yeah, go ahead. Jacob, can I please ask you to Sandeep, can you please mute? Yeah, thank you. All right. IISc Biochemistry, is there someone their hand raised? Yeah.
Hi, Ali.
Yes.
It was a fantastic talk and very happy to see you
Thank
here.
you.
And sorry for this disturbance.
No worries at all.
I just have a question on germinal centers, and you see that after natural SARS-CoV infection, there are like nice activation of germinal centers and formation of germinal center, which trigger more of high avidity antibodies. So can you comment on, is it possible to induce such germinal centers with like vaccines, and what kind of components is important in a vaccine to trigger these germinal centers?
Oh, yeah, that's a great question, especially regarding with such a new vaccine platform like the RNA, which we really don't know. We don't know in details on how exactly it does it. We don't think there's any special. We don't think there's any special requirement. Obviously, having more adjuvants or adjuvant-like activity in your vaccine antigen can drive a much better immune response and much more robust immune response. And can actually make the germinal center much more persistent. But there is no reason to think that, for example, a vector-based ad vectors will not generate also a robust immune response. Even an inactivated vaccine, whole virus inactivated, will also generate. Just we didn't have the side-by-side comparison with other vaccine platforms to really understand if there is any qualitative difference in terms of how persistent or how robust the germinal center is. Would be important to do that. Hopefully, when we get over with it, hopefully soon, we can start looking at more comparative studies to really look at that.
Yeah, thank you very much.
All right. Yeah, there's another question by Rashi, and then Girdhari has a question. Rashi, please go ahead.
Yes, sir. So one of my questions was, you observed like the lymph node size, right? So does
you need to speak up a little louder, Rashi. It's hard to hear you.
Hello, am I audible now?
Yes. Go ahead.
Yeah. So does the like the lymph node which you select for studying this particular effect affect the studies itself?
Are there any particular lymph node that we looked at? Is that the question?
Yeah, like was there a particular lymph node, or you had multiple lymph nodes that you looked at?
We looked, so the lymph nodes in the axillary region in humans, they are multiple groups. There are 5 groups of nodes. So the group that drain the upper arm, they call the lateral lymph node within the axillary region. There are, for example, there are nodes that called the pectoral cluster of nodes that drain the breast area, and there are other groups that drain the back of the neck. So the ones that we focus on, it's called the lateral group, and they are situated next to a very prominent lateral vein that those drain the deltoid muscle, and that's why we picked these nodes.
Thank you.
All right. Okay, Girdhari, go ahead.
Excellent. So fantastic talk. The question which I
Thank you.
have is that in terms of role of IL-6, which is also very interesting in COVID case, and we know that also the B cell require IL-6 for its development and differentiation. So the question is that the IL-6 receptor antibody, which is now thrown in the market or in the clinic, how relevant what is your opinion about using IL-6 receptor antibody, given the fact that is also contributing B cell development differentiation?
This is an excellent question. We have it becomes very complicated in humans. Obviously, it was used early on as a potential treatment in humans for COVID infection, and usually for severe infection. We obviously, if you do this in the context of vaccination, we know that IL-6 is important for plasma cell survival, and if you block that, you will interfere with infection. None of our participants obviously were on any of those medications, so we don't know what will happen if we had that going on. Likely we'll have really decreased the response, if not really abolished it. So we really don't have data on that. In humans and people who are infected, the problem with that is we cannot really dissect the impact of IL-6 receptor blocking versus really having individuals on ventilators, individuals responding to severe infection, having other issues. So we cannot dissect really what will be the contribution to immunity or blocking immunity to contribution to survival, which is, I thought, was hard to. We tried to study that early in the pandemic, but we couldn't really dissect the two parts together because lots of confounding factors with having someone viremic with severe infection and on ventilator and getting that IL-6 receptor treatment. So yeah, it's an important point, and I think it really needs a preclinical model to really dissect.
Okay, there are two questions by undergraduate students. Nikita and Vishal, please keep it short. Nikita, go ahead.
Hi, so I just wanted to know whether you observed any distinct gender-based differences in the study conducted and age-based.
Oh, that's a really important question. We have a low number of individuals, so we have a mix of male and females, and we didn't see anything striking, but the number is low, especially when it comes to looking at lymph nodes. We have 15 individuals, so it's not really something that we are not powered to really say something for certain. We didn't see anything striking within those 15, but obviously within all of our individuals are really in below 50 years old, especially the ones that we agreed to give a lymph node. So it will be very important in the future to study how the germinal center reaction happens and persists, and the robustness of it in individuals in the elderly age, because we know that there are intrinsic defects there. This is a very important question. We haven't studied that yet.
All right. Okay. Vishal, go ahead.
Yeah. So you said that the long-living plasmablasts play a major role in the vaccination response, and we know that this kind of they have a higher level of affinity maturation, and the antibodies produced by them bind with higher affinity. So if we consider the case of other viruses like HIV, which mutate faster, would this kind of be a compromise towards achieving higher affinity versus would it compromise on the breadth of the neutralizing?
Oh, yeah, again, it's an excellent question. We haven't. The thing is, our immune system handles what's presented to it, and it handles now those individuals that were presented with one form of spike, and they try to optimize or refine our responding B cells against that spike. But yes, there are the dogma right now in the B cell field is that the memory B cell compartment, which not the plasma cell, maintain a broader repertoire of B cells, of B cell receptors that didn't attain that much higher level of mutations compared to the plasma cell. And this, in theory, could allow those cells who could respond to future exposures to respond to a broader range of spike proteins. We are testing that right now in a way. Remember, our individuals are still participating in study. These are continuing study, and they are getting boosted with either the regular spike or spike based on the variants, or getting infected with any of those variants. So it will be interesting. We will be able to address that more robust in future studies. But it's such an important question that it will be beautiful to be, if this is the case, that really the memory B cells maintain the breadth and the plasma cells maintain the specificity. But it hasn't been shown definitively in humans yet.
Okay, thank you, Dr. Albedi. I have one, naive question. Given the fact that the mRNA vaccines are such potent antibody inducers, how do you explain the fact that Europe and US are seeing such high rates of infections?
So there are two things here. First is we have a 60% vaccination rate in the US. And so that's not 100%, and there's a huge difference. Second is, and it's similar in Europe, there are multiple different variations. Europe RNA wasn't really the main, AstraZeneca was the main, but still not really a much inferior vaccine. I think one thing that we should keep in mind in general about any vaccine is that not everyone vaccinated will have this robust response. There are a variety of factors that contribute to how robust your response to vaccination will be. I only presented a small subset of our individuals who are really healthy, relatively young, and there are really no other factors. They don't take any medication. There is no chronic conditions. There are lots of, and there is no.
Yeah, thank you.
So there could be different factors affecting how different people respond to vaccination.
All right. Okay. Thank you so much. I think this has obviously been a fascinating area and subject, and so lots of questions. I thank both the speakers, Sarah and Dr. Albedi. We'll move on to the next session now. I hand it over to the big. Thank you both. Thank you. Bye bye.
Thank you so much. Bye bye.
Thank you, Professor Geffen and Professor Albedi for the wonderful talks, and Professor Nandi for chairing the session. With this, we have come to end the session 5. Now I request Satyaranjan to kindly initiate the next session.
Thank you, Diksha. Now we'll proceed to the final session of the Centenary Conference Series. This session is focused on translational and disease biology. In the first part of this session, we'll have two interesting talks by Professor B.R. Prasad and Professor Sandeep Verma, and this part will be chaired by Professor Ganesh Nagaraju. I would like to welcome Professor Ganesh Nagaraju, who joined this department in 2007. His research interests revolve understanding DNA damage and response, and how various proteins play concerted roles in maintenance of genomic stability and tumor suppression in mammalian system. I would now request Professor Ganesh to kindly introduce the speakers.
Thank you, Satyaranjan. Welcome you all to this session. We have two speakers. The first speaker is Professor Prasad. So it's a great pleasure for me to introduce Professor Prasad. Prasad is an alumnus of our department, did his bachelor's and master's from University of Mysore, and obtained his PhD from our department with Professor M.R.S. Rao. Later, he carried out his postdoctoral studies at Fox Chase Cancer Center, Philadelphia, and Columbia University, New York. In 1990, he established his lab at Albert Einstein College of Medicine and has been working on viral determinants of HIV-associated neurocognitive disorders, effect of drugs abuse on HIV latency, beta chemokines, ESCRT factors, and viral budding. Currently, he is a professor at the Albert Einstein College of Medicine and has been serving in numerous committees. The title of his today's talk is Extrinsic Regulation of ESCRT Factors, HIV-1 Release and Replication Fitness by a B chemokine. Over to you, Prasad.
Thanks very much. I'm just going to start sharing my screen. Having some difficulty in sharing my screen. 1 second. There you go. Can you see my screen?
Yes.
Wonderful.
So you can go to the full screen mode. Yeah.
Great. Thanks very much, Ganesh, for such a nice introduction. And I would like to thank the organizers for this fantastic opportunity to speak at the centenary celebration of my department of biochemistry. So I'm going to be talking today about how the HIV-1 release and replication fitness can be regulated extrinsically by chemokines such as CCL2. And this is something really novel that we struck upon unexpectedly. Okay. Okay, I just want to begin by saying that HIV exists in multiple flavors. We have HIV-1 and HIV-2, but I'm really talking about HIV-1, which has 4 different clades, M, N, O, and P. Out of these, M is the most important and major clade. Each of these clades is, each of these groups rather, are single transmissions from monkeys to human in the form of SIV, then becoming HIV-1. So out of these 4 separate transmissions, M is the most successful because 95 to 97% of all HIV burden today is because of group M. And that is then subdivided into further clades, so A through J. And on the right, I'm showing you the proportion of burden caused by each of the clades. Notice that clade C is responsible for roughly half of the HIV burden in the world, whereas clade B, that many people study a lot because it's present in resource rich countries such as United States, Europe, and Australia, only causes about 10% of the HIV burden. And this shows where these clades are present. So for the purpose of our discussion, I want to point out the 2 clades, that is B and C. C is present in India and southern African countries and Ethiopia, whereas clade B, which is shown in green here, is present in North South America as well as Western Europe and Australia. My lab many years ago delved into the question of why is it that certain clades of HIV-1 in some parts of the world are hardly neurovirulent. For example, the HIV-1 in circulating in India, that's clade C. So we actually did a thorough investigation of this in collaboration with Dr. Uday Kumar Ranga, also in Bangalore at the Jawaharlal Nehru Center. And we published many papers both together with them and also independently in our lab. All of these pointed to the fact that there is a key mutation in the HIV-1 subtype C in one single gene, that is TAT, which seems to be responsible for recruiting macrophages to the brain and thus bringing HIV-1 to CNS causing eventually problems in cognitive aspects of the brain. So that work had been done over the years, and we were working on HIV-1 B and C from US and from India, and that led us to find a number of features that are unique to subtype C. So one of them is that the HIV-1B, upon infection of macrophages, causes induction of many inflammatory cytokines and chemokines, and the most prominent one is CCL2, which is responsible for recruiting macrophages. So you can see clearly that clade B infection of macrophages leads to significant production of CCL2, also called MCP1, whereas HIV-1C does not. Whereas the viral production in the 2 cases is more comparable than the CCL2 releases. So this got us interested in the question of what does CCL2 do to HIV-1 replication. And this is an old paper that showed that in viremic patients, you see high levels of CCL2. That raised the question, does CCL2 actually influence HIV-1 replication? And we investigated that question. So what you're seeing is a 30-day replication kinetics of HIV-1B or C. So the experiments are done in 4 conditions. You add nothing to the medium other than HIV-1 and macrophages. You see a peak at day 15. You add anti-CCL2 to the medium so that CCL2 would be neutralized. So it cannot signal through its receptor, the CCR2, then you see a tenfold inhibition of virus production. If you add excess CCL2 to the medium, there is even more virus production. And then we looked at the intracellular as well as extracellular virus. So when we looked at the extracellular virus, you see that compared to untreated, you see a significant reduction in the virus production. We actually saw no virus in the purified virus preps. Whereas intracellularly, you see accumulation of GAG polyprotein at a high level in the cases where you have added anti-CCL2. When you add CCL2, though, you see a significant small but perceptible increase in virus production. Subtype C, HIV-1 did not seem to care what we did about CCL2 levels, and there was no change in the precursor inside the cell or virus production in the medium. So then we hypothesized that this must be because of some event being blocked for the virus budding or release to occur. Virus budding, as many of you know, takes place for HIV-1, takes place at the plasma membrane. The GAG molecule, which looks like this with 4 different subproteins, accumulates at the membrane by attaching to the membrane through the MA, and then it assembles and then forms a curvature. You have a little bud neck, and this bud neck needs to be severed by a membrane fission event that is catalyzed by escort-3 proteins. And these escort-3 proteins look like spring structure. The spring-like structure then basically tightens itself, eventually allowing the virus to get out, get separated, and escort factors fall back into the cytoplasm. How does the escort-3 get recruited? Basically, it gets recruited by the P6 domain of the HIV-1 GAG. So P6 has 2 motifs that we know that can do such a function, PTAP and the LYPX. PTAP recruits this escort-1, TST101 that brings the entire complex, which then brings the escort-2 complex in brick red, which then brings this blue complex, which is escort-3. Whereas if you have the LYPX motif, then it recruits the protein called ALEX, which then can bring in the escort-3 directly. So it's well known that HIV-1b uses both of these. So we wondered if the HIV-1c does not have one of these. So we did a alignment and did a bioinformatic analysis that revealed that the PTAB was present in all of them. So here is an analysis of 700 or so B and about 500 or so C sequences. You can see that in the case of clade B, 96% of them have the LY. Here is the LY motif shown. Y is the critical of those 4 residues, whereas subtype C, 99% of them do not have that Y. In fact, we showed by using GST fusion proteins of GAG BP6 or GAG CP6 that only GAG B pulls down ALEX but not the C. So this then led us to ask the question, so what exactly is happening? So can you just have PTAP alone or LY alone? So what exactly are the requirements? So here is a series of mutants that we studied. We took clade B, which has both PTAP and LY, or clade C, which has only PTAP. We made mutants as follows. In the case of B, we used the wild type as is, and then we mutated the PTAP. We then deleted the LY, or we did both mutations. In the case of C, it only comes with one, that is PTAP. So this is a wild type. Then you delete the wild type, it becomes equivalent to the double mutant here. You can add back the LY, that becomes equivalent to wild type of the B, or you can keep the PTAP mutated and add the LY. So you have 4 combinations. So in each case, we did the transfection of HIV DNA into HeLa cells, added CCL2, which enhanced the virus production, or added anti-CCL2 that inhibited virus production. This is the control, which doesn't do anything. So without the PTAB, you still see the responsiveness the same way as this, although overall stunted, because PTAB is a major way of recruiting escort factors. But if you lose LY, there is inhibition of overall virus budding, but then there is no responsiveness whatsoever. Double mutant is dead. Whereas the wild type, subtype C, which has only PTAP, behaves pretty much like the delta LY, meaning if you don't have LY, it cannot respond by producing more virus particles. And if you add back the LY, it suddenly becomes responsive and looks identical to wild type B. If you lose the PTAP and add the LY, it still has a responsiveness. So overall, LY is needed in the virus to recruit Alex to the site. We also did the knockdown of Alex in the cell, and we find that without Alex, you see no responsiveness even for clade B. So then, what's the mechanism by by which this is happening? So is perhaps CCL2 is signaling the cell to make more Alex. So we looked for the Alex mRNA induction. There was no such induction happening. So then we looked at the protein. So there was an interesting clue here. We looked at the total soluble and pellet protein or insoluble protein under conditions of CCL2 treatment or the anti-CCL2 treatment. What we saw was that the total Alex never changed, but the soluble Alex seemed to be seemingly induced by CCL2, but it was not in induction as the total protein stayed the same. And the pellet fraction showed it was basically a mirror image of what we saw with the soluble Alex. So then we find that you can see that there's a time course induction or rather accumulation of Alex in the soluble fraction. This told us that Alex is moving from some insoluble compartment in the cell to the soluble compartment so that HIV can use the Alex protein for facilitating its budding. So here is a picture. I would like to set this up a little bit. Excuse me. So these are HeLa cells which have been treated with either CCL2, anti-CCL2, or the isotype antibody. And I want you to remember that the key to understand this is whether there is CCL2 in the medium or not. So in conditions of not treating with anything or adding CCL2 or adding isotype antibody, there is always some CCL2 because HeLa cells produce CCL2, so it's an autocrine reaction taking place all the time. But when you have an anti-CCL2 in the medium, there's going to be no CCL2 because it'll be completely neutralized. So then we look for Alex by immunofluorescence or actin because we had some clue that Alex may be present in the actin network. So you see that Alex is diffused in every case where there is CCL2 in the medium. Where there is no CCL2 in the medium, you see Alex is organized into some kind of a stress fibers and looks identical to actin stress fibers. If you merge the two, you see that everything seems co-localized very nicely. So this is a Pearson coefficient. And similar picture was seen in macrophages. I'm not going to show that. So this is our model at this point. CCL2 binds to CCR2, which is a receptor. Then there is a black box as to what happens that results in moving this little checkmark shaped Alex protein from actin compartment to the cytoplasm that then allows the recruitment of escort factors to facilitate membrane scission and the virus release. So I just want to take a small, diversion here to show you something very different. I earlier showed you how there are various subtypes of HIV-1 or clades of HIV-1 in the world and how clade C is dominant. So many people have wondered what's the basis for why clade C is so dominant in much of the world. So one hypothesis was that clade C somehow has an increased fitness. So people have looked at that and they hypothesized that clade C must be very fit. They did one-on-one competition, which is called a pairwise fitness experiment. And those experiments testing all clades within the group M that I showed you one-on-one showed that clade C actually is the least fit of all the clades within the group M, not the most fit. Then the hypothesis became that clade C probably is not overtly replicating, not announcing itself to the immune system so that it can linger on in the individual for a longer period of time and be able to spread to more individuals. That was the hypothesis by Eric Arts back then, who did extensive studies, published a couple of dozen papers on this topic. So he had shown that envelope glycoprotein gene may be one of the determinants, because when he swapped envelope, he could see fitness levels moves with it. So we hypothesized that the LYPX also contributes to fitness. So we compared clade C with clade B, and also we added back the LY, which is missing in clade C, to make a artificial virus that is plus LY. So what we see is that clade B is the most fit, clade C is the least fit, and you can actually improve the fitness by adding back just 2 amino acids. So at this point, we also noticed that in a collaborative study with Ujjwal Niyogi, we found that in drug failure cases in India, also in South Africa, you can see that individuals who suddenly stopped responding to antiretroviral treatment by increasing their virus loads and decreasing the CD4 levels, that they had acquired exactly where the LY should have been present, a tetrapeptide of this nature, PYRE, PYKE, or PYQE. We call them collectively PYXE. So we hypothesized that these insertions may allow the binding of Alex, regain ability to be more fit, and also respond to CCL2. And we created viruses that contain these 2 of the 3 insertions, and you can see that the binding, which was completely missing for the clade C, has returned to the level even better than clade B sometimes, as soon as you have the insertions. And we also looked at the fitness. This is the clade C, this is the clade B, and this is with the insertion of 2 amino acids, and these are the 2 tetrapeptide insertions. And we presume that if we add back the clade B envelope, it might actually overshoot the clade B fitness. We also saw that the intracellular GAG accumulates more in HIV-1C compared to B, and virus production is less robust in the case of C. All of these tell us that by regaining these tetrapeptide sequences, HIV-1C can become different and maybe more competitive. So this goes along well with the observation from Bangalore from Dr. Uday Kumar Ranga, who has shown that clade C can acquire an extra, already it has 2 NF-kappa B, 3 NF-kappa B sites, whereas clade B only has 2. And he has shown even a 4th NF-kappa B can be acquired. And other people have shown that clade C can also have duplications of PTAP motif to increase its ability to recruit TST101 perhaps. We don't know why it does that. So that's been observed too. So it looks like clade C HIV1 can do a lot of things by acquiring these extra sequences that allows it to become more fit. So I'm going to skip this summary because it was pretty straightforward, I hope. I want to tell you that the conclusions basically are that external stimuli such as CCL2 can modulate escort factors and affect virus budding and virus fitness. And this is basically an exploitation of a host innate immune response system by the virus. Also want to tell you that this is not limited to HIV-1. It's also seen in other retroviruses and many other RNA viruses and DNA viruses. Just want to conclude by adding a couple of things about what is going on in my lab currently. So you may all know about the CCR5 delta 32. That is a mutation in CCR5 that makes when you have homozygous condition with that mutation, HIV cannot infect those individuals. And when that mutation was identified, there was a spurt of activity looking for other polymorphisms in other chemokine receptors. And CCR2 was also thought to be a potential receptor, co-receptor for HIV-1, and they found one mutation as you know, all of these are 7 transmembrane domain proteins. So there is a mutation valine to isoleucine at position 64, and that had led to a delay in disease as pathogenesis, actually here measured by death. So this disease progression delay was attributed to poor binding of HIV to the receptor, but that turned out to be not true because this mutation doesn't affect the co-receptor function. So we hypothesized that the mutation is actually affecting the egress of HIV-1. So we hypothesized, we have tested this idea a little bit. I want to show you a couple of pieces of data. So what we did was we created HeLa cell lines expressing either wild type CCR2B or mutant CCR2B with the V64I. The wild type shows a similar pattern, CCL2 responsiveness by producing more virus particles with more CCL2 and very little virus or severe reduction in virus production when the CCL2 is depleted with anti-CCL2. Whereas HIV-1C has the same behavior as before. But now if you look at the mutant, you can see that even clade B has like 4-fold reduction in virus release, and more importantly, no responsiveness whatsoever to CCL2 levels. And HIV-1C remained the same. So to address the question how this may be happening, we looked at the mobilization of CCL2 by, mobilization of ALEX from actin compartment by the addition of CCL2 in these two cell lines. So as I mentioned before, only in anti-CCL2 condition you see complete localization of ALEX and actin as seen here. But when you look at the mutant cell line, you see that in all conditions there is a complete co-localization of ALEX and actin, suggesting that the binding of CCL2 to its receptor and signaling is very critical to allow for the utilization of ALEX by the virus for budding. So we are not only examining this mutation, we are also examining a CCL2 minus 2518G. This is a mutation in the enhancer which causes an elevated plasma CCL2 levels, and this allele has been shown to be enriched in people with HIV infection, at least in one big study. So we're looking at both of these in a cohort in the Bronx in New York, which is a nationwide network actually, MWCCS, Max Weiss combined cohorts study. The patients are beginning to be recruited, and we're going to take a look at what happens with these. So I'm going to stop at this point just by saying that there's a key role for signaling by CCL2 at the late stage of HIV replication. So suggesting that there is a value for targeting these molecules for therapeutics. There are many FDA-approved antiretrovirals, but there are hardly any drugs that are targeting at the late stage or very late steps. These are some of the people who worked on the project. The project was, much of it was really work by two individuals. Vasudev Rao is a postdoctoral fellow who is currently a program officer at the NIH, and a grad student who recently defended David Ajasin. And these are the four postdocs in the lab who are looking at the what I call the black box, which involves many aspects of how CCL2 brings about this ALEX movement. And Dr. Kalpana, I collaborate with heavily, and Dr. Ann Bresnik, who is an actin expert, who is also a partner in this study. I'm going to stop and take any questions.
Thanks, Professor Prasad, for the wonderful talk. Are there any questions? So IISc Biochemistry, so from my colleague, yeah.
Yeah, hi, Prasad, that was a great talk. So Saravanan here. So I have a question regarding this ALEX binding to F-actin. So when you add the CCL2, right? So do you think that cross-linking proteins such as alpha-actinin will play a role in bringing ALEX to the F-actin?
Yeah, not only alpha-actinin. So that's one of the proteins we are working on. Thank you for asking me that question. So it's been shown by another lab that ALEX binds to there is some crosstalk. So ALEX binds to 3 different proteins in the actin network. It's going to be actin, F-actin that is, and then alpha-actinin, and also cortactin. And the binding sites for all of these have been identified on ALEX, And in my lab, we are systematically creating stable cell lines, both HeLa cells and macrophages lacking the cortactin or alpha actinin. We can't do that with the actin, of course. So eagerly trying to find out what happens to CCL2 responsiveness.
And in connection with that, what happens that if you had a lat A to this system, and then are they disappears immediately, or they still have some association with it?
Yeah, it's a very good question. We have used latranculin A to do this experiment, and we can actually see that it releases Alex. And without latranculin, we don't see that. So it's actually part of the paper we published in 2019 in eLife.
Are there any more questions?
May I?
Yes.
Yeah, this is Subhash from NCL Pune. So far, you talked about polymerization and the actin binders. How about the actin nucleators?
For example, can you?
Well, you can take either formin or ARP2/3R associated complexes with crosstalk with Alex.
We have not seen anything yet, at least in the literature. So we're interested in this question in a really deep way. So we are looking to see what causes Alex to associate with, because what's amazing to see is that when you remove CCL2, there seems to be a complete association of Alex with actin network. So there is something that is being done. So modification of Alex, perhaps. So one of my postdocs is working on post-translation modifications of Alex, and another one is doing the partner study. So bringing down Alex binding partners. So in fact, we see many of those actin polymerization initiating proteins in our larger set of hits, but we don't know if they'll make it to the final hits or not.
Yeah, to add up, either the virus entry is Alex dependent?
No, it is not. No, there's nothing. So Alex only gets involved, as far as we know now, in the late stages during the budding.
All right. Thank you, Prasad. Nice talk.
Thanks.
Okay, so if there are no more questions, so let me thank Professor Prasad for the wonderful talk and go to the next speaker. So
thank you very much.
Professor Sandeep Burma. So it's a great pleasure for me to introduce second speaker for the session. Professor Sandeep Burma did his bachelor's and master's from BHU and obtained his PhD from National Institute of Immunology with Professor Saeed Hasnain. Later, he carried out his postdoctoral studies at Yale and Pennsylvania State University on transcriptional regulation. And subsequently, he worked with Dr. David Chen studying DNA repair at Los Alamos National Laboratory. In 2005, he established his lab at the Department of Radiation Oncology, UT Southwestern Medical Center. In 2019, he joined UT Health San Antonio, Texas, where he is a professor and vice chair of the Department of Neurosurgery. He is also a Mace Family Foundation Distinguished Chair at MD Anderson Cancer Center. And he has been working on DNA damage responses, genome stability, and cancer biology for more than 2 decades. And he has received several awards and serving in numerous committees. And the title of his today's talk is DNA Damage, Senescence, and Cancer. Great pleasure, Sandeep.
So thank you, Ganesh. Can you hear me just fine?
Yes, yes.
Okay, if something comes up and I get muted, just please let me know because I'm having trouble with my laptop. But again, thank you very much for the kind introduction. And also a very big thank you to the department chair and to the organizing committee for the invitation to present at this conference. And I have really enjoyed my previous visits to your department and your beautiful campus. And I really wish I could have been there in person to sort of share in the excitement of the Centenary celebrations. But hopefully, we'll be there. We'll meet again in the future once the pandemic settles down. And I'm really, excited about at least meeting with you virtually today. So as Ganesh mentioned, I moved recently to the UT Health Science Center in San Antonio, where my lab is located in this South Texas research facility in this STRF building. And my lab works on the responses of mammalian cells to DNA double-strand breaks. and the carcinogenic and cancer therapeutic implications of these breaks. With these three projects, which hopefully work in synergy. So we are basically interested in mechanisms of DNA double-strand break repair with the translational objective of targeting some of these repair pathways to augment radio or chemotherapy of glioblastoma, which, as you all know, are lethal brain tumors desperately in need of better therapeutic options. And Project 3 is the flip side of Project 2, where we are looking at the process of ionizing radiation induced GBM development, because paradoxically, ionizing radiation, which is used for cancer therapy, is also the only known risk factor for glioma development, which kind of speaks to the paradoxical nature of radiation. The fact that it is used for cancer therapy, but it can also cause cancer. And as I will talk about briefly today, it can also drive tumor recurrence. So today I'll just briefly talk about a short story from our glioblastoma project. And just by way of introduction, and this is something that most of you here are quite familiar with, GBMs are highly radioresistant brain tumors, which arise either from astrocytes or largely from precursor stem cells, either suddenly de novo as primary glioblastomas, which is what is more common, and sometimes they progress from grade 2 to grade 3 astrocytomas to grade 4 astrocytomas, or glioblastoma multiforme, or glioblastoma as it is now called, by the stepwise accrual of mutations. And when I started working on GBM at UT Southwestern about 15, 16 years back, the Cancer Genome Atlas Network had just come out with its characterization of its first cancer, which was GBM, which essentially reconfirmed the commonest sort of tumor suppressor losses and oncogenic events that drive gliomagenesis. So a lot of my work at UT Southwestern was essentially focused on trying to understand how some of these oncogenic pathways might contribute to radioresistance. And in that regard, it is important to point out that gliomas are really desperately in need of better therapeutic options because surgical resection of these tumors adds about 6 months to survival, and ionizing radiation then adds another 6 to 7 months to survival. Yet, in spite of decades of research, ionizing radiation is still the most effective therapeutic modality for these tumors. And the addition of temozolomide to ionizing radiation was considered to be a breakthrough, but it still adds only sort of a scant 2 more months to survival. But ionizing radiation remains the mainstay of GBM therapy, and any other sort of therapeutic modality, novel therapeutic modality, has to be compatible with the existing therapy, which is IR with TMZ, is the point I want to make here. So a lot of our early work was focused on trying to understand the possible crosstalk between oncogenic pathways driving gliomagenesis and DNA repair mechanisms in these tumors, which might contribute to radioresistance, and to the possibility of subverting some of these connections between the signaling pathways and DNA repair mechanisms in the nucleus in order to augment radio or chemotherapy of these tumors. But of late, we have become more interested in sort of understanding the forces that drive GBM recurrence after radiotherapy, because these tumors are radioresistant, but even if they respond initially to radiation, they recur within a few months, and the recurrent tumor is usually much more aggressive and much more resistant to further therapeutic interventions. So in that regard, it's important to point out here that these tumors are actually treated with a whopping dose of radiation with about 60 gray, given in 2 gray fractions. And the radiation volume actually includes a 2 to 3 centimeter margin of normal brain tissues surrounding the tumor, which is also evident here from the treatment sort of planning images here. And this is simply because these tumor cells are very infiltrative and they infiltrate into the margin. And so it is necessary to irradiate the margin in order to sort of get to these infiltrating tumor cells, which are not sort of removed by surgery. And the recurrent tumor usually arises in the margin. And so presumably residual tumor cells in the margin evolve under therapeutic pressure and they contribute to recurrence. But the other idea that we have been entertaining is the possibility that the high doses of ionizing radiation actually alter the tumor microenvironment. And in that manner, paradoxically, radiation might actually be driving recurrence by altering the microenvironment. So to get to this, we used our preclinical sort of mouse models, of which we have many. But in this case, what we did was we took C57 black 6 mice and irradiated them with 10 gray of radiation, waited 30 days. We have determined, we have done this at 15, 30, 45 days and such. And after 30 days, we implanted a very small number of GL261 syngeneic glioma cells, which are luciferase tagged and monitored for tumor growth. And we find here that if you radiate the brain first and then implant GL261 cells, as you can see here in the bottom panel, these tumor cells grow like gangbusters. As you can see here, if you compare the red and the blue plots, resulting in shortened survival of tumor-bearing mice. So something about radiation, pre-radiation, promotes tumor growth, and this has been seen in other syngeneic mouse models that we have queried. All of the data has been looked at in multiple lines and multiple mouse strains, so it's not strain-specific is the point I want to make here. And when we look at the tumors themselves, the tumors are a very different beast altogether. So they are obviously much larger. The tumors growing in pre-irradiated brains, they're much more angiogenic. They have a much more strong sort of stem cell phenotype. They're stain positive for SOX2. And very interestingly, while the tumors growing in unirradiated brains have a more or less more defined border, the tumors growing in the pre-irradiated brains are much more infiltrated. They're much more invasive. And I do want you to keep this sort of picture in mind because I'll come back to it in a second. So to understand what it was about radiation and the brain that contributed to tumor growth, we queried the radiated brains or mock radiated brains by RNA-seq and found upregulation of a number of genes in the radiated brains at 30 days post-IR. And interestingly, we found upregulation of a large number of tumor-promoting factors in the radiated brain. And many of these tumor-promoting factors, especially the ones that were sort of significantly upregulated in the brain, as seen here in this volcano plot, are very reminiscent of the so-called senescence-associated secretory phenotype proteins, or SAS proteins. And I'll come to that in a second. But this is something that most of you here are quite familiar with, senescence and SASP. So senescence obviously is a state of permanent cell cycle arrest that a cell goes into under either oncogenic stress or replicative stress, or in the context of irreparable DNA damage, and also in the context of, let's say, telomere shortening, that's replicative senescence. And these cells acquire this flattened morphology. They're no longer dividing, they're positive for beta-gal. And as we all know, going back to our college days, that senescence is considered to be a bona fide anti-cancer barrier because it prevents the damaged cell from proliferating further. But interestingly, there is a sort of darker side to senescence which has become evident in recent years, which many of you here will be quite familiar with. So one of the hallmark of a senescent cell is the secretion of these so-called SAS, sort of proteins. So this secretory phenotype involves secretion of cytokines, chemokines, metalloproteases, and this sort of SAS phenotype that is created by the senescent cell underlies a large number of disease pathologies, including age-related syndromes and neurodegeneration, because some of these SAS proteins can have very detrimental effects. So here we are looking at sort of antagonistic pleiotropy, really. And again, and just by the way, and I won't go into this anymore, but there are multiple, so while senescence is driven by P16 and or P21 dependent pathways, SASP itself is controlled by multiple pathways, but primarily by activation of the NF-kappa B pathway, by DNA double-strand breaks, such as those caused by radiation, and the DDR signaling that ensues from these breaks, as well as by the activation of the cGAS-STING pathway by cytoplasmic DNA. So senescence and SASP then have detrimental effects. And in the context of cancer, it has been shown for a number of cancers, including breast, lung, liver, and skin, that the secretion of sort of these SAS proteins by senescent stromal cells can promote the growth of neighboring tumor cells by promoting inflammation and tumor growth. And So this is sort of, in the context of SAS, this is the detrimental sort of the dark side of senescence, so to speak. So we naturally, given that we saw upregulation of a number of SAS factors in the brain, we naturally wondered whether ionizing radiation triggers senescence in the brain, which appears to be the case because we radiate the mouse brain with 30 days, stain for beta-gal. You see robust beta-gal staining in the brain. And by staining for other markers of senescence, such as P21 positivity or loss of nuclear lamin B1, we found that the astrocytic population in the brain is particularly vulnerable. For example, you can see here this GFAP positive green, sort of star-like cell, which is obviously senescent. And astrocytes have also been shown to be prone to senescence in Alzheimer's and other neurodegenerative disease models and contribute to neurodegeneration. So this is not surprising. And we find that this is dependent upon P21 because if you take P21 knockout mice and radiate them, you don't see senescence, you don't see appreciable beta-gal staining, you don't see loss of lamin B1. And in P21 knockout mice, you don't see this sort of robust promotion of tumor growth, which you do in P21 plus mice. So this is a P21 dependent phenomenon. And this again tells us that the tumor growth that we are seeing and the invasiveness that we are seeing is possibly senescence driven and possibly astrocyte driven, or at least in part astrocyte driven. And so in order to sort of see if astrocytes really can promote tumor growth, we carried out this hybrid in vitro, in vivo experiment where we cultured primary mouse astrocytes in vitro and irradiated them to trigger senescence. And after 10 days, we sort of co-implanted senescent astrocytes or sort of non-senescent mock irradiated astrocytes with tumor cells and monitored tumor growth. And you can see here how co-implantation with senescent astrocytes really augments the growth of luciferase tagged GL261 tumors in the mouse brain. So hence, we then focused a little bit more on these astrocytes, irradiated them in vitro, and of the SAS genes that were significantly upregulated in the mouse brain, the 14 genes that we sort of shortlisted, we verified that at least 13 of those genes were significantly upregulated in senescent astrocytes irradiated and pushed to senescence in vitro. And of these sort of 13 genes, for a number of reasons, for the moment, we chose to focus on HGF, or the hepatocyte growth factor, also known as the scatter factor, which activates a MET receptor tyrosine kinase, for a number of reasons. But to give you a couple, of all these 13 significant SAS genes, when we queried the TCGA database, we found that HGF was the most commonly altered in GBM. And again, here we are looking at radiated mouse brains, and I'm just showing you here by Western blotting upregulation of HGF, as well as, of course, P21 in different sort of individual mice that were irradiated. And HGF, as I mentioned, activates the RTK MET. And the HGF MET axis is pretty relevant to human GBM because MET RTKs are expressed in a good percentage, especially in the mesenchymal subtype of recurrent GBM and in recurrent GBM. And I forgot to mention in the beginning, gliomas are now sort of characterized molecularly and genetically into sort of 3 different subtypes, proneural, mesenchymal, and sort of classical. So MET is relevant to the mesenchymal subtype. And MET signaling has been associated with therapeutic resistance in GBM and with the maintenance of a cancer stem cell phenotype in these tumors. But from the point of view of, subsequent data that I'll show you, HGF is also called the scatter factor because HGF activates MET, and MET signaling promotes proliferation, scattering, and invasion of tumor cells. So that's one reason why, another reason why we were interested in HGF and MET. And so when we looked for MET activation in these tumors, we found that the tumors implanted into mock irradiated brains did not show appreciable MET activation, as seen here by phospho-MET staining. But the tumors growing in pre-irradiated brains showed pretty robust MET activation, which therefore sort of led us to ask whether HGF secretion by these senescent astrocytes or other cell types in the brain might be promoting tumor growth and invasion via MET activation, is the obvious question to ask, which we asked in vitro by irradiating sort of astrocytes and pushing them to senesce, which they do pretty rapidly within 10 days. And the induction of senescence here is queried by the loss of nuclear lamin B1. And we find that these senescent astrocytes do secrete HGF into the media here seen by ELISA. And when we take conditioned media from these senescent astrocytes and layer them onto GL261 cells or other CT2A glioma lines, you can see that conditioned media can activate MET in the receptor, in the recipient tumor cells. And so we next sort of carried out void and chamber assays to look at both migration as well as invasion. Invasion is not shown here, but essentially when we layer irradiated astrocytes in the bottom chamber and put glioma cells in the top chamber, you can see how senescent astrocytes in the bottom chamber can significantly push the migration as well as invasion of tumor cells. And this is in part driven by the HGF-MET axis because it can be attenuated, though not ablated, by the use of neutralizing HGF and anti-HGF antibodies, or by using a MET inhibitor, crizotinib. So obviously we are focusing on HGF-MET, but I do want to point out that there are multiple sort of signaling pathways I'm sure that are at play here between the astrocyte that is senescent and the glioma cell that it is pushing to proliferate and invade. And which then kind of brings me to this concept of senolysis, or even xenomorphics. So as many of you here would know, there is a sort of burgeoning interest, especially in the aging field, in senolytics. And senolytics essentially eliminate or are supposed to eliminate senescent cells or attenuate detrimental SAS while sparing normal cells. And so the first generation of senolytics were were discovered based upon sort of a mechanism-based process by which senolytics were queried that could target the anti-apoptotic pathways that senescent cells use to survive. And of these senolytics, I'll just focus quickly on navitoclax for ABT263. But now you have second-generation senolytics which are being identified by high-throughput screening, and there are other approaches at play too, such as the use of esa-beta-gal activated toxins. But these senolytics have been tested extensively in preclinical models, and these senolytics are also in a number of clinical trials, especially for age-related syndromes. And there is emerging evidence that senolytics can be beneficial for a host of human diseases, or age-related syndromes and neurodegeneration, including, as is listed at the bottom, fibrosis caused by COVID. So a lot of interest in senolytics at the moment, but we have just used this at the moment for sort of, from a more sort of basic standpoint, really. The next couple of slides are not really preclinical studies, but more sort of basic studies. So what we have done here is we have used ABT263 on navitoclax, and the reason why we chose navitoclax is because in a couple of other, a few other earlier studies using Alzheimer's models, people have shown that navitoclax can cross the blood-brain barrier and eliminate senescent astrocytes in Alzheimer's models, and thereby ameliorate neurodegeneration caused by these senescent astrocytes. So here in this case, we irradiated mouse brains and treated them repeatedly over 25 cycles, really, with either vehicle or ABT263. And we then looked at the senescent populations in the brain, and we see reduced esa-beta-gal staining and increased apoptosis in the brain, indicating that ABT263 might be clearing out some of the senescent cells by inducing apoptosis in these cells. And to look at the effect of this drug on tumor growth, we then carried out another experiment where it irradiate the mouse brain, clear away senescent cells with ABT263, and then implant a small number of GL261 cells. So this is not really a preclinical experiment because we are actually treating with the drug first and then putting in tumor cells. So the tumor cells do not really see the drug. And we find that pre-treatment with ABT263, if you compare the blue and the red plot, attenuates tumor growth and improves survival of tumor-bearing mice. And if you look at the resulting tumors, you find here that the tumors growing in ABT263 pre-treated mouse brains have are highly necrotic, as you can see here in the core, and the surrounding tumor cells are highly tunnel positive. So this kind of tells us that without this supporting mesh of senescent astrocytes, which you have removed by using ABT263, the tumor is not able to sustain itself and perhaps succumbs to apoptosis and necrosis. But again, this is not preclinical model. This is just to help us understand how these senescent cells are promoting tumor growth. But the preclinical studies are what we plan to do next. So this kind of brings us to our grand hypothesis, which is that radiation-induced GBM recurrence is a problem, and perhaps radiation-induced GBM recurrence involves cooperation between genomic changes in the tumor cell caused by radiation, such as MET amplification, which I didn't get into today, and senescence-associated changes in the tumor and microenvironment, such as the secretion of HGF by the senescent astrocytes, which then brings us to the concept of adjuvant senolytic therapy, which could be given after radiation. And this could be effective because adjuvant senolytic therapy will not only deplete the senescent tumor cells with tumor supportive roles, but will also eliminate that fraction of the tumor that has been also rendered senescent by ionizing radiation. So it's like a 1-2 punch is the idea behind adjuvant senolytic therapy, which we will test next. And this work was published just a few days back in Cancer Research, and it was very gratifying for us to find that our study made the cover of the December 1 print issue of Cancer Research. And going forward, we are really interested in senescence and its ramifications, which I could discuss with you on some other occasion. And I would like to end by thanking the people in my lab and my wonderful collaborators and funding from NASA and NIH. And I'd also like to acknowledge Elliot Fletcher, a postdoc in my lab at UT Southwestern, who essentially initiated this particular project. So thank you for your attention, and I'll be very happy to take questions.
Thanks for the wonderful talk, Sandeep.
Thanks, Anish.
Okay, there is a question from my colleague.
Yeah, hi, Dr. Burma. I was wondering, at least for some of the senolytics, I'm not sure about the ones that you used in your study. It is known that they also affect pathways other than the clearance of the senescent cells. So have you entertained the possibility that some of these observations that you are observing could be due to, I mean, of course, there is the senescent cells are getting cleared, but are there other pathways that could be helping as well? Have you looked into that, or any thoughts?
Yeah, that's a good point. So basically, many of these senolytics actually do either affect the anti-apoptotic pathways, like BCL-XL2 family proteins, which the tumor cells use to survive, or they can also affect certain signaling pathways, such as the D plus Q combination, again, pro-survival signaling pathways. So the best way to answer your question would be to use a genetic model, which we haven't done yet, where we can actually, let's say, use an ink attack mouse model to remove senescent cells genetically. But there is not yet any kind of P21 driven mouse model where we can remove P21 positive senescent cells. So the best answer would be to remove the cells using a genetic approach, failing which in this case, the basic idea here is that we are probably removing only the senescent astrocytes, although we cannot say that for sure. The only thing I can say here is in this scenario, we are treating with ABT first and then putting in the tumor cells. So we are really not affecting any of the signaling pathways in the tumor cells, and we are basically focusing on the clearance of the senescent cells. And in some ways, even if these senolytics do affect some of the pro-survival pathways in the tumor cell itself, that is not a bad thing. So many of the senolytics are actually anti-cancer drugs too, which have been repurposed as senolytics. So your point is absolutely valid, and but in some scenarios where the senolytic actually affects the tumor cell as well as the tumor microenvironment, you could have a a double whammy, which might be beneficial. But your point is absolutely valid, because of which there is interest now in also in what are called xenomorphics. So drugs that do not eliminate senescent cells but only get rid of the bad SASP while leaving the good SASP on. but valid point.
Thank you.
So Mani has a question.
Yeah, yes, sir. Thank you so much, sir, for such a nice talk. Actually, I kind of have a very basic question. So you talked about the molecules like HGF and its interaction with the MAT molecule, right? So the question that I have is that is it possible for these molecules to infiltrate to other tissues, and can these molecules ultimately lead into metastasis of cancer or other phenotypes?
So in the context, that's a good point. I mean, in the context of in the context of the brain, obviously, I think the effect of the HGF that is would be probably local, but
Okay.
yeah, I mean maybe not in the context of HGF. I do not know how far HGF travels and so on and so forth, and how much it can have sort of effects that are sort of long distance effects. But certainly that's a possibility. I mean, you could have so-called sort of abscopal effects where perhaps you could have long-distance effects, but I don't have an exact answer in the context of sort of brain tumors. But the other interesting thing that can happen is, which is, I'm sort of veering off into a totally different, sort of area, but the same point applies, is what is known as senescence spreading. So actually, senescent cells can actually induce senescence in neighboring cells, and this phenomenon called senescence spreading can actually amplify the senescence response, but again, sort of in a local manner. But I will not be able to, I'm not 100% sure whether secretion of HGF in the brain would also affect sort of distal organs.
Okay.
But
Thank you
something
so
to
much.
think about.
Yeah, thank you.
Sandeep, I have two questions. So the first question is that, is there any evidence in the glioblastoma patients? So the glioma, the tumors have arisen due to senescence of some cells in the astrocytes.
That's a very good question. So the thing is, so gliomas usually arise in older patients, right? So 60 plus years. And by which time you already have a good degree of sort of astrocytic senescence or even senescence of other cell types in the brain, which have been shown in the neurodegeneration field to contribute to sort of age-related neurodegenerative pathologies such as Alzheimer's. So there is a possibility that a degree of senescence in the brain could also promote gliomagenesis. So right now I'm talking about radiation-induced senescence promoting tumor recurrence, but it is also possible that tumor development could be induced by senescence, and that's something we really need to look into down the road as to whether using aged mice as an example to see whether there is tumor promotion. Also, it's a very good point, and it's a very valid point. And I'm sorry, you had another question?
Yeah, my second question is that your ABT 263. So what is the mechanism, how it is inducing apoptosis?
So basically, it sort of targets the BCL2-XL family of proteins, the anti-apoptotic proteins. So senescent tumors, senescent cells upregulate, anti-apoptotic proteins just so that they can survive long term in that state. So you're kind of targeting those anti-apoptotic pathways and inducing senescence, inducing death in the senescent cells. But at the same time, ABT263 is also used as an anti-tumor agent because tumor cells also upregulate, in some cases, anti-apoptotic pathways to survive. So in some ways, it could be a bit of a double, sort of a double whammy, really. So you could be affecting the tumor microenvironment as well as the tumor cell, which may not be a bad thing in certain contexts,
So wonderful, very informative talk and excellent talk, Sandeep.
Thank you.
And with this, let us thank both the speakers for their excellent talk, and then end the session and over to the big stream for initiating the next session.
Thank you.
Thank you, Professor Prasad and Professor Verma for the wonderful talk, and Professor Ganesh for chairing this session. Now I request Devanjali to start the plenary talks of the last day.
Thank you, Satyaranjan. Up next, we have plenary talks from two remarkable scientists in their fields, Professor James Westwood from Virginia Tech and Professor Maria Jason from Memorial Sloan Kettering. This session will be chaired by Professor P.N. Rangarajan. I would like to welcome Professor P.N. Rangarajan, Chair of Department of Biochemistry, with whom we have already been acquainted at the beginning of the conference. So I would request him to kindly introduce the first speaker.
Westwood?
Sorry, it was muted.
Can you speak? Yeah, Professor Westwood, can you log in and can you turn on the video, please?
I am here. Yeah, just a moment. Shall I share my screen?
Oh, there you are. Okay. I'll just briefly introduce Professor Westwood. It's my pleasure to introduce Professor Westwood, who is a faculty in the School of Plant and Environmental Sciences in Virginia Tech, Virginia, United States, since 2018. His research expertise includes parasitic plants and weed science, and has been working on deciphering how parasitic plants interact with their host to better understand basic plant biology and improve techniques for applied vegetation management. He has been a Fulbright scholar, and he is also a recipient of several awards, including the Virginia Tech College of Agriculture and Life Sciences Excellence in Basic Research Award, Henderson Award for Outstanding Faculty Member in the Department of Plant Pathology and Physiology and Weed Science. He's an honorary member of International Parasitic Plant Society. And today he is going to talk about the parasitic plant dodder, which is also known as cuscuta, how it uses RNAs to manipulate its host. I must mention Professor Westwood, I'm sure he's already aware that a lot of work on cuscuta was carried out in our department by Professor Mahadevan in the late 70s and 80s, in the early 70s and 80s. I think he's also here in the audience, and it's a pleasure to listen to your talk. Professor Westwood, please.
Thank you. Thanks for the kind introduction. And yes, greetings to everyone. I wish it was possible to be there in person, but sadly we are inhibited these days, but fortunately we can do this by Zoom. So yeah, I'm going to change things up. It looks like you've had a lot of talks relating to human medicine. So I'm going to take you not only into the world of plants, but into the world of parasitic plants. And I'm going to start actually with my acknowledgements to get these out of the way. And just to say that there's been a lot of people working in my lab over the years who've contributed to the work that I'm going to talk about today. Although much of it I will feature is from the work of Soyeon Park, who's here indicated by the yellow arrow down there, the photo. I've had good funding over the years from our National Science Foundation as well. So the world of parasitic plants. So what we know about plants are that they are very diverse, but most people don't realize that there are plants that are actually parasitic. And so there may be a greater awareness of this in India than there is in the US. We don't have so many problematic parasites here, but you can see from this slide some of the diversity. And about 1% of plant species have actually evolved to become parasitic, which is that they all derived from autotrophic green plants, the kind we're most familiar with. But over the course of evolution, they gave up that ability to perform photosynthesis to some extent or another. And they developed the ability to attack and gain resources from other plants. And so what I'm showing you here is a phylogeny of plants and all the families in red. And I apologize for the small font. You don't really have to read that, but you can appreciate here that within the whole plant kingdom, you have at least 12 times that plants have independently evolved this ability to parasitize other plants. And I'm going to talk to you today only about 2 of them. And so just a small fraction of the fascinating plants, but I'll talk about the Orbancaceae and Convolvulaceae here at the top, the boom rapes, the Enstriga, and the Convolvulus cuscuta. We are interested in these because they are weeds, right? A lot of them, they're all interesting, right? But only some are economically impactful enough to generate the kind of research money we need to study them. So here's some of the worst ones. There's cuscuta up on the upper left of your screen there, and that is one I will talk about most. But I first want to introduce you to this other important group of parasitic plants, which are the Orbancaceae, the root parasites. And so these include Striga down on the lower left there, and then the broom rapes, which are in the other pictures. And I'll say a little bit more about these. So first, when we talk about parasitic plants and the impact of parasitic plants, most people in the world would peg Striga, or the witchweeds, as the greatest impact, just because of their ability to devastate crops that are, and also they are in a part of the world that is, the farmers have least capacity to deal with this, right? So we're talking about Africa, and I do know that this has become a little bit of a problem in India as well. But what you're seeing here in this picture is the field on the bottom. It should be a cornfield in Kenya, but what you see is where there should be a corn plant there. There's a clump of plants, and they're surrounded by these lovely, beautiful flowers, purple flowers, and that's not the corn flower at all. That's the Striga flowers, and the Striga is attached underground to the root of the corn. And here it is. This is on sorghum, but if you dig this up, you find that the Striga there is attached, I don't know if you can see my pointer, on that over to a thin root of the host. And most of its resources are coming from that host. The Striga does make some green leaves, does a little bit of photosynthesis on its own, so it's we'll call a hemiparasite, but it absolutely needs that host for water and a lot of its carbon as well. Now, the related species that I mentioned are the broom rapes, the Orbankia and Philipponki species. And these are very similar, and they're from the same family as Striga. The big difference is that these are really holoparasites. They don't have any fully formed leaves, no chlorophyll, really. And so they're holoparasites. And they're also different in the hosts that they attack, whereas Striga was attacking the cereal crops, the corn, millet, sorghum, those staple crops that are really important for food in Africa. These parasites, the broom rapes, are attacking dicots. So these are broadleaf crops, tomatoes, sunflower, parsley, and beans. Now these can be important foods. They're more important as cash crops, and probably the ones that attack sunflower are the most economically important. And you find these predominantly in the northern part of Africa and Mediterranean, Middle East. So these two parasites kind of divided up the world a little bit geographically and botanically, and they're very interesting. And let me just say a couple more things about this. So here's one of these, Orbankia crenata, the crenate broom rape on broadbean in Kenya, and you can see this is a research plot, but that heavy infestation of the parasites is really a beautiful flower, I think. However, if you're a broadbean or fava bean farmer, you don't appreciate that. So that's crop destruction and total yield loss under a condition like that. So what has intrigued people for years about these parasites is this ability of the parasite to detect not only the presence of the host, but actually to identify a host root before germination. And the way the seed does this, it sits in the soil and it waits for a host root to grow past it. And as that host root exudes chemicals into the soil, the parasite pays attention and will respond to very specific chemical signals. And it can tell the difference from one host species to another. So it only germinates when an appropriate host root is nearby. And we've found these signals to be primarily strigolactone molecules, as you see here. And it turns out that strigolactones are actually plant hormones, and they're produced in the roots of plants. They are transported throughout the plant, and they control such things as plant architecture, how often that plant branches, right? So is it a single stem or multi-stemmed, and as well as root architecture. But also the strigolactones are exuded into the soil. And in the soil, they signal to beneficial fungi. These are arbuscular mycorrhizal fungi, which form a symbiotic association with the host root and greatly enhance the ability of the plant to thrive in the soil. And the parasite has been able to eavesdrop on this conversation and germinate in response to that. Now, I tell you this partly because this is my biochemistry connection. Since you're at the biochemistry department, I first learned about parasitic plants from this man, Larry Butler, who was a professor of biochemistry at Purdue University. And he was working on the stimulant, the germination stimulant for Striga. And he, even back at that time, so this was the early 1990s, and he was trying to figure this out, the biochemistry of this. And back then, we didn't know very much. Most of what I just told you was still a mystery. We wondered why plants would produce strigolactones or any chemical that would induce the parasite to germinate. And now we know much about this. And I will just wrap this up by saying that, that here we have this great example of plant-plant interaction. And if you are a young biochemist and you want something to do with your career, you could do a lot worse than to study strigolactones. There's still many mysteries out there to be solved about how they work and how they function. So let me kind of wrap up this section with just a brief life cycle and start to get onto the main message here. But what you see here is the life cycle of Philipponchus species, and we start with the seed and the germination signal I just told you about. This is plant signaling point number 1. So in order to germinate, there needs to be a signal received from the host plant that the parasite recognizes and causes it to germinate. But that is only the beginning of the parasite's ability to interact with the host, right? Because then it produces this tiny little root-like structure we call a radical, and that must then grow toward the host root, contact it, and develop the hostorium. That's critical as well. And this involves a second independent signal. And this one's poorly characterized. We know a few compounds that will stimulate development of the hostorium. We don't know really what the natural factor is, though. But anyway, the hostorium is this unique structure that only parasitic plants have. And it grows internal into the host, and it forms connection to the vascular tissue of the host. At this point, you have cell-to-cell contact where one plant grows invasively into another plant and makes those connections. I think of this as a point 3 of the plant signaling, because there has to be a mutual recognition, one plant of the other, and a coordinated growth. And then furthermore, there has to be further communication where the parasite somehow communicates to the host plant that the host should not defend itself, or somehow defeat the host defenses, and convince the host plant that it should export resources to the parasite, to treat the parasite as an extension of itself. So there's a lot of signaling going on here, and my career has taken me through many aspects of these interactions, but lately, and the thing I want to focus on now, are those later stage events. And this is where I'm going to take you to above ground, and we are going to consider the parasitic plant Cuscuta or daughter. And you see that here, and the parasite here is a twining stem, and it, in fact, once it germinates, does not have a root system anymore. And let me just introduce you to this with my video, because this is the best way for you to understand what I'm talking about. So this is I'm going to turn off the sound. This is on YouTube, The Attack of the Parasitic Plant. If you want to look at this later, you can just find that by Googling. So in contrast to the Orbancaceae parasites, The Cuscuta just germinates in the spring when other plants are germinating, and no special signal is involved. And yet the parasite still must find its way to the host plant. And notice here how it's growing towards those tomato plants. And let me actually pause this and emphasize a point. So I'm going to back it up a little bit. There we go. There's your parasites. They've germinated. And these plants, notice they do not grow away from the host plants. They somehow, before they even contact that host, they know where the host is and they grow towards it. They make physical contact and they wrap around. So we think that that attractive force is perhaps color, light quality, and also maybe volatile chemical signals given off by the host that the parasite is able to perceive. But once it makes contact, you can see how it tightens up the coil around that host stem. And then where it is attached from each of those bulges along the parasite stem, it's growing intrusively a hostoria into the host and making vascular connections there. And so here, not just one hostoria, but many hostoria connect that parasite. And if you're a farmer and you have this problem, you try to pull away the shoots, the plant can regrow from those hostorial attachments. So you see that there. But once the parasite is established, as I said before, the root system just senesces, so there's no more connection to the soil. The plant is getting everything from the host, and it branches and it grows. And each time it branches, at every node, it forms a new shoot, and each new shoot is capable of making a new connection. So at this point, you get basically a network, a web of Cuscuta attached. And this is growing on a Arabidopsis. We don't need to go through the rest of the video there. But from this video, you get an idea of really how that parasite behaves and how it lives. But let me diagram a little bit more the connections because this is important. So Cuscuta, as I've diagrammed it here, let me pick up my laser pointer. Here, growing on an Arabidopsis, and then we're showing it in cross section as it would be across one of these hostoria here that looks like that. And the way you can imagine this is those hostoria then grow into the host stem, that cross section, and there are cells of the parasite that grow either between host cells or actually right through host cells. And as they do that, they form, they lay down host cell on one side, parasite cell on another, and make these very tight connections. So the types of connections we have here are the following, where you have a cell of the parasite that meets the xylem, the water conducting tissue of the host, it will differentiate into the xylem. So this is just like your plumber's pipe, right? There's no barrier there to water flow between host and parasites. That will just conduct water from the parasite to the host. The phloem cells conduct the sugar photosynthase of the parasite. In Cuscuta, we see evidence that the parasite cells that contact phloem seem to turn into what we call transfer cells. There's no open connections that we've been able to report, but they probably pick up materials by cross-membrane transfer. However, there are also what's called interspecific plasmodesmata. This is where you've got host and parasite cells together, and they're spanned by these pores plants have from cell to cell called plasmodesmata that actually connect the cytoplasm from one plant to another, from one cell to another. In this case, you actually have plasmodesmata that span the Cuscuta to the host. And so this is the most intimate connection you can have in the plant world, right? Of cell-to-cell cytoplasmic continuity. And this allows the kind of exchange that I will talk about. We really don't know much more about how the exchange functions or exact routes that are taken for everything. But I want you to be aware of this in light of what I'm going to talk about. So now, Coscuta is also the only parasite that I know that makes connections to multiple hosts at one time. So rather than doing it in sequence, this one plant here is connecting, for example, a whole row of lentils together. Or in the case below, it's linking a fava bean to a couple different weed species. So you have these networks. And I want to show you this. This is Coscuta on cranberry. And this is in Massachusetts in the US in the Northeast. That should be a field of cranberry. So it should be a green field. In fact, it's a mat of Coscuta making millions of connections, linking together plants in some kind of incredible exchange. I don't know what's going on there, but I want you to also keep that image in mind as I talk about the things that we've been studying. So we've been interested in exchange of substances between the host and the parasite. So I'm going to run through quickly the list, and then I'm going to go into detail on a few things. So there's water. Of course, all parasites need water. That is probably the first and most important need. There's also sugars and small molecules. This is widely been recognized. Of course, the parasite needs that sugar, right? And I'm just illustrating this with an old picture from a paper showing the stain carboxyfluorocene, which is a phloem mobile dye. And you can see there's the host on the bottom, the parasite on the top of the image, and you see the green apparently moving through the hostorium in what looks like the vascular phloem tissues, right? So this has all been known for a long time. What was the main contribution really of my group to this field was the discovery that they're also exchanging nucleic acids. This was a graduate student of mine, Gunjun Kim, who did most of this work. And so we reported here, if you look at this simple diagram, this is our basic model here, right? So you've got a stem of Arabidopsis, the model plant for plant science, and coiled around that is the Coscuta. And you can harvest a bit of Coscuta out here away from the host. You can harvest the host away from the point of connection. We call this the interface, abbreviated I here. And then if you look at these different regions, and this is just the reconstruction of a host gene using high throughput sequencing. This is RNA-seq, basically, right? And so it is no challenge to reassemble this gene using either the host or the interface tissues here. But what was really amazing was to be able to basically reconstitute that host gene from transcripts in the parasite. So this was our proof that the parasite was taking up host RNAs, and not only a few of them, as was previously thought, but thousands of them. And this is just to show that we found that there were thousands of different RNAs transported from the host to the parasite, and also Coscuta RNA is found in the host. So the exchange is bidirectional, right? So it's not just that the parasite is taking only. For some reason, it's also letting some of its own RNAs get into the host. And I'll say more about that later. The other thing that we know moves is protein. And this is, again, an old experiment with the green fluorescent protein moving from host to parasite. Same as I showed you for that carboxyfluorocene. So you have the host down here, Coscuta up there. So the small protein that is soluble seems to, will clearly moves into the parasite. The other story is microRNA, and I will say more about that in a moment and give you more information on that. The last thing that is exchanged, though, that I want to mention, and I will not talk about further, is DNA movement. And we know that DNA is exchanged because we can find evidence of it, but it's not the same dynamic as I'm talking about for everything else on this list, right? All of those other things you can find in real time if you pick a piece of Cascuda off a host and look at it, right now, you can see the RNAs, proteins, microRNAs from the host. They're moving in real time. For DNA, you have to look into the record. So this involves studies of horizontal gene transfer. And for this, I've worked with my colleague Claude Pamphlis at Penn State, and he does this sort of analysis a lot. And so you have to look at signatures of historical DNA transfers. And what appears to be happening is that parasitic plants are able to take up large chunks of DNA from their hosts and incorporate them into their own genomes. And some of these appear to be used by the parasites to their benefit over evolutionary history. When we look for DNA in real time, we don't find it. It doesn't seem to be moving in the translocation stream or anything like that. But over evolutionary history, this has shaped the parasite genomes, and those parasites that are more dependent on their host pick up more of this horizontal gene transfer. So this is another interesting aspect of parasitic plants, but one that I won't dwell on because I want to focus more on these processes. RNA, microRNA, and protein that really shape, I think, the interaction between the host and the parasite. And let's say, there was a paradigm came up some years ago with microRNAs. And I'm going to talk about these first, because the idea that microRNAs can actually move between organisms and transmit information is a kind of a crazy idea at one point. Now it seems a little bit accepted. And certainly we know that plants and fungi are exchanging microRNAs, plants and insects. I mean, this is the whole basis of a corn rootworm inhibition in some crops in the US at least. We know that nematodes will take up RNAs from their hosts, and we know now that parasitical plants will take up small RNAs, microRNAs. But that work came out of a collaboration with my group and that of Mike Axtell, who's also at Penn State, and his graduate student at the time, Saima Shahid, who did much of this work. So Mike is an expert in small RNAs. Now he's also a parasitic plant researcher as well because of our collaboration. But so we had this system. We knew that the messenger RNAs moved. And we wanted to know, do the microRNAs also move? And we suspected that they would, because they're known to be systemically mobile in plants. We did the same experiment, basically, as I talked to you about before, harvesting different regions of the interaction. And instead of looking at messenger RNA, we looked for microRNA. So here's some replicates here of the different tissues, which I've already described. And the analysis showed that there was an accumulation of these 22 nucleotide microRNAs in the region of the interface. And so this is the region where the Cascuda and host are together with those hostoria embedded in the host. So hard to separate there. But what you see here on this figure is a blot, a northern blot of the microRNAs. So each of these is representing a different microRNA target that Mike and Saima identified, and they labeled them the microRNAs, and they did the plot. And we find out that they don't really move that far, but they seem to be confined to that region of the interface, but they're very abundant there. And so these are microRNAs encoded by Cascuda that have homology to host RNAs to the point that they should lead to degradation, right? And so that's what we're seeing here. Targets of those microRNAs were interesting, but I want to just, for the sake of time, mention just a couple. The botrytis-induced kinase is a defense-related gene, also shown to move in other systems, plant fungal systems. The most interesting, well, let me get back to that. There's also some auxin-binding protein RNAs that we found. Those are interesting as well. But the most interesting was the C element occlusion-related protein we call SOR1, which makes a protein that sits in the sieve elements of the phloem of a plant. And when there's damage to the phloem, that protein plugs it up like a glue to seal off the wound, keep the plant from bleeding out phloem. So it makes sense to us that this phloem-feeding parasite would want to shut down production of this phloem occluding protein. And so when we found that, we were very interested. So just to show you some more data from this figure, from this paper. So this just shows you that initial degradome that we characterized. And so here what you see is a Cascuda microRNA aligning to a region of host RNA, and there's the appropriate length and number of mismatches. And what you see from the figure is, again, these different regions of the host, the interface, and the Cascuda. And you see this small RNA-seq experiment, this degradome, only shows up in the interface region, and it coordinates specifically with a region of, this is the SOR1 gene here, outlined here, and where the microRNA binds is indicated in the red over here, and you find specific degradation at that point. So it says that this Cascuda-encoded microRNA seems to be capable of causing RNA degradation in that interface region. And to prove that this was functional, we did a couple of experiments here. And this one is to look at the level of gene expression of the target genes. And what you're seeing here then is quantitative gene expression on the y-axis here, and the controls are in the blue. The Cascuda-infested stems are in the red. This last one is a control genes, which was not a target of the Cascuda RNA. Most of the other RNAs that were targeted by the Cascuda microRNA show a decreased abundance in that interface. So again, so showing that there's something going on here. Then we got a hold of Arabidopsis mutant plants that are mutant in the specific genes that we're interested in. The hypothesis here is that if the parasite's trying to shut down these genes, growing the parasite on a plant that's deficient in those genes should allow the parasite to grow better. And that turned out to be the case for a couple of the genes. So here's the wild type, Columbia here. So this is normal, and we're measuring Cascuda growth on the y-axis. So here's normal Cascuda growth. On the SOR1 mutant, the Cascuda grew better, as well as on one of these auxin-binding mutants. Everything else, no signal there. The BIC1, it didn't grow as well, but that's because the BIC1 mutant plant, Arabidopsis plant, was stunted and feeble in growth, so did not grow very well. So that's an artifact of a poor host. The point here is that these this all aligns with our hypothesis of the parasite using those microRNAs as weapons against the host. And now the prediction you would make on that. So Cascuda has a broad host range. You would guess that then if the Cuscuta has this arsenal of microRNA weapons that it wants to use against the host, it needs to be targeting those very carefully. And so this experiment is just assaying a number of different host plants and asking the question of whether those host plants have the specific microRNA target site targeted by the parasite's microRNAs. The GAPDH here is a control, so you find that is not present in most plants. But for the other genes, these are the targets of the parasite, you do find that they have, many of them have the exact target needed that the parasite needs to shut down. And actually, this becomes more interesting in work that has not been published yet. It looks like these microRNA sites are evolving very rapidly, and that they are different parasite species, different parasite, even populations seem to have different combinations of the microRNAs, perhaps not exactly in the same spot, but attacking the same genes or same pathways. So we think that this really is a strategy for the parasite to control the host. And so my conclusion from this section is that the microRNAs are expressed in the hostorial tissues, and they are capable of affecting host gene expression, so seem to be acting here as an effector by the parasite to defeat the host defenses in this case. All right, so the next thing that I want to cover is the messenger RNAs. So the question has always been, and this has been a long-standing issue, I'm presenting this after the microRNAs because it's a little less clear, but the question is, these RNAs, are they just food? Are they consumed by the parasite the same way it consumes sugar? Or do they have potentially some meaning? And I think that, why not both? I think anything the parasite's taking from the host could contain meaning for the parasite. Certainly it can be degraded and used as carbon nitrogen source, but why not both? After all, our food, it not only carries nutrition, it carries meaning, right? You know what's going on by what you're eating. So just to run through some experiments here quickly, let's see, I'm going for a little over half hour. All right, this is an old experiment that we did just to see what is the fate of a host RNA in the parasite. And what you see here, if you take a piece of Cuscuta off of a host plant, it will live for days, and it's capable of forming new attachments during that time. So it doesn't die immediately. So we took Cuscuta off of the host. In this case, it was a tomato plant, and we measured the level of tomato RNAs in there, messenger RNAs. So what you see here in the square is a control endogenous Cuscuta gene, which is expressed over the course of a day. In contrast, the messenger RNA for this proteinase inhibitor gene that came from the host degraded rapidly over about 8 hours and largely disappeared, which tells me that it's being consumed or degraded somehow. So the question then, is it being translated, right? So they are being processed, not being translated. And for this work, we turn to the work of Fritz Kragler from the Max Planck in Germany, who also studies flow-mobile RNAs. And what he did was to generate Arabidopsis plants that have this fusion of a Gus reporter gene. So this is a commonly used reporter gene in plant science. He fused it to a tRNA motif, and he discovered that the tRNA motif was sufficient for conferring mobility on the RNA. So when he did an experiment like this, he made grafts then between Arabidopsis plants. So here's a normal plant, here's a plant expressing the Gus gene. And then if you grafted the Gus shoot on a wild type root, he got a result that looked like this, where a normal root should be clear, but he actually was able to detect Gus activity in that wild type root if it was grafted to a plant. So just demonstrating the ability of the tRNA motif to make this Gus gene to have a mobile RNA, mRNA. So we got those from him, and this is work by Soyan. And so here's the initial system was a 35S Gus. So this is just constitutively controlled Gus gene, or the Gus gene fused to the mobility signal, the tRNA. And so when we then attach Cuscuta and we section these and we stain them, we get the following situation. So here is what it looks like uncolored. So here's the host, looks round here because we're going cross-section of the host and longitudinal to the parasite. And the asterisks indicate the hostoria. And this is the control. So you see the host staining, but no sign of color in the Cuscuta. But where you have the tRNA motif, we see a color there. So this indicates that there may be translation in the parasite. And to do this again, even better, we fused some of those Gus ER constructs or Gus tRNA constructs to the endoplasmic reticulum targeting signal, so that the Gus, not only would it be not mobile, which is typically assumed to be not mobile, but it would be associated with a membrane, so held in place and not likely to diffuse at all. So a very similar experiment. Then we cut it in different sections here, longitudinal and cross sections. This time we separated the host and parasite before we did staining, so that we could not get any cross contamination from movement of the dye. And this just shows you those results, and they confirm what I showed you before. You've got the constructs that lack the tRNA motif. We don't see any signal of Gus, and nor do we see messenger RNA in the parasite. Whereas if we have the Gus expressing plants, we do see the blue indicating Gus in the parasite. And we do find the messenger RNA there. So for us, we think this is good evidence that long distance mobile RNAs from host to parasite can be translated and processed, which really it changes how you think about the RNAs moving. So it made us think that we should look at the proteins. I'll just say a little bit about proteins. Basically, we did the same experiment, only this time we used very sensitive protein detection, and we mapped that based on unique proteins encoded by either the Arabidopsis or the Cuscuta genome. We found nearly 500 Arabidopsis proteins in the parasite, and we found nearly 100 parasite proteins in Arabidopsis. When we cross-reference these with our database of mobile RNAs rather, so from our total mobile proteins, we found that about 2/3 of them, we had evidence that they have mobile RNAs. Some of them don't. And it is, based on what we know about the open connections between host and parasite, it's not unreasonable to think that certain proteins are going to be mobile, right? I already told you that we've known this for a while. But when we look into the specific proteins that we saw moving, the and we broke them down by size in terms of what was likely to move and what was not likely to move. We did, sorry. Here we have the small size range is about the GFP size, which we know is mobile. So a lot of those could be mobile. But when we get into larger proteins, like the size of Gus, these things, and here we have the Arabidopsis proteins moving into Cuscuta, and the other way, Cuscuta proteins moving in Arabidopsis. And we find evidence for very large proteins that seem like they could not possibly move. And so they are, sorry, all right. All right, and the other bit of evidence here for whether things could be mobile or translated is whether these proteins are membrane bound or not. And so for certain Arabidopsis or Cuscuta proteins that are mobile, always a fraction of about 20% are membrane integral proteins. Again, types of things that should not be freely flowing as proteins. So we think, to conclude this section, we think that we're seeing evidence that the translation of mobile RNAs is possible. And in fact, when we look at the proteins, we think that the best explanation for the presence of some of these proteins is that they were translocated as messenger RNAs and then translated to proteins in the recipient organism. And at this point, I want to just say one more quick story, and I think I will, for the sake of time, I will not dwell on this. But this is a protein that we found that we think is mobile from Cuscuta to the host. And it's, we call it GIP23. And the protein was found in one of our initial studies. There's actually two forms of it. And we did a bunch of work. And it's a protein that's found only in Cuscuta, not Arabidopsis. And yet it is found in our experiments. The RNAs are found to be mobile in the parasite. And we found that the assays of yeast-2 hybrid found that it associates with a gene in the jasmonic biosynthesis pathway of plants. And since I think many of you are not plant scientists, so the JA pathway is a defense response pathway in plants. And I think just to get to the bottom line of this story is that this protein encoded by the parasite moves into the plants, and it does impact the ability of the plant to, it does impact the physiology of the host. And we think it functions in defense response. I'm just going to jump to the main conclusion of this, is that this JA pathway controls many factors from flowering to plant defense. And we see these things affected by the GIP23 protein. And so we think this is an evidence that the parasite is manipulating the host through either a mobile RNA or a mobile protein, or both, really. And so my conclusion here, or kind of summary of what I've told you about, is I've got two model cells here of the host and the parasite. And in this diagrammatic figure, they both make messenger RNA, and we have an exchange of messenger RNA. And they also both make protein, and we can have an exchange of protein, but we can also have translation of mobile messenger RNA into protein. So we have a couple different ways for getting protein from one organism to the other. And furthermore, we have microRNAs moving specifically from the parasite into the host that are capable of inhibiting or silencing genes in the host. There's also evidence from other people that silencing constructs can work the other way around. And we're actually exploring this right now in terms of thinking about parasite control using a host-mediated gene silencing mechanism. And I did mention that there was evolutionary exchange of DNA. And so the final point was that this could be a way for parasites to communicate and maybe beyond that. And there was actually just a recent publication in Nature Plants just over a month ago where they found evidence for microRNAs that could be exuded from one plant, taken up by another plant, and actually act as a signal in the environment. And so the big picture here is that this may not be limited to parasitic plants. You may see something like this going on a bigger scale, and it kind of is shocking to think about. So with that, here's my final slide. I hope you take home the simple messages here that some of the world's most destructive weeds are parasitic plants, that the parasitic plants communicate with their hosts on many levels, and that RNAs in the form of microRNAs or messenger RNAs may function in this communication. And with that, I'm done. And I'm happy to take questions.
Thank you, Professor Westwood, for a very fascinating talk about the exchange of miRNAs and mRNAs and between the parasite and the host. We can now take a couple of questions.
Yeah. Hello, Professor Jim. This is a very nice talk and very insightful about the host-parasite interaction at different level. I have actually two questions. One is a rather general question. So basically, and in the first question, I would like to ask, so we have a lot of ecotypes of Arabidopsis. Are there any ecotypes, which is resistant to Cuscuta? And, by studying the genetics, can you get some information, how this plant is resistant to this Cuscuta, and can we, put forward for the agricultural benefit?
Good. Shall I answer this? Yeah, I'll so great question. So Arabidopsis is a wonderful resource, and there are many ecotypes, as you've mentioned, and we have not done the testing of all those ecotypes. I actually did it with the root parasite, the Feliponchi, and didn't find any resistance in Arabidopsis ecotypes. I had a postdoc who looked at a lot of defense mutants in Arabidopsis to try to find the sort of resistance you're talking about. And we found some that have slightly altered resistance. But the bottom line here is there's nothing really that jumps out at you and says, this is the ecotype or this is the gene for resistance. It's not that simple. Okay. So unfortunately, it didn't work out. But.
Okay, yeah. Anyway, so one question, maybe if I misunderstood, but so you said that the Cuscuta RNA, that induced the jasmonate signaling pathway. I mean the jasmonate itself, and then I think if I remember correctly, so the jasmonate also induced the lot of spine, and in the leaf and things. So isn't it like more contrary, like so that if they have like lot of from the outer surface, if you have all the barriers. For instance, Koi1, if you look at this leaf, actually the Koi1 Arabidopsis Koi1 means it's smooth. I think beside all the signaling, jasmonate signaling down, the leaf barrier, the protection against the pathogen is also not there.
Yeah. So this is something we still have to work out, right? And so as you mentioned, JA has many effects on plants. So what we believe is going on is that just in that region of the haustoria, maybe just a few cells away, the parasite is having this main effect. And it probably is not affecting JA signaling systemically throughout the host plant, but probably just enough in the region where the haustorium is embedded in order to manipulate JA-related defenses so that the parasite can live. So I'm imagining this functioning much like the microRNA in a kind of a localized response. The big effects happen when we do our artificial experiments where we constitutively express a gene throughout the plant. Then we get big effects, but I don't think that's how the parasite is working. So it probably would not affect other like leaf phenotypes, for example.
Great. Thank you very much.
Mani?
Thank you.
Yeah. Thank you, Dr. Westwood, for such a fascinating talk. So you talked about movement of RNA between host and parasite. So what I was just wondering about is, are there any changes in the decay rate of these mRNA when these are getting exchanged or moving to a foreign location?
So you asked about any changes in the RNA as it moves, or?
Yes, any changes in the decay rate of the mRNA, because you expect that decay. Yeah.
The decay of the RNA. We just did the one experiment to looking at. We know that the mobile RNA does decay. We have not been able to figure out exactly what is going on with that, or really how we're at the tip of the iceberg. We've not been able to study more than a few of these to see if some decay faster than others, or I guess that there would be differences, but we don't know much more about that than I've already said. It's an interesting question.
Yeah. Thank you, sir.
I have one question. So all the studies are done with Arabidopsis as the host. Do you see the same mRNAs, mRNAs trafficking if you use a different host?
So great question. We've done this in tomato, and we've done other experiments with a few other hosts. We did not find as much RNA exchange in tomato as we did in Arabidopsis, and that probably has to do with the fact that I think Arabidopsis is a little bit unusual in terms of its flow and biology. It tends to exchange things more, right? Plants are different. And so also Arabidopsis is not really a normal host for the Cuscuta. Whereas tomato, there may be some evolutionary history there. And so there may be some barriers, but I expect this to be different from plant to plant. Depending on the type of interaction, you will have different types of RNAs that move.
Great. So if there are no other questions, I thank Professor Westwood for a very fascinating talk, and thanks once again for accepting our invitation. Thank you.
Thank you, everyone.
And now we'll hand it over to the books for the next session.
Thank you, Professor Westwood, for the very interesting talk. And the final plenary talk will be chaired by Professor Satish Raghavan. Professor Satish Raghavan joined this department in 2006. His research group works on understanding the mechanism and regulation of DNA double-strand break repair, generation of chromosomal translocation, and its role in oncogenesis, and evaluation of novel small molecule inhibitors in cancer treatment. I would like to request Professor Satish Raghavan to introduce the speaker of our final plenary talk.
Thank you. Good evening to everyone. Good morning to Professor Maria Jasin as well. Finally, we have come to the last lecture of the Centenary celebration. In fact, it was started about 1 year and 10 months back, February 21, 2020, with a grand inauguration. Unfortunately, our activities were severely affected due to COVID situation, and ever since, of course, it was all through online mode. And of course, everything went well, and it was all in a good way. Today, I'm indeed honored to introduce Professor Maria Jacin, who will be delivering the real final concluding lecture of the centenary celebration of Department of Biochemistry, Indian Institute of Science, Bangalore. Professor Jacin is a biomedical researcher in the Department of Developmental Biology in Memorial Sloan Kettering Cancer Center at New York. Research in her laboratory is focused on the field of DNA repair through homologous recombination, with a particular interest in the role of homologous recombination in maintaining genomic stability. Maria obtained her PhD from MIT with Professor Paul Schimmel, and later she pursued her postdoctoral studies from University of Zurich with Professor Walter Schaffner, and from Stanford University with famous Professor Goldberg. Maria is considered as one of the outstanding researchers in the area of DNA double-strand break repair and genome stability. Some of her major discoveries include how DSBs are repaired within chromosomes, accomplishing the first gene editing experiment from her laboratory, and determining the role for the breast cancer suppressors BRCA1, BRCA2 in homologous recombinations. Maria is well-accomplished scientist. She has elected to National Academy of Sciences, USA in 2015, and the American Academy of Arts and Science in 2017. She was awarded with many prestigious awards, including Bezer Global Prize in 2018 and Shaw Prize in Life Sciences in 2019, to name a few. Today, title of Professor Jacin's talk is Protecting the Genome by Homologous Recombination. Now, I invite Professor Maria Jacin to deliver her plenary lecture. Thank you.
Okay, can you see my screen?
Yes, yes, we can, yeah.
Okay, thank you so much for that lovely introduction. And I had no idea that it was a concluding talk of the centenary celebration. So I feel particularly honored, and thank you again for the gracious invitation. I remember well the meeting at the Institute, I think it's 2015. And
2018.
Oh, 18.
Yeah,
okay,
yeah.
That makes more sense. Yes, that was one of the best conferences I had been to, and it was a lovely time in India. So I'm going to talk about two stories that may seem a bit disjointed, but they're basically related to how homologous recombination protects the genome. And so let me just start with a very simplified homologous recombination pathway. I know many of you are outside the field, and you may have learned about it in school, and it may be sort of a daunting process with all the DNA strands moving around. But I think one can think of it, at least in this context, as a relatively straightforward pathway where you have DNA damage. We're particularly interested in double-strand breaks because they're potent inducers of homologous recombination. There's an end resection step where 3' overhangs are generated in DNA. And this is crucial for the formation of Rad51 filaments. And Rad51, which is a homolog of the E. coli REC-A protein, forms a nucleoprotein filament on single-stranded DNA. And this performs the canonical step of homologous recombination, which is to invade a homologous DNA and form a D-loop. Now, there are many factors involved in these steps, and there are many factors involved with finishing up the Most of the time in somatic cells, we think of the repair as being very simple and straightforward, a small gene conversion event where the sequence in the unbroken molecule is copied in. But then there are also these crossover events that are switching the linkage. And these are drawn in two very close shades of blue to emphasize, at least in somatic cells, these events are primarily between sister chromatids. So you have two identical sequences, and then the identical sister can restore the original sequence in the broken sister. But crossovers are important between homologs in meiosis, which leads me to the first part of my talk. So I'll be talking about a recently published story in the first part, which is mutagenic outcomes at recombination hotspots, and how this will affect the transmission of the genome. And in the second part, I'll talk about a story on the tumor suppressor BRCA2. So the first part, I'll just right away mention the postdoc who did the work, Agnieszka Lukasiewicz, who's on the job market right now. And this is a collaboration with our long-term collaborator, Scott Keeney. So again, this may be something many of you have seen in school, but haven't really thought about much beyond, which is the complicated process of germline development. And this is so important, of course, because how the genome goes through these processes is how the genome will be transmitted to the next generation. So one would think that one would have a very protective system to protect the genome and so to safeguard transmission. But we know there are lots of errors, and these can impact progeny and populations. So I'll focus on spermatogenesis, where there are these diploid spermatogonia that are stem cells, but also undergo rapid divisions. And then meiosis, where the genome is halved to get haploid gametes eventually, and then the complicated process of actually forming a sperm. So the focus on this part of my talk is this meiosis step. And let me again emphasize how important proper treatment of the genome is at this time, because there's a very, very common, surprisingly common number of frequency of mutations and disorders that arise from mistakes at this time. So most of the time the process is very faithful, but there are times when it's not. So part of the issue, perhaps, for why there are problems is that meiosis relies on, surprisingly, the introduction of hundreds of DNA double-strand breaks around the genome. So we usually think of even a single double-strand break as being very problematic, but in this case, there are hundreds of double-strand breaks. And the reason is because, as I mentioned, they're potent inducers of homologous recombination. And during meiosis, this involves recombination between chromosome homologs. And so there are two major roles of recombination at this gross chromosomal level, which is pairing of homologous chromosomes. So these multiple double-strand breaks along a homolog pair ensure that they have interhomolog interactions to pair the homologs. And then the next important process is taking at least one of these homologous recombination events and undergoing a crossover event. And that's critical because it aligns the homologs of the metaphase 1 plate in meiosis 1. So in the absence of recombination, the chromosomes cannot properly segregate. So segregation is the key outcome of meiotic recombination, chromosome segregation. So haploid gametes result that have a correct haploid chromosome complement. So we're interested in this process at many steps, but especially at this very early step of double-strand break formation. And Scott Keeney was the original person to show that SPO11 is the protein that introduces double-strand breaks in yeast. And then we collaborated with him to look at the mammalian, the mouse protein. And so it's a topo2-like protein, and so it forms covalent intermediates with the 5' end of DNA when it cuts. And it's not removed in the standard topo way, which is to reverse the reaction. It's actually released from DNA with small oligonucleotides attached. And this has been very useful because we can sequence these and find out where recombination hotspots are at nucleotide resolution. And from there, the DNA longer range resection occurs and all the other steps of recombination. So here are what those oligonucleotides look like when they're labeled, when they're released with SPO11. And these were sequenced. And so here is one hotspot. So this is double-strand breaks do not occur throughout the genome, but they occur at hotspots. And this is one of those hotspots. And you can see there are two primary nucleotides where double-strand break formation is mapped, although there are smaller peaks that go out about a kb on either side, but primarily are encompassed within about 150 base pairs. So the model has been that although there's not so there are hotspots for recombination, but there's not a single nucleotide usually that's broken, but very preferred bases. And but there is some extension of double-strand breaks beyond that site. So one of the very striking findings from our collaboration was that the ATM kinase in meiosis, so ATM is required for meiosis, and there's a profound defect early in meiosis, which is that there are about 10-fold more of these double-strand breaks than in a wild-type mouse. And so ATM is probably known to many of you that think about DNA repair, double-strand break repair. It's activated by double-strand breaks and serves to phosphorylate a large number of substrates. And what we think is happening here is that ATM is sensing the double-strand break, and then it, in this negative feedback loop, acts to shut down further double-strand break formation. So there's a window of the number of double-strand breaks that's regulated by ATM, and in its absence, there are many, many more breaks. So this is an example of two hotspots that are reasonably close to each other. This is what it looks like in wild type and ATM. You can see that the location of double-strand breaks is fairly comparable in wild type and mutant, but these are normalized values, and you can see this hotspot goes up 9-fold. This hotspot goes up 5-fold in the ATM mutant. So it's interesting that the weaker hotspots actually go up more than the stronger hotspots. And this can be explained by this feedback loop where a stronger hotspot is going to be cut more readily and then be turned off more readily. So what happens to all of these double-strand breaks? There are several possible options. One is that they're not repaired at all. There are too many breaks to repair, but we do know that there's some gamma H2X that is found and evidence of chromosomal breaks. So some are probably not repaired, but many of them actually are. And so we can consider that, well, some of them may be repaired fairly normally, but if there isn't control over the numbers and perhaps the distribution, there can be problems. So for example, if both chromatids are broke at the same time or both homologs are broke at the same time, especially in this case, that could lead to problems in having an intact donor sequence for repair and recombination. And then double-strand breaks may be too close to each other. So we set out to look at this primarily from the standpoint of, do we find evidence for close by double-strand breaks? And would we get DNA fragments released so that these two ends can now join up? And so this is where this would be contrary to the normal meiotic program where double-strand break formation would lead to homologous recombination. But in this case, this would be a non-homologous end joining event that would lead to a deletion. And there's precedent for this in yeast, where the yeast homolog suppresses nearby double-strand breaks. But yeast primarily uses homologous recombination, even in quote somatic cells. And so end joining mechanisms are not that profound. But in mammalian cells, of course, we know that there are multiple end joining mechanisms. So Agnieszka, to address this question, first looked at the hotspot distribution. And what this represents is that each bar is the distance between hotspots, starting out with the bar between 1 and 5 kb. And you can see in wild type cells, about 6% of hotspots are that close to each other. But there are lots of hotspots that are even further apart. Whereas in ATM mutants, the number of hotspots that are close to each other, arising in part at least from the huge increase in double-strand breaks, is about 10%. So Agnieszka looked for hotspot pairs, and this is actually the one I showed you earlier. So these two hotspots are about 2 kb away from each other, 1.8 kb. And so the question was, could we detect deletion events between the two hotspots? And so I'll point out that if there were deletions, it would take out an exon. These two double-strand hotspots, double-strand break hotspots, flank an exon. And so this would be deleterious to the gene, as many deletions around the genome would be. So she performed a simple PCR, and what you can see is that in the presence of ATM, it's hard to see deletions, but in the absence of ATM, these are small pool PCR, she could readily detect deletion events. So this is a summation of those deletion events. So they are focused really around the double-strand break hotspot centers, although they're a little more spread out. So smoothing leads to this, where the breakpoint centers are fairly close to each other, although a little bit to the left or right. So there's quite a good correspondence where these deletions are relative to SPO11 breaks. So here are the numbers. So we can detect low frequency events in the presence of ATM, but they're much more prevalent in the absence of ATM. So how about some numbers? So we think about 1% of ATM mutant spermatocytes carry a deletion in one of these hotspot pairs that are close to each other. But of course, there are many more hotspots that are further away. And some controls, they're infrequent in SPO11, the one, so in the absence of double-strand break formation, this one may be a PCR artifact. It's not in the normal location. And also deletions can be transmitted into sperm. So although infrequent in wild type or heterozygous ATM cells, we can detect them being transmitted. So it's an interesting question then to consider. The way I've kind of drawn things is it indicates that these deletions are coming from two breaks on the same chromatid close to each other. But the other possibility is that they're on adjacent chromatids, and both would give rise to potentially the same deletion product, but the fate of the other sequence would be quite different. So in this case, where there are different chromatids, the deletion would be associated with a duplication. And in this case, with both of them on the same chromatid, there's the potential for release of a DNA fragment and potentially circularization of the fragment. So, Agnieszka performed inverse PCR to identify these events, and in fact was able to detect the inverse PCR product and to try to distinguish between duplications and circles. What she found was that if she digested the DNA with XO5, the number of events decreased substantially. And this suggests that circles, which are resistant to the nuclease, are about half of the total events, and duplications are about half of the other events. So these appear to occur at a relatively similar frequency by subtracting the number of products we get in both cases. So the deletion profiles are actually not identical. The ones that involve duplications often are right at the hotspots, but then they can have breakpoints further in, and you can see this more readily in these smooth curves. And that seemed to be surprising initially, because one would expect that duplication and deletion junctions or circle junctions to basically approximate each other. But we think what's happening instead is that there are actually not just two double cuts, as we call them, but multiple double cuts. And so that would lead to tandem duplications having more of a wider distribution away from the hotspot center. So the next question we asked in terms of two chromatid involvement, can these two chromatids be on homologs, or are they primarily within one homolog? And we used F1 hybrid mice for this, where there are lots of SNPs between the B6 chromosome and the DBA chromosome. And so we could amplify and look on either side. And almost all of the events were either on the B6 chromosome in red or the DBA chromosome in blue. We think DBA is more frequent because it's a hotter hotspot there. But we did get some events that involve non-homologous end joining between two homologs. So you can see those here. It's blue, the deletion, and then red. And then here, an interesting case where it's blue and then red at the hotspot, and then it goes back to red. So we think this might have been an attempt to replicate off the homolog, but then it aborted and went to a deletion. So, the bottom line from this again is that most of the events appear to occur between within one homolog, so between sisters or on the same chromatid. So as I mentioned, there are a range of hotspot distances from each other. And Agnieszka asked, well, let's go a little further apart. She looked at two hotspots that were 1.8 kb away from each other. What about 5 kb, 15 kb, or 20 kb? And she readily could find these events. And even looking at one that's 30 kb away, she could find deletion events. So certainly it doesn't appear that there's a restriction in sizes, but it appears that the further away you are, there is a decrease in frequency. So this summarizes what I told you. Deletions and duplications can be detected at double-strand break hotspots in meiosis, particularly in the absence of ATM, but at lower frequency in the presence of ATM in wild type or het, and they can be detected even at these more hotspot pairs, and most are intrachromosomal, so within one homolog, but they can involve homologous chromosomes. One thing I haven't mentioned yet is that most of the events are deletions between the chromosomes without any additional sequence, but about 10% of them do have insertions at the point of where the deletion is. And this is one of the most interesting types of insertions. So below is the hotspot pair that I've been talking about. And at the point of the deletion, there's actually an insertion of sequences from another chromosome. And this is not a random insertion of a random sequence that's inserted. It's actually from another double-strand break hotspot. And in fact, this is just a map of many of the inserts that we've seen. And you can see how often they occur at hotspots on other chromosomes. And so considering the inserts, so each insert here is blue. Sorry for the color change. And you can see these map right around the hotspot centers of these other hotspots. And even on the, we got insertions from the non-par part of the Y chromosome at positions known to have double-strand breaks. So that was particularly interesting finding for us. And but in a way, it is in keeping with the fact that there can be multiple double-strand breaks near each other, and then they release a DNA fragment, and so I think it's a very interesting question for us. How much extra chromosomal DNA is there in meiosis, and what's the fate of it? And I'll just quickly mention one other class of insertions we had, which is in fact from the same hotspot. So the sequence that was going to be deleted, much of it's deleted, but a part can be reinserted, but it's an inverse orientation. So we have a couple ways of thinking about how these might arise. But all except for one of our insertions were from either other hotspots or the same hotspot. And so it really emphasizes that double-strand breaks can lead to intermediates that can be mutagenic. And then finally, what I focused on was breaks at two hotspots near each other. And Agnieszka looked at a single hotspot to see if these, it's possible that some of these breaks are occurring on the same chromatid. And in fact, she was able to see them. And so we had deletions that fell into actually two size classes, 30 and 60 base pairs, although bigger ones were seen as well. And in wild type, when we saw these deletions, they were, this is normalizing the density. So the peak height here is the same. The 30 base pair ones were seen, but not the 60 base pair ones. So this suggests that there may be some guiding principle to getting breaks about 30 base pairs away from each other on a single chromatid. And that's been proposed actually in some of the yeast biochemistry, suggesting that SPO11 may be tethered to kind of a platform, and then maybe the then access to SPO11 is going to be at certain distances from each other. Oh, and this is just to emphasize how these breakpoints cluster at some of these smaller peaks. So the main hotspot area sequences predominate, but the smaller peaks may be used as well. So I'll conclude this part of the talk, and just to emphasize that germline mutagenesis can occur as a result of SPO11 cutting the genome. And we think these are particularly when at least two double-strand breaks are formed. And the deletions are found at a vastly higher proportion when ATM is deficient and double-strand breaks are increased, really emphasizing how double-strand break control is important in meiosis. So the surprising finding of hundreds of double-strand breaks around the genome, that's a lot, but having many more is really problematic. And yet they do occur, these de novo mutations do occur at a low frequency in normal cells. So they can occur even when double-strand break formation is controlled. Copy number variants then can arise in the genome as a result. We showed both deletions and tandem duplications. And then these released fragments can insert at other sites in the genome undergoing double-strand breaks. And then finally, of course, of quite important interest to us is how relevant is this to humans? And just about the time our paper was published, the Decode group in Iceland published long-range sequencing of Icelanders and identified thousands of, it could have been hundreds of thousands of various copy number variants in the Icelander genomes. And many of these are historical events, so they're seen in multiple people. But a class of these events were actually mapped to hotspots. And so we think this is probably evidence for, and doing some bioinformatics, it appears to be the case, that the events that we're tracking in mouse are actually happening in humans as well. So I'm going to completely switch gears in the last, I think I have 10 or 15 to 15 minutes to talk about what's been a kind of bread and butter in our lab for a long time, which is the tumor suppressor BRCA2. And this work is actually includes a meiosis component, but was really focused on what the role of this domain is in somatic cells. So as many of you are probably aware, homologous recombination deficiency has been associated with many tumor types, breast, ovarian, and other cancers. This is probably a particularly extreme case where about 30% of the tumors in ovarian cancers have mutations, either somatic or germline, in homologous recombination genes. And here's the breakdown in the ovarian cancer case, where most of these are BRCA1 or BRCA2 mutations, but in metastatic prostate cancers, one can also see contribution of homologous recombination gene mutations. And in this case, BRCA2 mutations are the most prominent. So we had demonstrated that BRCA1 and 2 are critical for homologous recombination, and that BRCA1 works upstream, and that's been verified by many subsequent studies showing that BRCA1 controls this end resection step. But today I'll focus on BRCA2, which is important for loading RAD51 onto single-stranded DNA. So it's one of these critical proteins that promote homologous recombination, and in a very direct manner at this step, which I said is so important for homologous strand invasion. And this shows why understanding homologous recombination and homologous recombination deficiency is so important. It's certainly important for understanding the initiation of tumorigenesis, but it's also very important for therapeutics. And these are homologous recombination deficient tumors, tumor cells here. And you can see that they're extremely sensitive to cross-linking agents and to PARP inhibitors, poly-ADP ribose inhibitors. So determining whether a tumor has homologous recombination deficiency is important because it would impact therapeutic approaches. So BRCA2 has been a difficult study, difficult protein to study at the biochemical level because it's very large and it has multiple domains and probably some disordered domains. And the early identification of this middle part of the protein as having several repeats was quite important because it was determined that these were interaction sites for RAD51. So having a direct role in promoting RAD51 function then was pretty evident. But based on this sequence conservation along the protein, Nikola Pavlitich's lab at Sloan Kettering decided to try and get a crystal structure of this segment of BRCA2 to try and understand its function. And the crystal structure was gorgeous. It's shown here. And what was really striking, this was a great case of the structure really informing function, is that there are OB folds that are known to bind single-stranded DNA. So there are 3 of them. And the Pavlitich lab did a co-crystal and could show that oligo-DT bound in exactly the same way as it would in other single-strand DNA binding proteins like RPA that have OB folds. So this suggested to us that a major role for BRCA2's BRCA2 in homologous recombination was to bind RAD51 and bring it to single-stranded DNA. And we tested this in an artificial way by creating this fusion between a BRC repeat, which would bind DNA, and then an RPA subunit, so it would bind single-stranded DNA. And the idea would be, could this peptide, artificial peptide, promote homologous recombination? And so we used our GFP reporter. And in fact, when we put in this artificial construct, we got a very much improved recombination efficiency that depended on its ability to bind RAD51. So our working model then was that the main role of BRCA2 is to bring RAD51 to single-stranded DNA. The important point that I mentioned at the very start to initiate strand invasion. And then we were thrown for a loop in that Alan Ashworth's lab, in studying BRCA2 revertant mutants in a BRCA2 deficient cell line, was able to find peptide reversions of this truncation mutation. And these revertants actually skipped over most of them, not all of them, but here are 3 different alleles that skipped over the DNA binding domain completely. And they were able to lead to PARP inhibitor resistance and to cisplatin resistance. So how can this be the case? And is this just a tumor phenotype, or is this something relevant in normal cells? So that's the second part of the talk. And so to address this question, we use CRISPR-Cas9 in mouse embryos and precisely deleted this domain as shown here. So a deletion of these exons leads to an in-frame fusion, and this is completely deleted for DNA binding. There's just this little segment that starts the DNA binding domain, but doesn't have any function in DNA binding. So a surprise was that the mice were born and viable. So they were born at normal Mendelian ratios and showed no overt phenotype. So They are normal size. Even more surprisingly, they're not tumor predisposed, so at least out to 600 days. And we could not find any sensitivity to mitomycin C, so a cross-linking agent. So they have a similar profile as the heterozygous control. Now, we kind of pushed this a little bit and looked at introducing another mutation, and that's in RAD51 itself. RAD51 heterozygous zygosity is not associated with any phenotypes, but when we cross it into this background, now we find that the mice are hugely sensitive. So although mice are viable and the delta DPD is quite functional in many contexts, it can lead to severe defects when the mice are challenged. And so here, it's not surprising that we're not really seeing anything of a recombination defect, or certainly not much. So recombination is fairly normal in frequency, although we did see a reduction in RAD51 focus formation, and this is in line with if RAD51 is further perturbed, it will create problems. But it's interesting, even if RAD51 focus formation upon ionizing radiation is compromised, it doesn't really affect simple gene conversion events very much. So we looked also at micronucleus formation. So this is a standard readout for genomic instability. And there was really very little indication of increased micronucleus formation in the mutants. So when the DPD was deleted, however, again, RAD51 heterozygosity increased the level, and that it's not significant for it's significant relative to heterozygous animals in both males and females. So again, it's a case of the peptide is certainly functional, and it goes quite far in supporting normal recombination activity. But when the system gets compromised by having the amount of RAD51, then clearly issues start arising. Another surprise was that deletion had no effect on fertility. We could see some small reduction in testis size, but the mice were fertile with no drop off of litter size. And you can see here why that might be the case. RAD51 focus formation is now, in this case, in contrast to the somatic cells, relatively normal. Although interestingly, DMC1 focus formation is somewhat reduced, but apparently this reduction is not compromising fertility. So DMC1 is the meiotic homologue of RAD51. So both are critical during meiosis, but clearly you can drop down the number of DMC1 foci. But if considering this compromised state of DMC1, we asked if we perturb the system by heterozygosity, would we have a greater effect? And in fact, it was like with RAD51 heterozygosity, but even more extreme. Now the testes are extremely small, and there are issues going through meiosis. So So that's the work on the mice that the BRCA2 protein that's deleted from the DNA binding domain gives rise to relatively normal mice, but the mice are kind of at the cusp of genomic stability, because perturbing it by heterozygosity of either RAD51 or DMC1 can create significant issues. So one last analysis of these mice is that, of this mutant, is to look at embryonic stem cells, and we could, BRCA2 is normally essential in embryonic stem cells as well as mice. And we found that, perturbing BRCA2 by deleting the DBD had no effect on cell proliferation or cell cycle profile, although now in this case, we're starting to see a little bit more of a phenotype with homologous recombination. So it's reduced about 40%. And now this seems to make the cells sensitive to DNA damaging agents, including this PARP inhibitor and cross-linking agents, cisplatin. So to summarize this part then, I think I've emphasized the mouse part enough that the mice are pretty normal and surprisingly not cancer prone, at least in this model. But we can sensitize, excuse me. Excuse me. We can sensitize the mice by heterozygosity in RAD51 or its parallel. Excuse me a moment. And in embryonic cells, which divide at a much faster rate, we don't see any problem in cell division time or distribution, but we do see a mild homologous recombination reduction, about 50%, and this leads to sensitivity to agents. So the requirement is for BRCA2 DNA binding can change in cell types, and like other studies we've done, we see more profound impacts within embryonic cells than in somatic cells. So this raises a question: how is BRCA2 getting to DNA damage sites if it cannot bind DNA? We focus a lot on the PALB2 interaction, and PALB2 is another tumor suppressor that links BRCA1 and BRCA2. So I want to thank the people in my lab who have done the work I already mentioned, Agnieszka, but Travis is the one who did the bulk of the work that I showed you on the BRCA2 DNA binding domain, as well as Marina Marcet in Scott Keeney's lab. And I also want to mention Deepika, who's an alumni of your program, and I hope at some point she'll be able to give an alumni talk as well. All right, thank you so much, and I'd be happy to answer any questions.
Thank you very much, Professor Jasin, for the outstanding lecture. I think we have few questions. Sandeep.
Hi, Maria. I really enjoyed your talk. I have a ton of questions, but maybe I'll come back later. So, but my first questions were regarding the first part of your talk, and do you have some idea? So, two questions here really. So, do you have some idea as to how ATM sort of inhibits SPO11? Sorry, go ahead, and then I'll come back with my second question.
Well, that's an easy one. No, it's not obviously a direct phosphorylation, but
it's not okay
because we've done SPO11. We made SPO11 mice, and we've done a lot with SPO11, and obviously it's a catalytic component of everything. We focused on SPO11, but actually there are lots of proteins required for double-strand break formation, and one in particular has a lot of SQ motifs, and there's also the so SPO11 works at hotspots, so there's a protein PRDM9 that defines where these hotspots are, and so there's another level of protein control. So one could imagine a lot of different candidates, or maybe even a set of candidates. So it's a really interesting question, but we haven't had the bandwidth to solve it.
I see. And really quickly, just another related question was the increased sort of deletions that you see. Is that only a consequence of increased breaks, or is it also possible? And here again, I'm kind of ignorant. But is it also possible that it is also because of a role of ATM in sort of downstream events in meiotic recombination, which is now sort of precluded by the ATM deletion?
Well, it's interesting because ATM, as always, is very complicated. And so, as you know, one of the things we were interested in is ATM's role in endosection, like in the context of BRCA1 mutations. And others, of course, have talked about ATM and endosection. And so some breaks are not very well processed, or appear not to be very well processed at all. But other breaks are hyper processed. So how that next step I believe is likely to influence some of these events. So if there's not a lot of processing, that may lead them to be more likely to be joined together. Although we do see that there's some spread out of the junctions, which again could be multiple double-strand breaks, or it could be that there is some end processing. But it's kind of interesting that loss of ATM in these end resection assays in meiosis, we see two defects, a minimal or absent end processing, but also an increase in processing, hyper end processing.
That's interesting. Okay.
Thanks,
all right, thank you.
Yeah. I see biochemistry. Is it Ganesh?
Yeah. Yes, Satish. Yeah. Very nice talk, Professor Maria. So my first question, Sandeep asked. I have two more questions. So my question is that you showed with your first part of your talk that the copy number variation as well as tandem duplication with ATM minus mutant, when you analyze the track lengths and things like that. Have you analyzed the track lengths and then the copy number variations, whatever you observed?
So in doing those distribution maps, we can see some smaller deletions and some larger deletions, and then that's also seen in the tandem duplication. So as I mentioned, you might expect if breaks are right at the hotspot center and they're precisely joined, we'd have about a 1.8 kb deletion. And in fact, the deletion size is not too far from that in most of the maybe 100 base pairs to the outside. But the tandem duplications appear to be different in size because there are more breaks internally, leading to different amounts of duplication.
These are, HR mediated duplications, possibly?
Sorry, what kind of duplications?
These tandem duplication, do you think that these are, HR mediated, possibly?
Oh, it's interesting considering the role of HR, but no, the simple answer would be no, we think these are end joining mediated. So just the opposite part, if you have two breaks, a break on each of two chromatids and you join them together, you have this option of duplicating. And that's going to still be an end joining event. And there are no substantial segments of homology there for the homologous recombination. But you do raise an interesting point, because in the events that we saw with homologs, so those are deletion events, and we would have put them in the case that we saw, at the beginning of being simple deletions, but some of them did involve some copying from the homolog. So that's clearly homology based. That was a perfect copying. So we can identify those events because we have SNPs between homologs, but how often the standard deletions we're looking at involve some homologous recombination, we don't know, but in the end, it has to be joined by end joining.
Okay, my last question is that in the last part of your talk, so you showed that BRCA2 DNA binding deletion, so there is no HR deficiency, so significantly, but you see so great sensitivity with ICL agents as well as with a PARP inhibitor. How do you explain that?
It's a much more compromised situation. So when we so in the ES cells, there's about a 40% reduction in recombination, so it's not a lot, but it's obviously still significant, and it's something. And when you have PARP inhibitors or cisplatin, and you have the large number of lesions around the chromosome, we think that's going to be quite important for the deficiency in 40% of recombination. If half of those lesions are not repaired, then you're going to have sensitivity.
Thank you.
Okay, thank you, Ganesh. There is one question in the chat box. This is from one of our PhD students. She asks, the distribution of recombination hotspots, is it random in genome, or hotspots are more frequent in certain genes?
Hi, Diksha. Thank you for the nice question. Yeah, I didn't have time to go into it, but as I mentioned briefly, hotspots are not random in the genome. They're at sites that are bound by the PRDM9 H3K4 trimethylase. And so these hotspots, so that's a histomark that is often found at gene promoters, but the PRDM9 promoted ones are not at gene promoters. They're away from gene promoters, and like in the cases that in the hotspots I showed, they were in genic regions, but in introns, and there are also a lot of intergenic regions. So there's no, as far as I know, they're not, hotspots are not evenly spread in the genome. There are some regions that have more hotspots than others, and ATM is actually a regulator to some extent in these more hotspot rich versus hotspot cold regions.
Sure. Just one related question. So do you find some sort of secondary DNA structure motifs in those hotspots, or you have not looked at?
Genome-wide, actually, we have not, at least I've never seen any data looking at that. So PRDM9 is a zinc finger protein, and it has 12, well, it varies. It has like about 12 zinc fingers. And so it's basically recognizing DNA through a zinc finger binding domain. And that having so many zinc fingers is what helps to give it, it can go to lots of spots, but not every spot. So it defines the set of hotspots around the genome. But one thing I didn't mention is, I focused on, a normal cell has about 300 double-strand breaks, but there are about 15,000 hotspots around the genome of various hotness, various strength. And so only a subset are going to be used in any one cell, but you have many more hotspots that can be used in other cells. So the recombination pattern between any two cells is going to be completely different because there are so many different hotspots. And then that, of course, will help reassort linkage groups that's beneficial in evolution.
Right. Maybe a last question. So do you find those deletion junctions, what you described in the first part of the talk, they're all purely NHEJ, or do you also find microhomology, at the joining?
I should have left that slide in for you. So, there's no particular microhomology. But if you do the, how does it look compared to random? There's more, but it's not huge. But on the other hand, in one of our hotspots, we have many events, like 10% of the events will be pretty much the same. And that's because there's a microhomology cluster, like 7 base pairs of identity, and then another base pair, and then 5 base pairs of identity. And so in that case, there is some substructure to the genome. So the presence of repeats can impact things. So that can have an effect on outcome.
Sure. Thank you very much. And thank you, Professor Maria Jessin, for accepting our invitation and giving this outstanding, insightful lecture. We are really grateful. Thank you very much. Back to Bix.
With that wonderful talk, we have reached the end of the session and the end of the centenary celebrations. What a marvelous journey it has been, full of enlightening talks and interesting insights into the world of biological research. This program would not be the success that it has turned out to be without the active participation and immense efforts of many people, including the organizers, speakers, students, and the audience. Now, I would like to request Professor Deepankar Nandi, President of BICS, to kindly present the vote of thanks for the event.
Thank you very much, Devalina. It's my great pleasure to deliver the vote of thanks at the end of the BC Centenary activities. It has been indeed a long journey, starting from February 2020, when we had our first physical meeting celebrating the centenary year of the biochemistry department. This was soon followed by the pandemic from March 2020, and all this threw us into a tizzy and disrupted our original plans. However, we had to adjust and revise our plans, and there are several people to thank. First, I would like to thank all the alumni, faculty, postdoctoral fellows, and staff members of the department in helping us with all the organization that have gone into managing the entire activities. Some have contributed articles in the Coffee Table book and in the Reminiscences book, as well as given financial support for the activities of the BC Centenary. I would also like to thank the IISc administration for being supportive, and Professor Rangarajan, the director IISc, for agreeing to release the Reminiscences book on 10th December 2021. Special thanks to Digits for the supply of the Zoom license that has allowed for the talks to be broadcast to the entire world. It has been a great opportunity for us to listen to speakers and broadcast these to an audience covering the entire world. But it has also been challenging to deal with. As seen today, we had to deal with Zoom bombs, and some of us were dealing with it for the first time. I'm happy that the organizers have managed to deal with these initial attacks, and post the initial attacks, we were saved from any further attacks. I would like to thank the previous chair, Professor Jay Bhaskaran, who initiated the activities, and the current chair, Professor Rangarajan, who's been spearheading the Centenary efforts. Several faculty have been closely associated with running the meeting, and special thanks to Raj, Sandeep, and the 4 new faculty known as the Tetrads, Sarvanan, Deb, Kesav, and Mahipal. Together with the students, we have enjoyed these interactions while spending long hours during the meeting. A special shout out needs to go to Professor D.N. Rao, who conceived of the BC Centenary activities and pushed ahead using his contacts with alumni and coordinating with members here in the administration as well as in the department. He spent a lot of time and efforts in guiding these activities. Currently, he's in the Chalakere frame of mind, but we thank him for all his guidance, time, and support. We have had a number of talks, including ones by Nobel laureates during this phase. A big thank you to all the speakers for giving their time and support for the talks. None of these activities would have been possible without the support of the staff and students of the Department of Biochemistry. These activities have been guided by the Biochemical Society. Now, there have been administrative changes, and there are 2 administrations in BIGS to thank for. The first administration consisted of Kshitija, Mani, Swagat, Sheetal, and others who played stellar roles. The current webinar has been organized by Sananda, Sweta, Debraj, Debanjali, Neetu, and others. The new BIGS administration have made the transition really well, and thanks to them for the smooth running. In case I missed anyone, my apologies, but I'd like to thank everyone who has been connected with these activities. I was glad that there were no major hiccups during the meetings, including this one, although as seen today, there have been challenges. All good things need to come to an end, and this is the final program celebrating the centenary activities. I would like to thank you all for being with us until the very end. You are the ultimate supporters. Thank you all. We will now look forward to the next 100 years of the Department of Biochemistry. Thank you very much.
Thank you. Thank you, Professor Nandi.
Bix, can you please take over?
Thank you, Professor Nandi, for your vote of thanks. And thank you to everyone for joining us in the landmark event of the Biochemistry Centennial Celebration. As a scientific community and a department, we are looking forward to carrying on the legacy of 100 years of hard work and aspiration. Good night, everyone. Thank you for this wonderful gathering.